Good morning, sir. Sir, the session will start in once the speaker has joined around 9 o'clock. We'll start, sir. So your mic is on mute, sir. So I'm not able to hear you, sir. Yes, sir.
I'm just checking the video.
Yes, sir. That's why I want to just clarify, sir. Now your audio and video is good, sir.
Okay, sir. Thank you. Thank you. I'll wait.
Yes, sir. Thank you, sir. தேவாலய இப்போ சின்னதா இருக்கும். Good morning, sir.
Good morning.
Good morning, sir. Your audio is clear and your video is clear, sir.
Okay, that's great. And why?
Sir, you can minimize this. Yes, sir. You have already started to share the screen, sir.
Yeah, I want to share the screen.
Sir, you can minimize this and then you can open the PPT, sir. You can minimize
I can. Yeah, your voice is like breaking. Yeah, it will take some time to load. Yeah, now you can see my presentation.
Yes, sir. I can see the presentation, sir.
Okay, great. Okay, let me know when I like to start. Okay.
Yes, sir. Sir, you can stop to share the screen now, sir. We will introduce you. We'll introduce the chairpersons, and the chairpersons will introduce you, sir. After that, you can start it, sir.
I will stop sharing.
Yes, sir. Thank you, sir.
This is Lee Winner, how to stop the sharing.
Sir, it has already stopped, sir.
Okay.
Yes,
That's
sir.
great.
Just 10 more minutes, sir. We will
No,
start
that's
the
okay,
session at
fine.
9.
You just tell me, like, no, I may be not in the picture, but I will, as quick as you tell us to start, I can come, I can do that thing. I am just sitting by the side. Okay?
Okay, sir. Thank you, sir.
Okay, thank you.
Good morning, ma'am. Welcome, ma'am. Ma'am, we will start in 2 minutes, ma'am.
Yeah, good morning.
Good morning.
Fine.
No.
Good morning, all. This is our fourth international e-conference BMSECON 2023, and we are at the final day. And today we are going to start with an excellent presentation on the topic of role of chondroitin sulfate in pathophysiology of COVID-19, induced respiratory failure and lung fibrosis by Dr. Sumit Bhattacharya. For this interesting session, I would like to invite two chairpersons to chair this session. To introduce our first chairperson, I would like to invite Dr. Gopinath, our head and professor of the Department of Physiology of AVMC. Sir, over to you, sir.
Thank you, Dr. Frank. I'd like to invite our first chairperson for this session on third day of BMSECON. It's Dr. Pravati Pal, Professor, Department of Physiology, JIPMER, Puducherry. Madam has obtained her MBBS degree from VSS Medical College, Sambalpur University, Orissa in 1993. She obtained her MD degree from JIPMER, Puducherry in 1998. She had an outstanding academic and research career. Her field of interest are electrophysiology, physiology of nerve and muscle, endocrinology, and neurophysiology. She is the co-author of our textbook of practical physiology for undergraduate students, which is the most popular book followed by the students. She has published more than 135 research papers in national and international journals. Next slide, please. She has been awarded Dr. N. Batmanabhan Memorial Award for the best research work in physiology from the Indian Association of Biomedical Scientists in the year of 1998, and Professor K.P. Puttaraya Award of APPA, Best Teacher in Physiology, and Shiksha Ratna Puraskar Award of India International Friendship Society, IIFAS, New Delhi, and Vaidyanathan Venkata Subba Reddy Medal for her best performance in the postgraduate examination in Pondicherry University. And Madam has many other awards to her credit, and faculty in charge of the Cardiovascular Research Lab and Animal Research Lab in JIPMER, Puducherry. Welcome you, ma'am, for this wonderful event to chair the session. Thank you.
Thank you, sir. Welcome, ma'am. And next, I would like to invite our second chairperson, Dr. R. Venkateshwara Babu, Professor and Head, Department of Respiratory Medicine, Indira Gandhi Medical College and Research Institute, Puducherry. Sir is a renowned person in Pondicherry, and he has obtained his MBBS degree from JIPMER in 2000. And he has obtained his MD degree in TB and Respiratory Medicine from JIPMER, Puducherry again in 2005. And he is a Vice Chairman of NTEP State Task Force Puducherry and Treasurer of Association of Pulmonologists of Puducherry, that is APP. And his work related to COVID-19, he was a nodal officer for Puducherry for clinical case management during COVID-19. He had experience of working in dedicated COVID hospitals of Puducherry throughout the pandemic. I welcome you, sir, for this wonderful session. Now, I would like to invite our own Pravati ma'am to introduce our speaker. Ma'am, over to you, ma'am.
Thank you. First of all, I thank the BMSECON team for this 4th international conference for inviting me to chair this prestigious session. I especially thank Dr. Lakshmi Jatia and Dr. Gopinath. Now I have the pleasure of introducing Dr. Sumit Bhattacharya, research scientist, J.C. Brown Medical Center, Chicago, who obtained his BSc with honors and MSc in Human Physiology from Kolkata University. He obtained his PhD in Medical Sciences from AIIMS, New Delhi, and completed his postdoctoral fellow from Loma Linda University, California, and postdoctoral research associate from Northwestern University, Chicago, USA. He is currently working as Associate Professor Research, Section of General Internal Medicine, Department of Medicine, University of Illinois at Chicago. He has more than 25 years of postdoctoral experience in basic and clinical research development in several renowned universities. He has extensive experience in cell biology, molecular biology, immunology, biochemistry, cancer biology, endocrinology, and metabolism, has more than 15 years of teaching experience, and has 75 research papers in peer-reviewed journals to his credit. He has more than 80 abstracts and presentations in national and international symposium. He is reviewer of several journals, mentors of many undergraduate and graduate students, trainees, judge of many symposia, investigator and co-investigator of many grants, and examiners of many interviews and tests. And he is applicant of 3 US patents. So for this today's session, we have these illustrious speakers. Over to you, sir, Dr. Sumit.
Thank you for such a nice introduction, and good morning to all participants. Let's start with the presentation. Everything is visible now?
Yes, sir.
Okay, thank you. The title of my presentation is Role of Chondroitin Sulfate in Pathophysiology of COVID-19 Induced Respiratory Failure and Lung Fibrosis. Pulmonary fibrosis, one of the major complications post-COVID-19 infection. And this study is addressed to find out the molecular mechanism in which induced this pulmonary fibrosis. In a recent report, it was demonstrated that about 55% of the COVID-19 survivors developed pulmonary fibrosis. If you go in details, these are the CT scan of a same patient A and B before COVID-19 infection, and CD are the basal view after infection, and EF are the coronal view of the after infection. And you can easily find out that at A and B, it's showing the clear normal lungs, whereas in CD, there are septal thickening, consolidation, and parenchymal infiltration, bronchitis is everything, which suggests the development of pulmonary fibrosis. If you go further, the lung biopsy of COVID-19 lungs, you can see that diffused alveolar damage, which is known as DAD. You can see the deposition of fibrin and hemorrhage. You can see that there is a premature fibroblast and honeycomb-like remodeling. To go further, this group demonstrated that the heparin sulfate content and chondroitin sulfate content in the BAL, that is the bronchial lavage fluid, is increased after COPD. And the content of chondroitin sulfate, it was negatively correlated with the forced expiratory volume, which is one of the basic parameter of lung function. Very recently, it was also found the CST15, one of the carbohydrate sulfotransferases, is highly involved to bring the signal for pulmonary fibrosis. It's induced the expression of chondroitin sulfate E, and it activates the expression of several molecule which is known as profibroblastic. In this context, we started our work. So before directly go to our work, let's see what is chondroitin sulfate and how it is synthesized in our body. Chondroitin sulfate is, the chondroitin part is basically synthesized from glucuronic acid and galactosamine, and in the presence of chondroitin synthase enzyme. And then it is sulfated by several sulfotransferases enzyme, which is CST11, CST15, and several others. In this sulfotransferase reaction, the donor of the sulfate group is a molecule which is known as PAPS. And the PAPS is synthesized by two enzyme, which is known as PAPSS1 and 2. So basically, chondroitin synthase, PPAS1 and 2, and the sulfotransferases are the key regulatory enzyme of chondroitin sulfate biosynthesis. So after chondroitin sulfate is biosynthesized, it is linked to the its core protein to form chondroitin sulfate proteoglycans like Neurocan, Versacan, and aggrecan, etc. and etc. So from this idea, we thought if there is an accumulation of chondroitin sulfate in the COVID-19 infected lungs, there is some changes in the chondroitin synthesis. Either it is increased due to increased activity and expression of the enzyme like chondroitin synthase, PAPSS1 or 2, or increased in the activity and expression of sulfotransferases enzyme like CST11 and CST15, or there is a decrease in degradation of chondroitin sulfate by specific sulfatase, such as aryl sulfatase B. With this hypothesis in mind, what we did, we collected the lung tissue from several patients suffered from COVID-19 infection and stained them immunocytochemically with chondroitin sulfate, aryl sulfatase B, and CST15, which is a sulfotransferase. And what we found is both chondroitin sulfate and CST15 is increased significantly in comparison to the normal lungs, whereas aryl sulfatase B staining is significantly less with comparison to the normal lungs. If you go to the vascular smooth muscle cells of the lungs, we also found the CST15 expression is increased, which is suggested by the more intense staining. If you go further, you can see here the chondroitin sulfate immunostaining in the normal lungs. This is the early stage of COVID-19 infection. This is mid stage, and this is the very terminal stage. So the chondroitin sulfate staining is increased as the disease progressed. On the contrary, if you see the aryl sulfatase B staining, we can see that a lot of staining in the normal lungs, which is decreased when there is COVID-19 infection, and it is progressed with the progression of the disease. And in the final stage, there is hardly any aryl sulfatase B staining. With this observation, we thought that, okay, that it's fine that chondroitin sulfate is increased, sulfotransferases are increased in the chondroitin sulfate, regulating enzymes are increased probably. So what are the molecular mechanism by which each COVID-19 infection and increasing the chondroitin sulfate biosynthesis as well as decreasing the its degradation? To address this question, what we did, we cultured normal human small alveolar epithelial cells and treated them with a synthetic protein, which is actually the receptor binding domain of the SARS-CoV virus. In some cases, we treated the cells with interferon beta before the SARS-CoV spike protein in treatment, which is onwards I will call SPRBD, which stands for SARS protein receptor binding domain. And we treated the cells before treatment with SPRBD with interferon beta. And later we'll see what this interferon beta role in this process is actually increases one molecule, that is ACE2, that is angiotensin converting enzyme 2, which is the main carrier protein for SPBRD, which facilitate the entry of this protein inside the cells. So after the treatment, we traced out several signaling mechanisms by which the sulfotransferase expressions are increased and the sulfatase expressions are decreased. we used inhibitor specific for several signaling pathway like SB203580 for P38 MAP kinase. We did it cis3 inhibitor for SMAD3 and so on. Now what we found? We found that treatment of the cells with SPRBD significantly increased the content of ACE-GAG, that is sulfated GAGs, and chondroitin sulfate in the lungs or the lung cells. And this increase is further pronounced when the cells are pretreated with the interferon beta. Moreover, when we examine the expression of three important enzymes related to chondroitin biosynthesis, that is PAPSS1, PAPSS2, and CSY, we found the SPRBD induced the expression of this enzyme, and that induction is stronger when there is pretreatment of the cells with interferon beta. Coming to the sulfotransferases, we found the similar trend. SPRBD induced the expression of CST15 and 11, and that induction is stronger when the cells are treated with interferon beta. We also measured the sulfotransferase activity, which shows the similar effect. The SPRBD induced the sulfotransferase activity in presence and absence of interferon beta. Then we measured the ARSB activity and expression in such a count. And we found on the SPBRD actually decreased the aryl sulfatase B in the cells, and that inhibition is stronger when it is pretreated with the interferon beta. ARSB expression follows the same trend that SPRBD reduced its expression, and it is much stronger when there is a pre-exposure to interferon beta. So, in this point, we found that two of the sulfotransferases and some enzyme of the chondroitin biosynthesis is increased in expression and activity, and the sulfatase expression and activity is decreased. But how it is happening? So we try to find out the cellular mechanism or signaling pathway responsible for these changes. So, but before that, we try to see one thing, that what interferon beta is doing. Why the pretreatment with the interferon beta actually potentiating the effect. So what we did, we checked the AC2 expression when the cells were treated with SPRBD alone or in presence of pre-exposure to interferon beta. And we found interferon beta actually inducing the expression of ACE2 protein but SPBRD neither alone nor along with interferon beta has any independent effect. So this pre-exposure to interferon beta actually increasing the carrier protein, which further facilitating the entry of SPRBD inside the cells. To find out the cellular signaling, what we did, we first tried to find out one major signaling molecule in the cells, that is P38 MAP kinase, and we try to determine the phosphorylation or activation of P38 MAP kinase after the treatment. And what we found is the SPRBD actually inducing the phosphorylation of the P38, and that induction is stronger when there is interferon beta pre-exposure. And specific inhibitor to P38, that is SB203508, it totally abrogated the response, whereas SMAD3 inhibitor, that is CYS3, has not any significant effect. We also tried another specific inhibitor to P38 MAP kinase. This is more specific to alpha P38 MAP kinase, that is known as P8797-804, and it shows that it also totally abrogated the SPRBD induced phosphorylation of P38. But when we treated the cells without ACE2 expression, that is by doing silencing or knocking down ACE2 expression with the specific silencing RNA. So we also found that as ACE2 is not there, and there is less entry of SPBRD, so the inducing effect is really compromised. One thing is important in this slide, that SMAD3 inhibitor has no effect on the phosphorylation of SPRBD, whereas the P38 inhibitor has a significantly reduced the effect. Next, what we did, we tried to measure SMAD3 phosphorylation, which is, you all know that SMAD3 is one of the transcription factor, and it's very important in the cellular mechanism. So we found that SPBRD inducing the phosphorylation of the SMAD3, and it is totally nullified in presence of CYS3, which is a specific inhibitor to SMAD3, as well as it is inhibited by the P38 inhibitor ASP. So from this slide, right, we've hypothesized possibly that phosphorylation of P38 brings the phosphorylation of SMAD3, because as when we used CYS3 in the previous slide, right, we didn't see any change in the phosphorylation of the P38. But when we used here CYS3, this is and PSB, so we found there is a significant decrease in the effect. Next, we went to measure for promoter activity of the two important sulfotransferases. They are CST15 and CST11, and we found that SPBRD and a combination of interferon and SPBRD both induced the promoter activity for CST11 and CST15, and which is totally abrogated in presence of the SMAD3 inhibitor CYS and P38 inhibitor SB. Finally, we try to measure P38 and SMAD3 inhibitors, how they act on the expression of CST15 and CST11. We found that SPBRD, as I told before, induced the expression of CST15 and CST11, and either in presence or in absence of interferon beta, and the specific inhibitor to P38, it actually totally antagonized the effect, and it's nullified the effect also. Whereas RORAC inhibitor NSC is not showing any significant effect. So it is mediated through the P38 MAP kinase inhibitor. When we did the similar experiment with the SMAD3 inhibitor, we found the similar result, which suggests there is a pathway involved, the phosphorylation of P38, as well as phosphorylation of the SMAD3. We used another inhibitor, which is P8797-804. told before that this is a specific inhibitor to a specific subtype of the p38 MAP kinase, that is alpha p38, and that shows the similar effect with as ASB, the pan inhibitor of the p38 show. So possibly the p38 MAP kinase involved in this pathway is the alpha p38. To summarize what I told till now, we may say that interferon beta actually increased the ACE2 expression, and by binding with the ACE2 expression, the virus enter inside the cells, and this SARS protein then induce the phosphorylation of p38, and this phosphorylation of p38, it actually induce the phosphorylation of the SMAD3, and this SMAD3 then act on the DNA to induce the CST15 and CST11 promoter, and finally the expression of CST15 and CST11. So we have now some idea how the SARS protein induced the expression of sulfotransferases and how it can increase the chondroitin sulfate biosynthesis. But we don't have any idea of one of our previous finding, how the sulfatase activity is decreased, which is a decrease in degradation of the chondroitin sulfate. We tried several things, but we failed to get any positive result. Finally, we find out that if we treat the SP cells with SPRBD, it actually reduced the C-terminal phosphorylation of a very important protein of the cells, which is known as retinoblastoma protein. So SPRBD alone or in combination with the interferon beta actually reduce the C-terminal phosphorylation of the SPRBD, and that effect is actually totally abrogated when we treated the cells with the p38 MAP kinase inhibitor, that is SB, here and here. But that has no effect when we treated the cells with its MAD3 inhibitor, that is CYS3, which suggests that this pathway may not be involved or SMAD3. Furthermore, when we try to see the phosphorylation of N-terminal of the retinoblastoma protein, we are surprised to see that SARS protein treatment actually increase is the N-terminal phosphorylation of the retinoblastoma protein, and which is also abrogated in presence of the specific inhibitor. So here what we found that SPBRD treatment and causes is reduction in the C-terminal phosphorylation of RB protein and increase the N-terminal phosphorylation of RB protein. Now, what does that mean? To explain that, let me say something about these molecules. Normally, what happens? There is a very important transcription factor known as E2F. This E2F is mostly regulated to the cell signaling and regulation of several other related genes. But normally, this E2F, if in the cells, remain in the inactive stage, and it's in a bound form with the retinoblastoma protein. So retinoblastoma protein actually sequesters the E2F by binding with it. When there is a stimulation from cell signaling molecules like the CDK4, cyclin D, or this thing, there is C-terminal phosphorylation and of the retinoblastoma protein. And this C-terminal phosphorylation of the retinoblastoma protein actually causes conformational changes, which release the free and active form of E2F in the cytoplasm, which then go to nucleus and bind with the DNA and there and regulate several gene expression. But if there is a phosphorylation of MAP kinase P38, that also causes the N-terminal phosphorylation of the retinoblastoma protein. This N-terminal phosphorylation actually predominates, which causes is what? It reduces the phosphorylation of the C-terminal phosphorylation of retinoblastoma protein, and it actually inhibits the release of free E2F in the cytoplasm. So, if there is a N-terminal phosphorylation of retinoblastoma protein, there will be less PE2F in the cytoplasm, and less will go and bind with the DNA, and less will be its physiological function. To test our hypothesis, what we did, we tried to find out the binding of E2F with the DNA by a oligonucleotide binding assay. And we found that SPBRD actually significantly reduced the DNA binding of the E2F, and that effect is stronger when in more SPBRD is entering into the cells due to the pretreatment with the interferon beta. And that effect is also nullified by the specific inhibitor to the P38 MAP kinase. So that suggests actually P38 phosphorylation may be one of the major cause of this E2F binding reduction. To prove this is observation furthermore, what we did a chip assay. This chip assay is designed and for the basically the ARSB promoter. And we found that that SPBRD actually reduced the binding of the E2F with the ARSB promoter, and that is totally counteracted by the inhibitor of the P38 MAP kinase. So, from this study, we concluded that E2F binding is reduced to DNA, as well as E2F binding is reduced in the ARSB promoter. Then what effect you will find in the ARSB promoter? So we checked the ARSB promoter activity. We found that ARSB promoter activity is reduced in presence of SPRBD, and that reduction or inhibition is further stronger when there is a pre-exposure to interferon beta. And that inhibition is totally dependent upon the phosphorylation of P38, and then because that specific inhibitor is actually totally nullified that inhibitory effect. Finally, we checked the ARSB expression and in presence of this inhibitor, and we found that SPBRD reduced the ARSB expression, and that reduction is counteracted by the when we inhibit the P38 inhibitor SB, but not affected by the RORAC inhibitor or NSC. We also tried the specific inhibitor for alpha P38 MAP kinase, and we found the similar effect as the PAN inhibitor. And finally, we tried the inhibitor of SMAD3, but that SMAD3 inhibitor has no effect. SPBRD reduced the ASB expression, and SPBRD plus CIS3 has no significant effect, which suggests this pathway possibly don't involve like the other half of the phosphosmad. It's basically probably involved the transcription factor E2F. To summarize is that what we can say, when there is P38 phosphorylation, the N-terminal phosphorylation of RB is increased and C-terminal phosphorylation of RB is decreased, and release of active E2F is decreased, and finally less E2F go to the nucleus and less E2F bind to the ARSB promoter, and there is less ARSB expression. Now, we also tried some of the chemicals or drug which is used in the earlier phase of the COVID-19 infection for or maintaining the patient. Like one of them are desiloratidine, which is an antihistaminic agent. And what we found, the desiloratidine actually in a very high dose was partially recover or partially counteract the action of SPBRD. If you see this SPBRD, this phosphorylation is increased, whereas was when we use the desiloratidine, it's recovered not fully but partially recovered, and similar trend is observed when there is pre-treatment with interferon beta. We try to find out similar effect on CST15 and CST11, and we found that desiloratidine also do the same. It's partially help the cells to recover the expression of CST15 and CST11, and SPBRD increase the expression, and when we treat it simultaneously with desiloratidine, it's trying to bring it down, but it cannot reach the control level. It's partial on only. And it is doing the same thing for ARSB expression, and normally SPBRD reduced ARSB expression, and the desiloratidine only partially recover it towards the control level, not fully. So as it's affecting in both the chondroitin sulfotransferases as well as ARSB, it is probably acting at the level of P38 phosphorylation. Next, we tried another medicine, which is monansin, which is also used in the earlier phase of the COVID-19 epidemic, and which is a polyether antibiotic, right? And we found this also reduced the expression of CST15 and 11 when it is induced by SPBRD. SPBRD induced the expression here, and monansin in bring it down on, and when there is interferon beta pre-exposure, or it's dose dependently reduced, it's effective. But it has no effect on aryl sulfatase B expression, and SPBRD decrease ARSB expression, and whereas that monansin has no significant effect. There is no significant difference between these two. So that suggests that it may act at the level of SMAD3, so it can affect the expression of CST15 and CST11, but cannot counter the effect of aryl sulfatase B. So here, if I summarize, that SPBRD treatment of the lung alveolar cells increase the expression of CST15 and CST11, which are the two major sulfotransferases, as well as it's already I shown that it increased the chondroitin biosynthetic enzyme. So increased is the chondroitin sulfate biosynthesis in total, whereas it's decreased the degradation of chondroitin sulfate by reducing the expression of aryl sulfatase B, which also help to build up chondroitin sulfate within the lungs. So this buildup in the chondroitin sulfate actually being one of the primary criteria for the lung fibrosis. Now, another major thing during COVID-19 infection is a systemic inflammation. So all our data, what I shown you either from the human sample or from a in vitro study. So we try to do something similar or in the animal model. But in the animal model, you want to do work with SPBRT, it's very technically difficulties are there. There is a lot of restriction and other things. So what we did we use another systemic inflammatory agent, which is known as carrageenan, and we fed the animal for 6 weeks with carrageenan and see its effect on P38. It shows that this systemic inflammator increased the phosphorylation of P38 as well as it decreased the DNA binding with the E2F, and it is increased the expression of CST15 and CST11, and as well as decrease the expression of aryl sulfatase B. So to summarize that, in the SPBRD treatment actually upregulate the sulfotransferase enzymes and chondroitin sulfate biosynthetic enzymes and decrease the sulfatase enzymes. And there, by that, there is a building up of the chondroitin sulfate of in the lungs, which may be one of the major point and to bring pulmonary fibrosis as a post-COVID-19 infection. Last but not the least, I should thank my mentor and collaborator, Dr. Tobacman, and our Swedish collaborator, Alexander Ekonov, who actually provided us with the COVID-19 samples from there and others. And thank you all for patiently hearing my talk. So I am open for question if you have any.
Thank you, sir. Thank you for the wonderful presentation, sir. Now over to the chairpersons for discussion. Ma'am, you can start with it.
Thank you, sir, for this elaborate cellular and molecular pathways that you have investigated of COVID-19 lung fibrosis. We knew that diabetic patients had more, they were more prone to COVID. People with some immunological disorder, they also were more prone to COVID. But these pathways that you have investigated, that is interferon beta and role of chondroitin sulfate in further molecular mechanisms, I understand and I hope that this will open up for further drugs, that discovery of new drugs, which can directly act on these specific ligands or specific substances that are involved in the pathogenesis of lung fibrosis. So I will just like to ask one clarification, because we work a lot on diabetes, and my question will be, because this chondroitin sulfate having direct link derived from glucose. So
I understand that. Yeah, please go on.
Are there studies specifically differentiating diabetic population and other people who had lung fibrosis, and what is the chondroitin sulfate level and how it has impacted this lung fibrosis?
Yeah, actually, previously we shown that inflammation, it's published, I think, 6-7 years ago in the Journal of Diabetologica, that in the diabetes patient, and if you treat the patients in with a diet which is without an additive, food additive, which is, I just showed, it causes lot of systemic inflammation, used a lot of in our food as a stabilizer, and all other things, that is carrageenan, which is used in our food as additive. If you provide pre-diabetic patients with this carrageenan, if you can see that their HB1AC and other things are improving, and they are much better than and before. Why I bring that point, I will tell you that in those patients, if we also measured the chondroitin sulfate and aryl sulfatase B, and aryl sulfatase B is lowered over there, and actually aryl sulfatase B lowered, and chondroitin sulfate, and actually basically chondroitin 4 sulfate increased in these patients. And this it is a human trial. It's a small pilot experiment, about 30 people. We did it for each group, and we found a significant difference. If we don't use the food additive containing food and other food, normal food, which is inducing those things, you will get a better result apart from drug. Okay. And I just like to add few things, like as you told about diabetes, even the smoking increase interferon beta, which is one of the major concern related to COVID-19, that is reported by several other groups. Okay, and there is more like evidence for this pathway, is that it's through the angiotensin in converting enzyme 2. So it's also related to blood pressure regulation. Like you all know that hypertensive patients are more vulnerable to COVID-19. So it's, to me, it's not a single pathway, but what we try to find out is one of the pathway. It's a multifactorial things. It may interact one to other in any place, but this is one of the major thing, because this is ultimately induced you lung fibrosis. So even you survive from COVID, you may face those problems. That is the first report I shown you, that about after release from the hospital, 45 of the patient here, they showed the significant changes in the lungs with the lung fibrotic changes. So that is the importance of what we found it over here.
Thank you, sir.
So thanks for the wonderful study and wonderful presentation, sir. I have a query, sir.
Yeah,
We
sure.
have
Please
started.
go.
We have started seeing COVID cases again in Pondicherry. 2 days back, we had a COVID pneumonia. So, with regard to your study, at what stage can we intervene, or any medication is we can try at present to decrease the development of COVID fibrosis?
I am not a clinical person. I cannot tell you what I showed it to you. That is deslorudine or monensin. They don't have a total effect. The deslorudine I used as a clinical person, you probably understood, better than me. That's a very high dose. So, yes, and that is the effect is also partial, and you cannot extrapolate all of the in vitro study in the in vivo situation, because it's a multifactorial, as I told. So right now I cannot suggest anything about the clinical thing, but one of the thing I know, it's that in even COPD, even in the other lung disease, even in the cystic fibrosis, if you measure the pleural fluid or the bowel, you will see there is thickening, and that thickening is the major cause, is the increase in chondroitin sulfate and heparin sulfate content. So that much I can tell you clinically. My knowledge is limited about other things.
Yes, sir. Maybe hopefully in the near future we will have some drugs, sir. So
Yeah, the inhibitors I use you cannot use systematically because they are toxic. Like the P38 inhibitor and all this. So people has to come out with some inhibitor which is not toxic and can be used clinically.
yes, sir. I totally agree, sir. Thank you, sir. Thank you.
Yeah.
Just one more query, sir. I hope
Sure,
that
sure.
will be related to this matrix metalloproteinases,
Yeah.
which will be directly linked to this chondroitin sulfate. So in long fibrosis, the level of matrix metalloproteinases and chondroitin sulfate.
Yeah, they are involved, especially MMP2 and MMP9. When one of your faculty member, Dr. Shushmita, asked me to present over here, I asked her that what type of presentation you need. I actually right now I'm working on two field. This is on COVID-19. Another is melanoma. I work on melanoma, and in the melanoma system, recently one of our 2023, I think in August or September, we have a publication in BBA. But okay, and you can find out that in melanoma cells, there is a reduction of aryl sulfatase B and an increase in chondroitin sulfate, which actually upregulate the expression and of um, uh, the metalloproteases, especially MMP2 and MMP9. Basically, you may say that, oh, you are involving everything with chondroitin sulfate. It's not that. Chondroitin sulfate and heparin sulfate, they are two major components of our cells. So if there is any cellular changes in the cell membrane, that signal has to go. That can go to several pathway, and that may be cause of one thing to other. In which disease, what is predominant, we don't know exactly. Cell culture system can allow you to study one-to-one, not like a in vivo system. So it is changing maybe in the cell culture system, but it may be not that effective in the in vivo system because other pathway will take care of that. But definitely there is a correlation between chondroitin sulfate and MMP2 in this development. Yes.
Okay.
Thank you.
Okay, thank you, sir. Thanks for a wonderful presentation and an eye-opening presentation to us about all the aryl sulfatase and chondroitin sulfates. And I hope everybody has clarity on that. Now, we would like to, for this wonderful presentation, sir, we know it's too late for your time. It's 10:23 at Chicago, and thank you so much for bearing with us for this time. We would like you to appreciate with an e-certificate and a virtual memento, sir.
Thank you. Thank you very much. But before that, I like to thank your organization. Thank you. Thank you very much to share my research finding with you. So I am really thankful for this your international meeting, and I'll hope to join it personally someday.
Definitely, sir. Definitely. We are looking forward to it, sir. Thank you, sir. And we'll also like to appreciate our chairpersons, Dr. Pravati Pal ma'am, with an e-certificate and a virtual memento. And our next chairperson, Dr. Venkateshwara Babu. Thank you, sir.
Thank you.
Thank you so much. Thank you so much, guys. That's the end of our session. Let's all try to meet up in the valedictory, which will happen today evening at 4:30. Thank you.
Thank you. Bye.
Thank you, ma'am. Thank you, sir.
Thank you.
Dr. Suresh sir.
Hello,
Sir, shall we start, sir?
am I audible?
Yes, sir, you are audible.
Oh, okay, okay. We can start.
Yes, thanks. Good morning, all. We'll continue with the next scientific talk on the topic of study of phosphatase estradiol receptor and non-coding RNA interaction in breast cancer. For today's scientific session, we'd like to invite two chairpersons with us. First, I would like to invite Dr. Deepthi Shastri ma'am, Deputy Dean, Vinayaka Mission Kripananda Warrior Medical College and Hospital, Salem. Madam has obtained her MBBS degree in 1990, Bombay University, and MS degree in Goa University. Madam has more than 30 years of teaching and research experience. Madam is a member board of management, VMRF, academic coordinator, VMRF. Madam is the PhD supervisor, editor-in-chief in National Journal of Medical Science. Madam is the editorial board in National Journal of Clinical Anatomist. She is a president in Association of Anatomists, Tamil Nadu, Puducherry. Madam has more than 40 publications in national and international journals, indexed journal. I welcome you, ma'am.
Thank you. I just want to make a small correction. Many of them are former editors, okay? Different
Okay.
periods of time. Thank you for the introduction.
Thank you, ma'am.
Yeah. At the outset, I would like to, are you going to introduce Dr.
Yes,
Vidya?
ma'am. Yes,
Yeah,
ma'am.
okay.
Yeah. We'll also welcome our next chairperson, Dr. Vidya CS, Professor and Head, Department of Anatomy, JSS Medical College, JSS Academy of Higher Education and Research, Mysuru. Madam has more than 20 years of teaching and research experience. She has completed certificate course on e-learning and prepared a digitalized histology CD called Histology Learning Made Easy for MBBS student. Madam is a Vice President of Karnataka Chapter of Anatomists, Executive Committee member of Society of Clinical Anatomists, editorial member of Karnataka Chapter of Anatomy Journal, and Madam is a life member of Karnataka Chapter of Anatomists, SOCA, ASI, and South Indian Medical Legal Association. Madam also a reviewer of National Indexed Journal. I welcome you, ma'am.
Thank you, Doctor. So I request Dr. Deepthi Shastri ma'am to introduce today's speaker, Dr. Suresh.
Thank you. At the outset, I would like to thank the organizing chairperson of BMSECON, Dr. S. Silan Kadir, Professor and Head Department of Anatomy at AVMC, for giving me an opportunity to introduce such a distinguished speaker of this session, Dr. Suresh PS. I deem it a privilege to introduce the illustrious Dr. Suresh PS to the audience. Dr. Suresh is an associate professor at the School of Biotechnology, National Institute of Technology, Calicut. He has completed his UG from Christian Medical College and Hospital, Vellore, and PG from JIPMA. He was the best outgoing student and the university topper in both the degrees and was awarded Dr. Ramani Pulimur and Satyabhama Murli Rangarajan gold medals, respectively. He worked as a postdoctoral research scientist in the Department of Pharmacology and Toxicology at the Medical College of Wisconsin, USA. He is a member of various international science societies. He has been an invited speaker and has delivered talks nationally and internationally at the Max Planck Institute of Molecular Biomedicine, Germany, the University of Helsinki, Finland, Tokushima University, Japan, Medical College of Wisconsin, USA, the Ohio State University, and the Wexner Cancer Center, USA. He has published 56 peer-reviewed research articles in various internationally reputed journals. He has received awards from the Japan Science Society, Japan, the Endocrine Society, USA, and has received fellowships and honors and serves as an editorial board member in 8 journals, related to his research. We are delighted to have Dr. Suresh deliver his talk on the topic Study of Phosphatase Estradiol Receptor and Non-Coding RNA Interaction in Breast Cancer. The screen is all yours, sir.
Okay, thank you, ma'am, for the kind introduction. And am I audible? Clear enough?
Yes, sir.
Okay. Thank you very much for the kind introduction, ma'am. And also, I thank the organizers for giving me this wonderful opportunity. So give me a minute. Let me share my slides. Okay. Is it in the screen? Is it the full screen show, ma'am?
Yes, sir.
Yes.
Okay. So I can continue. Okay. So once again, thank you, organizers, for giving me this wonderful opportunity for delivering my talk on the topic, the non-coding RNA and phosphatase interaction in breast cancer. So before I start, let me introduce our institute to the audience. Our institute is a National Institute of Higher Importance, and it has been ranked 23rd in the NIR of Engineering 2023. It is also nationally second rank in architecture, and also in innovation, it is ranked 8th. So this is the introduction about our institute where I work, and little bit just showing some glimpses of our lab activities before I proceed to the topic. So our lab is a molecular oncology lab. We basically work on 3 cancers: endometrial, breast cancer, and ovarian cancer, mostly gynecological tumors. And the areas of research, we work on non-coding RNAs, epigenetics, signal transduction, and drug delivery system for cancer. And the last 5 years, we have got several grants that supported our research from BST, DBT, iSTEM, UGC, FIST, and SAP. And with this help, we could do some research in our lab, meeting all these objectives. And today I'm going to share some studies which we carried out in breast cancer. And this audience, I don't think I have to introduce the burden and the disease of, breast cancer. And I'm just going to go through swiftly. The Globocon 2020 shows the breast cancer incidence is this much percentage. You can see that pie chart. And there are many subtypes of breast cancer based on histological aspects and also based on the hormone receptors. You classify pathologically different kinds of breast cancers. And various risk factors are there for breast cancer, like family history, gene mutation, late childbearing, early menstruation, late menopause, obesity. And obesity is one of the important factor that we also try to address how obesity causes endometrial cancer. I think that I presented in one of the meeting here. But there are various risk factors as shown in the picture. And also the diagnostic tools involves the history, age, examination, mammography, ultrasound, FNAC, and other things. And treatment strategies varies based on the condition and the surgery, chemotherapy, radiation, and targeted therapy. And what our lab is mostly interested in, the molecular markers for the breast cancer. So can we find a molecular marker for early diagnosis and also which can serve as a marker for prognosis? That's what our lab aims to look at as a research objective in our laboratory. Now, a little bit closer details about this, little bit numbers. If you look at the numbers, the breast cancer people, you can see it's almost like a second highest level. So first highest level in the estimated new cases, but estimated deaths is the second high to the different kinds of cancers. So we can understand the burden of the disease, but the national statistics if you look at the Indian statistics, of course, this has been picked from the source, which is shown below. And you can see in 2018, I was trying to look at the data, recent data, but it was not clear to me. And also, they have a subdivided into urban and rural. But that's why I quote this reference for 2018 statistics about new cases. 162,468 new cases have been registered, and 87090 deaths have been recorded, and it's the second highest in the world in that year, to indicate the burden of the disease in India. And also 1 in 28 women is likely to develop breast cancer during her lifetime. And in urban areas, 1 in 22 women is likely to develop breast cancer during her lifetime, as compared to rural areas where 1 in 60 women develops breast cancer. So this is national statistics as taken from this statistics site. So having known this burden of the disease, so one of the important challenge for the treatment of breast cancer is the drug resistance. So, for example, a tamoxifen drug is commonly administered drug for breast cancer, and there are known breast cancer which can resist tamoxifen therapy. So we would like to address this tamoxifen therapy. Can we identify a molecular predictor of tamoxifen resistance in breast cancer? Can we identify a protein molecule? Can we identify a gene signature in a breast cancer that can help us to understand if this breast cancer is going to develop the tamoxifen resistance or not? See, most of the breast cancer treated with tamoxifen don't show resistance at the early stage. And as the treatment progresses, the breast cancer can start developing tamoxifen resistance. So this was one of the primary things which we try to address. And also in parallel, we try to address the signaling pathways associated with tamoxifen resistance, and what protein molecules and what gene signature contributes to the tamoxifen resistance in breast cancer. For which I would like to give a background about certain signaling pathways and nuclear receptor story. The nuclear receptors, I'm sure the audience knows about signal transduction pathways that happens in a cell, which executes various functions for a normal cellular functions, physiological and also cellular functions. The nuclear receptors are a kind of receptors which belong to large superfamily of evolutionarily related DNA binding transcriptional factors, and they regulate gene expression, and the receptors may be present in the cytoplasm or nucleus. And ligands, most of the ligands for nuclear receptors are lipid soluble, so they can cross the membrane and bind to the receptors which may be present in the cytoplasm or nucleus. So the name also tells us certain things about nuclear receptors. The final execution of its action will be at the nucleus. So it can execute by mediating certain gene transcription in the nucleus. So the nuclear receptors are very important in terms of being a target for various disorders. And of course, tamoxifen is actually targeting the nuclear receptors like estradiol receptor. So, and if you look at the nuclear receptors as such in breast cancer, some of the nuclear receptors are pro-proliferative and some of them are anti-proliferative. And pro-proliferative, it augments the proliferation of the cells. And some of the nuclear receptors are anti-apoptotic, which means it prevents the apoptosis of the cells, which is a programmed suicide death of the cells. And some of the nuclear receptors are pro-apoptotic. I put it in a green things because some are good for the cancer and some are really bad for the cancer cells to progress. So the anti-proliferative examples are estradiol receptor beta, androgen receptor, PPR gamma, retinoic acid, vitamin D receptors, and these are all under the category of nuclear receptors. And other examples for pro-proliferative, anti-apoptotic, and pro-apoptotic, as you can see from the slide. So there are nuclear receptors that can help breast cancer to progress or grow or proliferate. There are nuclear receptors which can be antagonistic to the breast cancer growth. Now, Since my research involved estradiol receptor, which is a nuclear receptor, I would like to explain or show the domain structures of this nuclear receptor. As it is a protein, it contains many domains. And it's a nuclear receptor, so the domains are particularly organized for one domain to bind the ligand and one domain to bind to the DNA. As I mentioned before, nuclear receptors execute the function in the nucleus. It can be like a transcriptional factors that bind to the DNA response elements and can mediate the gene transcription. So some domains that's been represented here, like usually it's an amino terminal and carboxy terminal. That's about a protein structure. And also the domains like nuclear exit signal or nuclear localization signal. You can have domains A, B, domain C, domain D, and domain E and F. And each domain that connects DNA binding domain and the ligand binding domain. So there are also signal sequences present in those domains like nuclear localization signal, which will help them to move after the ligand binds, that's the estradiol, binds to the estradiol receptor in the cytoplasm. It can go to the nucleus through the signal, that's called nuclear localization signal. And once it has finished the job in the nucleus, it can just come out of this nucleus to the cytoplasm. So there is a continuous nucleus cytoplasmic shuttling of estradiol receptor, which is maintained or which is regulated by these domains of specialized amino acids which mediate this function. But however, in added to this complexity, the post-translational modification that determines the function of this estradiol receptor. What are post-translational modifications? Anything that's added to the proteins after the protein is made, like methylation, like acetylation, like many polymetallation. There are many post-translational modifications. In this protein, estradiol receptor also contains a lot of amino acids like serine, lysine, and arginine, and that undergoes post-translational modification, which gets phosphorylated. As you can see in domain A and B, for example, it contains serine as residues 104, 106, and there's a number of the amino acid that's being present in the domains, and these gets phosphorylated. And you can see the other proteins, other amino acids in other domains also getting phosphorylated. And this we group as post-translational modification. And this post-translational modification in turn determines the function of this estradiol receptor. And one important function of this estradiol receptor is this post-translational function modification control the nucleocytoplasmic translocation. So it controls the nuclear cytoplasmic shuttling. And this is very crucial for nuclear receptors because when it moves from nucleus to cytoplasm or cytoplasm to nucleus, it executes various functions. And this function and also the activation of genes. So for example, in breast cancer, the estradiol is a stimulant. And estradiol receptor is an important target for treatment of breast cancer. But if you look at the function of the estradiol receptor in context of breast cancer promotion, the more the estradiol receptor that stays in the nucleus mediates more and more gene transcription that promotes this breast cancer growth. So if the estradiol receptor, liganded estradiol receptor, is more present in the nucleus on the DNA response elements, it's going to make more and more genes or proteins and genes, and then they make proteins, and then proteins are going to help the breast cancer growth and proliferate the breast cancer growth. Now, a little bit about the signaling pathways of the breast cancer. As you know that breast cancer, in breast cancer, what happens, or in normal cells, estradiol is normally required for many cells to have its function, like endometrial cells or bone tissue or breast tissue. Normally, it has got its pathway, and that pathway is represented in your left side. And estradiol, as it's a nuclear receptor, and estradiol hormone is a steroid hormone, so it can cross the membrane. And once it crosses the membrane, it binds to the estradiol receptor. And estradiol receptor in unligated state, it's usually bound with heat shock proteins. That's being represented as HSP. Heat shock proteins is complexing with ER in unligated state. But estradiol, when it enters the cell, it binds to it, estradiol receptor, it dissociates from the heat shock proteins, and the liganted estradiol receptor in the dimeric form enters into the nucleus, and you can see it's binding nicely to the DNA response elements. And then what it does is to enhance the gene transcription. And these are estradiol regulated genes. And those genes are normally required for functioning of any estrogen response tissues. And in breast cancer, this is going to augment the breast tissue proliferation, breast cells proliferation. And the post-translational modification of estradiol receptor is also shown the above slide. You can see the DNA binding domain, and lot of signaling molecules post-translationally modify these estradiol receptors, like protein kinase A, ERK signaling pathways. These are different signaling pathways. And I'll just come back to one important residues in the estradiol receptor, and that's being regulated by SRC kinase, which is being shown in the AF2. AF2 stands for activator function 2, activator function 1. There are two activator functions which controls the ligand-dependent mediated regulation of estradiol receptor. You can focus on this tyrosine 537 residue that's being present at this AF2 domain, and that will be the focus of this talk, because we attempted to see how this tyrosine 537 phosphorylation regulates the nucleocytoplasmic shuttling of estradiol receptor, and what protein can modulate its regulation. Having talked about the estradiol signaling pathway, it's almost like a nuclear signaling pathway, but there is this called as classical nuclear signaling pathway, but there is also a non-classical signaling pathway of estradiol receptor. And in non-classical signaling pathway, the estradiol receptor, liganted estradiol receptor, combines with other molecules like Jun and co-activators, and they can bind to the AP1 region of the gene, and they can promote the gene transcription in other way. And this is not a classical way, because it's bound through other complexes and then mediate the gene transcription. But classical pathway is always that estradiol receptor binds to the estradiol response elements, direct binding to the DNA, along with the co-activators. That's being shown in this diagram. Now, this is a complex slide which shows how much the different crosstalks happens in the estradiol signaling pathway. I'm not going to go into the detail, but I would like the audience to appreciate the complexity of estradiol signaling pathway and its crosstalk with various other pathways like EGF, epidermal growth factor receptor pathway, insulin-like growth factor pathway, IGF, and all these pathways through MAP kinase. You would have heard about something about this kinase pathways from the previous speaker, PI3 kinase pathway. These pathways can also modulate the estradiol receptor actions because they are going to phosphorylate the estradiol receptor. As I mentioned, the post-translational modifications of estradiol receptor can be regulated by these pathways. So in other words, the estradiol signaling pathway, it's not one kind of, probably I would say a unidirectional or a single pathway. It is always in complex or in crosstalk with other pathways, and that can make into a classical action or a non-classical action as well. Now, the most important is what happens to this pathway in breast cancer, and what are the existing drugs for targeting this breast cancer. So you can see there are two on the right side of yours, the steroid selective estrogen receptor degraders or modulators. These are the classification of certain drugs under this category. The like fulvestrant, FUL is being abbreviated there. The fulvestrant can degrade the estradiol receptor. As I mentioned before, estradiol receptor and estradiol is required for normally functioning cells. But in breast cancer, it acts like a fuel. So it just makes the cells to proliferate. So one of the targeting ways is to block the action of estradiol receptor. And under this category, you have several drugs like fulvestrant, which is a degrader of estradiol receptor. And this degrades the estradiol receptor through the ubiquitin system. So there is no availability of estradiol receptor. for mediating its action. The other modulator is the tamoxifen, and tamoxifen is a similar structural analog of the estradiol receptor. And since it is a structural analog of estradiol receptor, it can bind to the estradiol receptor directly by displacing the estradiol or competing with the estradiol. So, excuse me, when tamoxifen is bound to the estradiol receptor, it's like analogy is that it just sits on the chair but doesn't perform the action. So it occupies the chair of the estradiol but doesn't perform the action what estradiol does. So the tamoxifen bound to estradiol receptor goes to the nucleus, but it does not do gene transcription because it recruits the co-repressors. Their co-activators can be classified as co-activators and co-repressors. Both are under the category of co-regulators. Especially for nuclear receptors, we need both. Okay, so the co-activators are those which augment the activation of this pathway. Co-repressors will block this pathway. So what is the way the tamoxifen acts is, tamoxifen sits on the chair for estradiol, goes to the nucleus, but it recruits co-repressors. So there is no activation of the pathway that estradiol used to do. So there is no active gene transcription of estradiol. So there is no estradiol responsive genes that gets upregulated. So there is no cell proliferation or whatever it does, the estradiol responsive genes has to do, it's blocked. So this is the two approaches, some of the two approaches that how it manages the breast cancer, and these are already available and practiced drugs. So you can antagonize this estradiol receptor in two states, that's premenopausal state and postmenopausal state. Of course, the cancer can happen in premenopausal state as well, but the contribution of estradiol is significantly different in premenopausal state and postmenopausal state. If you look at premenopausal state, ovaries contribute to the production of estradiol mostly, and in postmenopausal state, the peripheral adrenal sites and adrenal gland, and liver, muscle, and fat. So these are peripheral sites that contributes to the, of course, aromatase is one enzyme which can control the testosterone to estradiol. So the suppression of this estradiol can also be done by, you can do a GnRH inhibitors, and GnRH, as you know, is a gonadotrophin releasing hormone from the hypothalamus, which can block the pituitary activation like FSH, which can promote ovaries to produce the follicles and then estradiol. So GnRH inhibitors are ovaries removal. And in case of postmenopausal, aromatase inhibitors is tried out maximally because the peripheral sites contribute to the production of estradiol. And these are something which I talk. Of course, other than the fulvestrant and tamoxifen, which are only for estradiol receptor expressing breast cancer, as we already know, triple negative breast cancer, which does not express E2, P4, progesterone, or estradiol, or HER2, human epidermal growth factor receptors. So in that, the significantly, we cannot use this inhibitors for in that cases. We cannot use this fulvestrant or the tamoxifen in those cases. And there are additionally, experimentally, there are various drugs selectively chosen for triple negative breast cancer and estradiol positive, estradiol receptor positive breast cancer. There are some small peptides which can block the co-activators, and these are all in research, and also some of them are in clinical trials. So we have some small molecule inhibitors which can block co-activators action, and some of them are research. Now, where does this problem comes in this targeting? Like say we talked about fulvestrant and tamoxifen that can effectively block the estradiol receptor, and they can stop the breast cancer growth. But as I mentioned in the earlier slide, one of the problem is the breast cancer learns how to resist this tamoxifen. And that is called as tamoxifen resistance. So drug resistant. Any cancer, the major problem is resistant to chemotherapeutic drugs. So the tamoxifen is also initially, whichever is working, it becomes, the cells become resistant to the tamoxifen. And various molecular mechanisms have been correlated or have been figured out why these cancer cells, how these cancer cells become resistant to tamoxifen. And that's being put as a diagram here. And there are various pathways that cancer cells activate or upregulate so that it can resist the tamoxifen. And 4 points are there. Of course, these are not the only points which makes the cells, breast cancer cells, tamoxifen resistant, but it's correlated very well and experimentally proven for this phenomenon. So the loss of estradiol receptor, some breast cancer which expresses estradiol receptor, the receptor becomes extinct or lost. And endocrine adaptation, so there may be an adaptation or pharmacological tolerance or P proteins that's getting overexpressed, which makes the efflux of this tamoxifen and alteration of co-regulatory molecules or proteins, and some activation of the pathways like MAP kinase and PI3 kinase pathways. All these have been shown to promote the cancer cells to have the tamoxifen resistance. And this is one of the important problems that will be challenging for the clinician to administer the tamoxifen drug for the breast cancer patients who show this resistance. Now, the question for us was, so of course, the first question is, what makes the breast cancer cells resistant to tamoxifen? Of course, the literature shows many molecules that plays a key roles in tamoxifen resistance. But the question also is important to ask whether can we predict or can we identify patients? It's like a precision and also personalized. Can we predict a patient who is going to resist tamoxifen in the long run or in the early stages? Which molecule, which protein molecule in the breast cancer can predict the tamoxifen resistance, and which molecule can augment the tamoxifen response? These are the questions that can definitely help for the breast cancer treatment with tamoxifen. Can we predict a protein molecule that can help tamoxifen sensitivity, which is already present in the breast cancer cells? Can we predict a patient who is going to develop tamoxifen resistance? All very important key questions. Now, I'm going to present some data from these 3 papers that we published on this protein molecule called as protein tyrosine phosphatase. Okay, and this molecule, this protein molecule, how does it regulate breast cancer? For first question. The second is, how does it sensitize the breast cancer cells to tamoxifen? Is it playing a major role in tamoxifen sensitivity or resistance in breast cancer? And what is its expression profile in Indian patients, breast cancer patients? And also we talked about the crosstalks with other signaling pathways this protein plays so that they do this job of tamoxifen sensitivity or tamoxifen resistance. So I'm going to present few of data from these 3 papers. Now, I want to give an introduction to PTPH1. That's the protein I'm talking. PTPH1 also called as PTPN3 protein. And this is a tyrosine phosphatase. And this protein, actually what it does is it removes the phosphate group from tyrosine residues of the amino proteins. So tyrosine is one amino acid which gets post-translationally modified by phosphorylation. And this protein is a tyrosine phosphatase, which removes the phosphate group from the tyrosine residues. So it's mostly present in the cytoskeleton, and it got its cooperation with P38 gamma and also vitamin D receptor and various other molecules. It cooperates and collaborates for promoting cancer the interesting part of this protein is, in some cancer, it acts like a tumor suppressor. In some cancer, it acts like an oncogene. And it has a dual role. So it's very difficult to figure out whether it can classify PTPH1 as an oncogene or we can classify PTPH1 as a tumor suppressor protein. So these are some of the features of this protein. Now, the first experiment we do is, just to make simplified way of understanding is, if I want to address a protein molecule, what it actually does to breast cancer cells, okay? The simple experiment to do is genetically overexpress this protein or knock down this protein in breast cancer cells. So if you genetically modulate this expression of these proteins in breast cancer and try to understand the phenotype of the cells, we can figure out what kind of role the protein can play. So the summary of this data, this is experiment where we cultured the cells in the presence of tamoxifen and also genetically modified these cells to overexpress the PTPH1 protein or knock down this protein. So we are manipulating the cells. One is overexpression, another one is knockdown of this protein. And then expose these cells to tamoxifen and see what happens. How do the cells respond to tamoxifen? And these are breast cancer cell lines, MCF7 cells and T470 cells. This is a colony formation experiment. We call it colony formation experiment. And what we found out is if we overexpress this protein, tyrosine phosphatase, that is PTPN3, the cells shows enhanced sensitivity to tamoxifen. If we knock down this protein, the cells show less response or less sensitivity to the tamoxifen. The message is PTPH1 does something to the response of the cells for tamoxifen. That's what this experiment, we got this experiment. Now we have to know the mechanism, how this protein can mediate this effect. So we did some experiments in the MCF7 cells, and we also carried out an in vitro assays. Since I mentioned to you this protein is a tyrosine phosphatase, and also I have showed you that estradiol receptor protein contains tyrosine, we wanted to address whether this protein is doing something to the tyrosine residues of estradiol receptor. Very straightforward question, because tyrosine residue is present in the estradiol receptor, and tyrosine receptor undergoes phosphorylation, dephosphorylation in estradiol receptor. Since PTPH1 is augmenting the tamoxifen response, we try to understand if this PTPH1 can dephosphorylate the estradiol receptor. So this experiment is all about an in vitro experiments where we expressed PTPH1 protein, and then we incubated the cell extracts with the PTPH1 and try to look at the phosphorylation status of the estradiol receptor. And when we try to look at the phosphorylation status of the estradiol receptor, as you can see the arrow mark here, this protein, almost the tyrosine phosphorylated status of estradiol receptor gets decreased. And we also have shown in by immunoprecipitation experiments that the tyrosine phosphorylation decreases in MCF7 cells, both by in vitro incubation with cell extracts and PTPH1, GST PTPH1 protein, and also in vivo proteins. We showed that PTPH1 decreases the phosphorylation of tyrosine 537 residue in the ER. So we got two things, important things, that PTPH1 enhances tamoxifen sensitivity, and PTPH1 has the potential to dephosphorylate the tyrosine 537 that's present in the estradiol receptor. Now, all in vitro data should be validated by in vivo experiments. That's what classical experiments are being designed. So we also did it in mouse, that where we created xenografts of T470 cells. In T470 cells is a breast cancer cell line. We made a xenografts in the nude mice, which is immuno suppressed mice. And that mice, when we administer the tamoxifen, actually we created the xenograph by a cell line which overexpresses PTPH1. So we can genetically overexpress PTPH1 in these T470 cells. After overexpression, we created xenografts in the mouse. And then we looked at the response of this mouse xenografts to tamoxifen. As you can see from the graph, the summary of this data is, when we administer tamoxifen to the mouse that's being made of xenograft made of T470 cells with overexpressed PTPH1, it showed a very nice response to the PTPH1. As you can see, the red plot here, the tamoxifen treatment decreases the xenografts. We assess the xenograph sizes. Basically, this is measured tumor volumes. After xenografts, we administer tamoxifen at different time periods. We measure the tumor volume, and that's an indication of how much the tamoxifen is working for this xenografts. And also we did histological studies and to show how the tumor cells are affected by the tamoxifen. The summary of this, the experiment, the data summarizes to us that even in vivo data, the xenografts that overexpresses PTPH1 when these mouse are treated with tamoxifen, they show a great response to the anti-cancer activity of tamoxifen. Whereas if you show the PTPH1, less expression, that you can see as a control of green. You can see the green, that violet one, that's not showing response. In fact, the growth of the tumor is increasing. So even the in vivo data confirm to us that the protein, if it is overexpressed in the T470 cells, and that's going to show a good, nice anti-cancer response of tamoxifen. And now, 3 things we got to know. One is the PTPH1 increases the tamoxifen sensitivity. The second, PTPH1 decreases or modulates the phosphorylation status of tyrosine residue in the estradiol receptor. The third, even the in vivo tumor xenographs showed a good response to the tamoxifen if those cells are highly expressing PTPH1. Now we have to show a critical mechanism. How does this tamoxifen sensitivity really happens? Now, the Castoria et al. In 2012 published a paper which showed that if the tyrosine residue in GR, so tyrosine residue in the estradiol receptor modifies its position in the cell. In other ways, what I want to say here is, if the tyrosine residues in the estradiol receptor gets phosphorylated or dephosphorylated, whether it can determine the localization of estradiol receptor. Where, for example, estradiol receptor can be in the nucleus or can be in the cytoplasm based on the cellular state, based on the liganded state, based on the signaling pathway activation state, based on the phosphorylation and dephosphorylation of tyrosine 537 of the estradiol receptor. That's what these authors have shown. These authors have shown clearly that if the tyrosine 537 is phosphorylated or dephosphorylated, it can alter the localization of estradiol receptor. If it is phosphorylated, the nuclear localization is less. If it is in the 537F, which means this ER is mutated from tyrosine to phenylalanine, and then they did the experiment and their localization changes. You can see 0, 30, and 60 minutes after estradiol treatment. Just focus on this data, nucleus data. You can see the focus of this nuclear data. You can see by 60 minutes, the ER, which is a wild type, which has an intact tyrosine 537 residues, is almost gone in the nuclear fraction. This is a Western blotting data, which is taking proteins from nucleus, cytoplasm, and lysates, whole cell lysates. We can see it's almost gone in the nucleus. But in the phenylalanine, which is tyrosine mutated to phenylalanine, this is almost being present at 60 minutes. So which suggests that the tyrosine 537 residue of estradiol receptor determines its localization, whether it's in the cytoplasm or the nucleus. So we asked a question in our experiments, and we did a self-fractionization experiment. And this is the data, which is a Western blotting data, which is to understand the expression of the proteins. And we did genetic manipulation in the T470 cells and MCF7 cells, Teton PTPH1, which overexpresses the PTPH1 expression. And the summary of this experiments, what it suggested to us is, when the PTPH1 is overexpressed in breast cancer, the localization of ER is mostly, the localization of estradiol receptor is mostly in the nucleus. You can see this data. Just to avoid the complexity of other, just focus on this data. Here you see nuclear fraction, minus and plus indicates presence of PTPH1. Okay, if there is no PTPH1, the nuclear localization of this ER is less. But if there is a PTPH1 more in these cells, the nuclear localization is more. That's being shown even in the other cells. You can see this data, minus and plus. The ER PTPH1 is there, more ER is in the nucleus. And that's being shown in other experiments where we knock down this PTPH1 and we can show there is a decrease in the ER. So in other words, the summary of these experiments, we did a cell fractionization. We collected proteins from cytoplasm. We collected proteins from nucleus from the breast cancer cells. And those cells were genetically engineered to overexpress or knock down of this PTPH. Then we found that in the nucleus whether the ER is more in the presence of PTPH1. That's what we found out. When you have more PTPH1, ER is always present in the nucleus. When we have less PTPH1 nuclear, the localization of ER is very low in the nucleus. That's what these experiments, cell fractionation experiments, suggest to us. And since it is a tyrosine phosphatase, I'm sure phosphatase is an enzyme. So what is the critical amino acid that regulates the activity of the tyrosine phosphatase? We made some mutations in the PTPH1 protein, and then we overexpress that PTPH1 mutated proteins. This is all possible by genetic experiments or genetic manipulation of cells and molecular cloning. So we did this experiment and found a critical residue that's being present in the PTPH1 that regulates the activity of this tyrosine phosphatase. And also we looked at this turnover of PTPH1 by a cyclohexamide chase experiment, which means the cyclohexamide is given to the cells and then looked at how much PTPH1 is present at different time points. It's basically it inhibits the translation of the proteins, and we looked at the dynamics of this protein presence in the breast cancer cells like MCF7 cells, and another types of cells is 293 cells. And we looked at the turnover of this PTPH1 protein, and which suggested to us that the mutated ER in the GFP mutated ER, the phosphorylation of the turnover of the protein is much more. So the experiments suggested that the PTPH1 is not unidirectionally communicated to the estradiol receptor. There is a bidirectional communication between PTPH1 and the estradiol receptor, which means PTPH1 controls tyrosine phosphorylation of estradiol, and estradiol receptor in turn alters the turnover of the PTPH1. So both have a bidirectional communication. That's what we figured out from this experiment. Now, summarizing this data, basically we summarized based on these findings. I'm just excluding some of the data, but like on ER2 signaling pathways. But what I like to summarize to easily understand the implications of this research is, let's see the usual pathway of estradiol in a breast cancer cell. The estradiol just crosses the cell membrane. It's a lipophilic hormone or steroid hormone, crosses the membrane. And in the nucleus or in the cytoplasm, it can bind either way. You can see a liganted estradiol receptor goes to the nucleus, and this is a DNA. And you can see the liganted estradiol receptor binds to the DNA into a response element, estradiol response elements. And along with the co-activators, it's going to mediate the gene transcription, many gene transcription. Now, we saw one thing, that PTPH1 makes ER to be more in the nucleus. It will not be allowing the the nuclear exit of estradiol receptor. So the PTPH1 is present in the cytoplasm. Let's say the normal pathway, how does it happen is, once the ER binds to the DNA, it gets phosphorylated at tyrosine 537. This tyrosine 537 phosphorylation makes them to export to the cytoplasm. So you must know that anytime the activation and deactivation of pathway should happen. It's not always there should be a signaling pathway activation. So here also, the signaling pathway of ER stops by phosphorylating tyrosine 537 in the nucleus. After phosphorylation, it moves to the cytoplasm. This is normally happening by deactivation or regulation of this estradiol signaling pathway. But what PTPH1 does in the cytoplasm is, it dephosphorylates 537 and pushes back to the nucleus. So these are the facts. Number one, the estradiol receptor binds into the DNA response elements in the nucleus and mediate gene transcription. Number two, in the nucleus, the phosphorylation of tyrosine 537 by SARC kinases moves the ER out of the nucleus to the cytoplasm. This is the normal way of stopping the estradiol receptor signaling pathway. Fact three, in the cytoplasm, if PTPH1 is present, this PTPH1 dephosphorylates the tyrosine 537 and pushes back to the nucleus. This is what normally happening in the estradiol. Now you can question how PTPH1 is mediating the sensitivity of the drug. Now you imagine a situation, the breast cancer patients are given tamoxifen. Now the tamoxifen is what? The structural analog of estradiol. So it can occupy the seat of the estradiol. It can go and bind to the estradiol receptor and goes and binds to the DNA response elements as similar to this pathway. And it cannot mediate any activation of this pathway because it is going to recruit co-repressors. So there is no gene transcription being mediated because this is a suppression, because co-repressors will suppress this pathway in compared to this pathway. Now, but the tyrosine 537 can get phosphorylated and pushes back this ER to the cytoplasm. Now in this cytoplasm, this complex will come out. Now the advantage for us in this case is the more the time of this complex sits in the nucleus on the DNA response elements, the more is the beneficial for the therapeutic aspects, because it's going to inhibit the gene transcription, and this gene inhibition of gene transcription doesn't allow the ER responsive genes to come out, and this is very useful for the breast cancer in fact. So you can imagine the desirable effect for this side is to have more and more time of this complex to sit on the DNA response elements in the nucleus, so there is no gene transcription. To have that, this should be here only. For that, the PTPH1, when this complex gets phosphorylated, 537 moves out of the nucleus, the PTPH1 in the cytoplasm pushes back to the nucleus and more sits on the DNA response element. So we can understand overall, we can understand this pathway is such a way that in tamoxifen given situation, tamoxifen bounds to the estradiol receptor and binds to the DNA response elements, does not mediate any gene transcription. But tyrosine 537 gets phosphorylated by Src kinase and it is shuttled to the cytoplasm because of this phosphorylation. But PTPH1 in the cytoplasm can dephosphorylate the 537 and pushes back to the nucleus, and it sits on the DNA response elements. The more the time it sits on the DNA response elements, this complex, along with the co-repressor, is more beneficial because there is no gene transcription. There are no protein products that's going to be translated and mediate the proliferation of the breast cancer cells. So this is what we figured out, and it's one of the important way how the PTPH1 modulates, and that's what we got. The PTPH1 is there; it enhances the tamoxifen sensitivity of the cancer cells. Now, going on forward, all this data has been done in breast cancer cells and nude mice in animal models. Now, we would like to look at this expression of this PTPH1 in the breast cancer tissues. Now, coming back to this, we took some breast cancer tissue samples from patients, and we also got different categories of patients. As I already told you, the breast cancer can be subtypes based on the nuclear receptors, based on histological aspects and all that stuff. We did the expression of PTPH1, and this is a simple immunohistochemistry analysis of this protein, PTPH1. And we looked at the expression of this PTPH1 in different clinical cases and correlated with the clinical characteristics of the patients. And where we show there is some kind of correlation between the presence of estradiol receptor or human epidermal growth factor receptors. And we show there is a differential regulation of this PTPH1 protein. And you can see most of the localization of this protein is cytoplasmic. And we see the number one fact, the PTPH1 protein is highly expressed in the breast cancer tissues. And these are Indian breast cancer patients. Number two fact, these are differentially expressed based on the presence of nuclear receptors like estradiol receptor and progesterone receptor. Number three, it's also correlated with the KI index or the proliferative index of the breast cancer patients. These are some of the data we got, and I'm not showing much of the other clinical correlation, but this is an interesting data we done in Indian patients, which showed abundant expression of PR. We are trying to make more sense out of it. As I asked in the first slide, the question is whether this protein can predict or classify the patients who can be responding to the tamoxifen better than the others. Can we have a tamoxifen therapy as a personalized therapy which can show much better response based on this PTPH1 expression? These are questions. Of course, this cannot be done very shortly. We need a long-term follow-up and other things which we will try to work out. And we also looked at the genetic expression of this protein. As I said, any gene can be genetically controlled or epigenetically altered. Okay, I'm sure epigenetics, you are all aware of the epigenetics, which means the expression of gene is altered without changing the DNA sequence. So we looked at genetic expression, genetic factors of PTPH1 expression, and also we looked at the epigenetic control of PTPH1. And this technique is a methylation specific PCR, which we make a bisulfite treatment of the DNA isolated from the tumor samples. And bisulfate will convert the CPG islands. And after conversion, we have two designed primers, which detect specifically methylation of CPG residues, specifically CPG residues. So the basic facts is the CPG islands are present in the promoter region, and they are the one which gets methylated. And methylation is one of the important epigenetic modification, which will alter the gene expression. So we also try to look at this epigenetic alteration of the PTPH1 expression by a methylation specific PCR, and we didn't find any significant epigenetic mode of PTPH1 regulation. So we concluded that the epigenetic mode of regulation of PTPH1 is not prevalent in the breast cancer samples. How much time I have, ma'am? Can I move or can I stop here because of the time constraint? Hello?
Sir, you had 45 minutes actually for presentation, and I think you have a few minutes, sir, 5 minutes probably.
No, I will leave that 5 minutes for discussion. Maybe I will stop here by stating certain facts now. I'll conclude my talk. I'm not going to present the other part of the work since we are running short of time, and I would love to have an interaction as well. The fact is, what we have identified is we have identified a protein molecule in breast cancer that can augment tamoxifen sensitivity, or that can. And second is the mechanism of this PTPH1, how does it make tamoxifen sensitivity is through the phosphorylation and dephosphorylation of tyrosine 537 residue of estradiol receptor. Third fact is PTPH1 modulates the nuclear cytoplasmic translocation, nuclear cytoplasmic shuttling of estradiol receptors, which is very key determinant of drug sensitivity, tamoxifen sensitivity in the breast cancer. Fourth fact is we have also profiled this expression of this protein in breast cancer patients, Indian breast cancer patients, and also have a correlation with the clinical characteristics of the patients. The fifth fact is we try to understand, since we are asking a question whether this can be a predictor of tamoxifen sensitivity, we have to know the genetic and epigenetic control of this protein. So we try to ask one way of epigenetic mode of regulation of this protein by methylation specific PCR. And that doesn't suggest us that the epigenetic mode of regulation is a key determinant way how the PTPH1 is regulated. So, so in the long run, what we just desire, or we are ambitious. Probably we can say we are ambitious, or probably, but it needs a coordination, cooperation from the clinical data and other things, etc., etc. Will be. In the long run, is there a way we can predict or segregate patients into two groups? High expressing PTPH1 can show more tamoxifen sensitivity group, and low expressing PTPH1 and low tamoxifen sensitivity group, and also tamoxifen resistance, who can develop, who cannot develop. This is all our long-term plans in this project. Thank you once again. I stop here, and I am looking for some questions to answer. And once again, I thank the organizers and also the patient listening, the audience for the patient listening. Thank you very much.
Thank you, sir. Thank you for the wonderful presentation. Over to the chairperson for discussion.
Myself, Dr. Vidya. I couldn't find any questions in chat box as such. On behalf of organizing committee, I congratulate Dr. Suresh and I express appreciation for your excellent talk on breast cancer. Your areas of research and depth of understanding was commendable, and the breast cancer burden, risk factors, and how the tamoxifen resistance occurs, for the drug resistance occurs, and signaling pathway, and your area of research was very neatly explained. Once again, congratulate Dr. Suresh.
Thank you, ma'am. Thank you, ma'am.
Just one question, sir.
Yes, ma'am.
Is your work, research work, is funded research project or something like that?
Oh, okay. I think it's a good question. 1 minute, ma'am. Let me. My slides are visible?
Slideshow is not there.
Not visible, sir.
No, let me put an acknowledgement slide. I forgot to acknowledge. That was the last slide of mine. But since you have asked me, I think it's very important to acknowledge the funding agencies. This study is funded by DBT, 2 projects from DBT and UGC and also DST SERB. This project is funded by these funding agencies. And also I'd like to thank different people in this regard, which is very important. My PhD scholars, Dr. Abhishek Shetty, who is now in Johns Hopkins, USA, doing his postdoctoral work, and Sanu Thangachen is my PhD scholar who has done some work of it, and my postdoctoral mentor, Dr. Chen, from Medical College of Wisconsin, and some of the portion which I showed, a molecular cancer therapeutics paper, has been generated from his lab, funding from NIH and other agencies. And also, I thank my current institute for support and some of the investigators for sending us some cell lines and other things. And thank you for asking this. It's very important for me to acknowledge. And it's a funded, yes, it's a funded, coming back to your question. Thank you, ma'am.
Excellent, sir. Thank you. Thank you. I thank all the organizing committee members for giving me this opportunity to chair this wonderful session.
Thank you, ma'am. On behalf of 4th International Conference 2023, we'd like to appreciate the resource person, Dr. P.S. Suresh, sir, with the e-certificate and virtual memento. Thank you, sir.
Thank you very much. Thank you.
We'll also appreciate the chairpersons with the e-certificate and memento, Dr. Deepti Shakti. Thank you, ma'am. Also appreciate the next chairperson, Dr. Vidya CS, with the e-certificate and the virtual memento.
And also I thank chairpersons also for conducting this session very well and the kind introduction, Deepti ma'am and Vidya ma'am. Thank you very much.
Thank you.
Thank you, sir.
Thank
That's
you all.
updated in end of session.
Sure, I will leave. Thank you.
Good morning, everyone. So we'll continue with the next session. The next session is on trans fats and neurochemistry, the biochemist prescription. So I welcome our speaker, Dr. Sukanya Gangopadhyay ma'am, as well as our chairperson, Dr. Gautam Sarkar sir. First, let me introduce our chairperson for the next talk. It's Dr. Gautam Sarkar. So he has graduated from Medical College, Calcutta, post-graduation from AIIMS, New Delhi. He's at present working as a professor in the Department of Biochemistry, Arubadai Medical College Hospital in Pondicherry. He has served in the post of HOD post in various other medical colleges throughout India, including Andaman and Nicobar Islands. He has received the Young Scientist Award at the 16th International Conference of International Union of Biochemistry and Molecular Biology at New Delhi, which was held in the November 1994. He has contributed to the chapter on the structure and function of cell membrane in the medical physiology textbook by S.S. Sarkar, CBS Publishers. He's published many papers in many reputed national and international journals. He's a very passionate teacher, as well as he's a very good research. His main interest is in research. So with this small introduction, I welcome Dr. Gautam Sarkar sir to take over the stage. So over to you.
Thank you, Dr. Manju. It's my proud privilege to introduce Dr. Sukanya Gangopadhyay. So I have a list of her achievements, which are a bit too much actually for me. So I'll be showing a short summary of whatever has been given to me. So she graduated, that is, she did her MBBS from Lady Hardinge Medical College, Delhi University. Postgraduate, that is MD from VP Chest Institute, Delhi University. Done MBA in Total Quality Management. Awarded Fellowship of International Medical Science Academy. Awarded membership of National Academy of Medical Sciences. She is currently working as Associate Professor in the Department of Biochemistry, Vardhaman Mahavir Medical College and Safdarjung Hospital, New Delhi. She has a lot of post-MD experience. She is an NABL technical assessor, life member of several professional bodies, publication in different international and national journals. And her research interest is in nutrition and genetics in health and disease, and as well as medical education. She has received awards in national conferences, attended numerous national, international conferences and workshops, organized workshops on laboratory accreditation at BMMC, a co-coordinator of medical education unit at BMMC, avidly taught medical students and several other courses, guided MD thesis, worked in government and private laboratories, Lady Hardinge Medical College, and guest lectures and CME talks. My God, too much. But above all, that is a formality. But above all, I have to add from my side, she's a remarkable human being. Okay. Now, Dr. Sukanya, this stage is yours.
So thank you so much. Thank you so much. I'll share my screen, sir.
Yes, ma'am. Yes, ma'am. Please go ahead.
Ma'am, the slides are showing?
Yes, ma'am.
Yes, okay. Thank you. So at the very outset, I would like to express my gratitude to Dr. Sarkar sir for giving me the opportunity to express my views in this highly coveted conference with a wide audience. And also I would like to thank Madam Manju Madam for her cooperation during this entire preliminary phase before the actual time of the conference. So Madam had been a great help. And also thanks to the vast and diverse audience for giving me their precious time for listening out my thoughts. And I would like to rekindle your attention towards a very prevalent, well-known, yet a frequently ignored issue, the issue of trans fats. So let us revisit what we know and what is new with fats, nerves, and our overall health. We will be dealing with the following questions. What is the effect of trans fats on neurodevelopment and mental health? Does it really matter to us as adults? What is new in trans fat research? What should be the dietary balance of fat classes? And what about the fat-rich diets for weight loss and health, which are in vogue nowadays? Just for the reminder, the chemistry part of the lipids for the non-biochemists out there, let us quickly revise what all we have learned as biologists and medical scientists, that SFA is saturated fatty acid, monounsaturated fatty acid, polyunsaturated, trans fatty acid, essential fatty acids, which are not synthesized in the body, required in the diet, and the different types of the presentations of the fatty acids in the body, that is the glycerol backbone ones, the sphingolipid backbone ones, the glycerol backbone triglycerides, We all know that they have the three glycerol, three fatty acid chains attached to them. One of them or all of them may be polyunsaturated. So these polyunsaturated, monounsaturated fatty acids give a bend or kink to the base glycerol molecule. So the plasma membrane and wherever these fatty acids are situated, the fluidity depends upon that. And these, let me also remind you about these trans fatty acids. In the trans fatty acids, the kink of the cis double bond is absent. It is a virtual pseudo straight line. So that comes the problem. And here the health issues can arise. Also, let me remind you about the different types like the oleic acid, linoleic acid, arachidonic acid, and also the medium chain triglycerides and the omega classification. This I'll deal with later. But this is the main we have to remind ourselves before the topic begins. So what trans fats are? In the past century, we have seen a steep rise in the world population, putting a toll on the food supply. The mass production of fertilizers and genetically modified crops have somehow managed to feed the masses. But as necessity is the mother of invention, the methods to increase the shelf life of life-giving food is constantly being invented, reinvented, and industrialized. We have the courtesy of the patent for the hydrogenation of fats to Mr. Wilhelm Norman way back in 1900s, where the shelf life of fats was able to be increased, so giving a help to the storage for the food of the population. But as we know that industrial boons often tend to have the serious flip sides. So food with long shelf life definitely shorten the lifeline. When the turning point has come for the trans fats, when US FDA bans the trans fats in 2015, they had given a 3-year window period for the US food agencies to remove all the trans fat from the circulation of the human population. There were various studies backing up this ban. There were many animal studies in which they show that medium chain trans fat diet induces liver fibrosis and non-alcoholic steatohepatitis NASH in animal models. This and then other studies in the year 2012 have shown that dietary trans fatty acid induces NASH is normalized when the trans fat is removed from the hepatic lipid pools. Then there are other studies which show that trans fat supplementation exacerbates the behavioral biochemical changes in models of mania. And then influence of trans fat and omega-3 fatty acids on the preference of psychostimulant drugs in the first generation of young rats was shown. Also, it modifies the brain-derived natriuretic factor, mRNA, in the hippocampus of animal models, showing that trans fat has effect in the brain neurotransmitter organization as well. And finally, there was another study in the year 2012 which showed that could the trans fatty acids induced movement disorders. So not only the nerves, but also the functioning of the muscles were also shown to be probably affected. And in the year 2022, January, Indian FSSAI has also stated that all food items should contain less than 2% trans fats in the diet. So what are the risks we have with the fatty acids? There can be coronary artery disease, diabetes, obesity, liver dysfunction, infertility, acne, certain cancers, breast and prostate in particular, and brain and neurological dysfunctions. These all are rampant. And why the risk occurs? Why trans fatty acids are causing these risks? Because the trans fat get incorporated in the cell membrane, distorting the structural organization of the cell membrane. Thus, the signal transduction of all the cells may get affected. So what is the scientific proof? What exactly happens? The trans fat may disrupt the body's ability to metabolize the essential fatty acids. They impair the metabolism of the long chain PUFAs, the long chain polyunsaturated fatty acids. They raise the LDL, the bad cholesterol, by decreasing receptor-mediated clearance, by decreasing the LDL ApoB catabolism mechanism. They also decrease the HDL or the good cholesterol. Thus, both ways, they are increasing the LDL-HDL ratio to the highest terms. Trans fats, rather than anything else, can increase the LDL-HDL ratio. They increase the CRP and other inflammatory markers in the body, various parts like in the liver, in the arterioles, in the pancreas, and also in the brain cells. The inflammatory markers have been seen to be risen, and it promotes intra-abdominal deposition of fat. There is a term called industrial trans fat, which means the thing which was patented way back in 1900s, where hydrogenation of fat was promoted at that time very much before this 2015 mandate of being banned. So this industrial trans fat, which was produced by the hydrogenation process, that is the main culprit. And on opposite to that, there is a natural ruminant trans fat, the vaccinic acid. Mostly the industrial trans fat is the elidic acid. And the natural trans fat, which is present in very low amounts in animal diet, milk products, and meat products. These natural trans fats are generally present in the food items. If we take a balanced diet, then around 1 to 2% of this trans fat can be, the calorie can be produced from these naturally available trans fats. But research, I will show in the later slides, that research show that these natural trans fats, as compared to the industrial trans fat, do not harm us by these above given parameters so much like the elidic acid-like trans fats. So these industrial trans fats promote inflammation and endoplasmic reticulum stress much more than anything else. They promote the fat storage in liver at the expense of adipose tissue. So if there is excess calorie, excess fat, it will be promoted to be stored in the liver, giving the fatty liver and NASH-like symptoms. It stimulates the cholesterol synthesis pathway also, so generating the inherent lipids as well. How it does so? By activating the steroid regulatory element binding protein mediated gene regulation. They also impair the post-insulin receptor binding signal transduction. They interfere with the liver enzyme delta-6 desaturase, which converts the essential fatty acids to prostaglandins, and the paracrine hormone, the paracrine system is thus affected. So the trans fat has been shown to interfere with the liver desaturase enzyme as well. And another thing, interestingly, it activates the apoptosis signal regulating kinase, MAP kinase pathway, and thus increases the apoptosis in the livers and other organs. The trans fat increases the CETP activity, which could increase the transfer of cholesterol esters from HDL to the LDL, thereby contributing to the decreased HDL levels and increased LDL levels. So ultimately, this trans fat is making our body's fat proportion towards the bad side. The LDL-HDL ratio is very, very high when the trans fat intake is high. There is a correlation with cancer too. The breast cancer and prostate cancer, it has been shown to have this effect with the trans fat, but exactly how the research is still ongoing. Exactly the mechanism might be by this apoptosis signal regulating kinase ASK1 pathway, might be by some other pathway, which is still not very clear, but large population studies have shown that trans fat is increased in such patients. Now coming to the what happens to the what are the risks to the brain? There is a risk to the neurodevelopmental disorders, ADHD, depression, mania, decreased memory, and Alzheimer's disease. So from child on to adults, nobody is spared. how the process is affecting, there is decreased long chain PUFA secretion in the breast milk, thus affecting child intelligence. That means when the mother having a fetus inside is having the high intake of trans fats. In that case, the LC PUFA, which is normally secreted in the breast milk, that will be affected. So that PUFA concentration or secretion in the breast milk will decrease. So that will affect the child's intelligence. Decreased conversion of the essential fatty acids to long chain PUFA will affect the brain signal transduction and also the neural membrane structure. So ultimately, it is not only decreasing the secretion, also it decreases the production. The conversion of essential fatty acids to LC PUFA is very much affected with the higher intake of trans fat. This type of trans fat substitutes for DHA, that is the essential fatty acid docosahexaenoic acid, in the orbitofrontal cortex of the brain. So this DHA is very important in the new nerve generation and the neuro membrane regeneration. So this will substitute itself for the DHA and thus creating havoc in the memory, and also it decrease the sensitivity of the neurons. They incorporate themselves into the brain cell membrane as well. They alter the ability of neurons to communicate, thus diminishing the mental performance. It has been shown to cause substantial brain shrinkage, and also the neurotransmitters like serotonin production is very much affected in the brain. So trans fats are in the brain causing so much of harm that not only the child is being affected, the depression, mania, and Alzheimer disease is affecting the adults as well. But how is it so? How the adults are actually affected? Why do we have to really worry? Because as we somehow know that the brain is formed by 2 years of age. By the adulthood, not much of the new brain formation is there. So what is the basic reason behind that? Because 70% of the brain is fat, and adult brain cells are generally, they are fixed, no doubt, but yet the connections, the neural connections, they regenerate. The neurogenesis in adults generates the functional neurons from the adult neuron precursors throughout the life in certain restricted brain regions in the mammals. The brain cell turnover is 1.75% per year in adults, which is approximately 700 new neurons are being formed or produced per day in certain brain restricted regions only, not the entire brain. The cortexes do not change often, but in the orbitofrontal cortex and the dentate gyrase of the hippocampus, they are the most affected, where around 1500 new neurons per day are being produced. They are responsible for the new episodic memories and spontaneous exploration of novel environments and other functions of the brain. This turnover of the brain in adults is similar in males, females, and people more than 50-year-old. Everything, the turnover is very much similar. Neuroplasticity is always occurring in the adult brain, which is the ability of the neural networks to change through the growth and reorganization. So adult brains are obviously affected. They are in a constant equilibrium, and the new connections are always forming. What is the latest research on the fats, the trans fats, and other fats on health? So far, we have seen that trans fat, the prevalence of trans fat yet being banned, but still not being banned in all the countries of the world and not completely out of our diet. The products, the food products, they claim to have trans fat less than 2% on paper, but how actually they are able to reduce the trans fat without decreasing the cooking techniques or increasing the shelf life? That is the main question. So not only we should be concerned about the trans fat, but also the other fat. We have to focus on what should be the quality fat in our diet. various researchers have said that the dietary fats are associated with inflammation. Not only the trans fat, but they have also focused on the N6, that is the omega-6 series of the PUFA. So the omega-6 series is also probably affecting the inflammation in the all over health of the individual. Certain studies in 2021, very latest, have shown that ruminant derived TFA, that is the vaccinic acid, they have a different composition with the industrial TFA, which is having the conjugated linoleic acid. So studies are being done to show the effect of trans fats on the human health. What are the consumption patterns? The comprehensive toxicological analysis of trans fats revealed that a pro-apoptotic action specific to the industrial TFAs are counteracted by the polysaturated fatty acids, especially the omega-3 types. So the omega-3 fatty acids have been shown to cure or have a reverse effect on the industrial trans fatty acids. What is the mechanism of action of trans fats? That is being reviewed by an author in 2020. What are the trans fatty acids that facilitate the DNA damage induced apoptosis by the JNK-SAB-ROS positive feedback loop pathway? And certain studies have also shown that exposure to trans fat during the developmental period in animal models, in the Drosophila model, it alters the composition of fatty acids in the head and induces a depression-like behavior. So the the link has been the scientists, the researchers are constantly trying to establish the solid link of trans fat with the brain system. And in a study in 2022, the researchers have seen that diets rich in the N6 PUFA, saturated fatty acids, and trans fatty acids definitely directly increase the neuroinflammation, whereas the diets rich in monosaturated fatty acids and omega-3 PUFA and the sphingolipids can do the reverse effect. They can decrease the neuroinflammation. A higher dietary intake of omega-6 results in the excessive incorporation of these molecules in the cell membrane and various pathological consequences because of the pro-inflammatory the eicosanoids. So we can see that the entire system of fat-induced health damage is probably somehow mediated by the pro-inflammatory factors. So what should we do? Should we have a totally fat-free diet in adults? Should we totally stop taking the fats in our diet? Because already the dietary components in milk, nuts, seeds, they already have the proportion of the fatty acids. So we should also know what is the goodness of the fat. The fats are the storehouse of fat-soluble vitamins, antioxidants. The essential fatty acids are very useful in neural regeneration for paracrine functions with the help of the prostaglandins, cell membrane regeneration. We also saw an article on the food for mood, which says that the omega-3 series members like the docosahexaenoic acid, eicosapentaenoic acid, DHA, and EPA, which are the omega-3 series essential fatty acids, they provide fluidity to the cell membranes, thus facilitating certain processes, including neurotransmission, ion channel flow, thus increasing the brain activity, the regularization of the brain activity. And thirdly, we also know that there are non-oil fat sources, milk, fish, nuts, seeds. Oil as a cooking medium. It reduces the requirements of high temperatures of baking or the cooking in the oven, high temperature cooking in the oven. These things can be avoided if we use oil as a cooking medium. The benefits of the ratio of PUFA, MUFA are that they increase the LDL clearance, they increase the concentration of the HDL in the blood, they activate receptors So this polyunsaturated fatty acids, specifically in the omega-3 series, they have shown to increase the good fat proportion in the body. And there are yet to be discovered signaling and receptor pathways by which the overall health of the individual may be determined by the fat in the diet are still ongoing. So just because we have seen certain negative effects of the fat in the health may not mean that we decrease or make the oil concentration in our diets totally zero, because certain oils like the mustard oil, the coconut oil, they have antioxidants, they have vitamins in them. So how will we get those fat soluble vitamins? And these antioxidants and anti-cancer elements are very important to keeping the body overall healthy. So what are the right fats? To understand the right fats, we have to see the 1/3 rule in the adult diet, where 1/3 of the calories should come from fats, and 1/3 of those fats should be saturated fats, and 1/3 PUFA and 1/3 MUFA. So that is a 1/3 rule in the adult diet. So the NCEP, the National Cholesterol Evaluation Program, they have suggested this 1/3 rule. And we also know that there is a correct omega-3 and omega-6 ratio. This omega-3, omega-6 ratio in the PUFA intake is very, very important in avoiding, preventing the neuroinflammation and the other cardiac and muscular inflammation, which may lead to certain diseases. Because omega-6, if taken in a huge amount, will counteract the benefits of omega-3. It is important to take omega-6, but the proportion, the ratio has to be correct. So flaxseed oil, canola oil, fish oil, they are all the richer sources of omega-3 rather than omega-6. And this omega-3 is destroyed by the heating process. And also there is another thing coming up, the medium chain triglycerides. Coconut oil, which is rich in the 6 to 12 alipathic chain, caprylic acid, caproic acid. This coconut oil is rich in medium chain triglyceride, which can be very easily digested in the in our digestive system, so that the absorption of the fat becomes very easy. The requirement of absorption for the lacteals and the lipoprotein requirement is negated. And this type of medium chain triglycerides, if taken in high amount in the diet, higher amount, proportionately higher amount, will treat steatorrhea, digestive absorption disorders, and many more. What are the NCP guidelines and for the dietary balance? They say that the PUFA should be less than 10% of the total calories. Trans fat should be less than 1%. And this 1% of trans fat is very quickly reached by the ruminant, naturally provided trans fats in the milk and meat. So the industrial trans fat actually should be zero in our diet. The NCEP guidelines also say that MUFA should be 10 to 15% of total calories. Saturated fatty acids have to be very less, at less than 7% of the total calories. The SFA, MUFA, and PUFA proportion should generally be 1:1.3:1. And a word on omega-3. The omega-3 present in flaxseed oil is very high in alpha-linolenic acid. And the fish oil is more rich in the DHA and EPA, that is the docosahexaenoic and the icosapentaenoic. Both these DHA and EPA are not actually categorized as the essential fatty acids, because they can be produced from the ALA, that is the alpha-linolenic acid. The DHA and EPA can be produced from ELA, but the conversion rate being very slow. So that is why DHA, EPA are preferably taken in the diet also, so as to have a higher concentration to combat the neuroinflammation produced by the other harmful fats, which we invariably tend to take in our diet. The Mediterranean diet has shown to reduce cardiac events. They have a high concentration of olive oils and the PUFA and MUFA in their diet. They cook their diet at a lower temperature. The Mediterranean diet is nowadays showing to be having a better effect on the cardiac health. But actually what it is doing, the keto diet, it controls morbid obesity, but in keto diet, we are totally ignoring the carbohydrates at all. Not even fructose or fruits are being taken. And the MCT diet, that is the medium chain triglyceride diet, it is shown to treat malabsorption, upregulating the mitochondrial biogenesis, that thus improving the exercise tolerance. So these three types of diets are rich in the fats, but we should take whatever is suitable for our climate and our type. So in conclusion, we should be very careful to choose what we eat. We should restrict the calories from fats to 1/3, taking into consideration the 1/3 rule. We make that that 1/3 portion which we are taken has to be rich in most useful types of fat. We should have to focus on the quality of food, and we have to take special care of the yet to born and the newborn brains. And the 2% calorie limit by trans fat is already reached by the naturally occurring ruminant vaccinic acid present in milk and meat. So industrial trans fat or hydrogenated trans fat has to be consciously kept to totally zero in our diet. Thank you very much. And thank you again for giving me this opportunity to speak in this coveted this conference.
Thank you, ma'am. Over to you, sir.
Thank you, Dr. Sukanya. It was very, very interesting indeed. And may I ask you a few simple questions? Of course, I have several.
Sure, sir.
A practical question. The practical question is that, most of the food items that we Indians take, they are not packed food. I mean, in urban situation, of course, it is packed. But when you see India in general, it is not packed food. We have so many other foods that is available, such as we have samosas and this and that, which definitely contain a lot of trans fatty acids, I'm sure. And we have no means of really finding out how much they are. So do we have any established labs where you can send us samples for getting the trans fatty acids measured? That is one question of mine. And what is the fate of trans fatty acids in the body? Suppose we end up having it. Okay. And at one juncture, we think of stopping all trans fatty acids as far as possible. Now, what happens to the trans fatty acids that you already have in the body? I mean, do they take a lot of time to move out of the body? And I have several this thing. Of course, I'm not asking all of them. You told that the omega-3, omega-6 fatty acids present in fish oils, they are destroyed by heat. So should we be resorting to having fish oils only in the form of capsules, or should we be looking forward to having it in raw fish? Because raw fish, if we cook fish, then we don't have it anymore because it is treated by heat. So we can take the Japanese foods like sashimi. So, of course, the effect of refining on oils, there are many more, but so much for now.
Yeah, sir, yes. Sir, first of all, coming to your first question, as you asked that the measurement of the trans fatty acids, the labs where we can send the samples for measuring our trans fatty acids. Actually, the purpose of my talk was to initiate a discussion on this aspect, because trans fats is, since they have banned it and so much of the industry depends on it. And at the outset only, I told that the world population is increasing, and such processes of industrialization keeps our population alive. So having the natural food 100% may not be feasible for the entire population. So storage and increasing shelf life options have to be revisited. And basically, I could not myself find any labs which are doing the testing. Yes, there are labs which test the food sample. The food sample is being tested by the GC techniques, the gas chromatography, etc. But the serum trans fatty acids testing, NABL or any lab in India, I could not find. So maybe it is a food for thought for the biochemists of the future, that we can initiate or validate certain tests for this purpose. And the second question that you asked was, you asked that omega-3 and omega-6 is you said that it is influenced by the cooking. It will be damaged by the cooking purpose. So should we resort to fish oil capsules or the Japanese sushi technique of eating fish only or not? So basically, sir, what I feel that since it is of course better to have food in the raw form, but certain amount of low heating techniques to cook the fish that could be practiced, like the south in the Keralites, they eat their cook directly in the gravy. They cook their fish directly by boiling in the gravy, not frying. Frying attains a higher temperature, and boiling up to maximum 110 or 120 maximum. So that ways we can preserve some of the omega-3. Not exactly 100% will go waste. And eating raw has its own challenges. Our body over the time, over many generations, and over the age also has been tuned to take it cooked, because the raw ingredients need more digestive difficulties. The difficulties in digesting those will be more. So, and also the toxin accumulation part has also to be seen. So the traditional ways, I think, are better rather than the modern fads of frying or deep frying or packaged foods. And your third question, sir, I cannot recall. Could you please remind me one more time, sir?
I think let me just see. Refining of oils, of course, that you did not answer as of now, but I bring in one tiny question before I conclude, that I think we would like your next talk to be on nutrition during pregnancy, because that is the time the brain is being formed.
Yes.
So this is something that we totally ignore,
yes,
isn't it?
Yes.
And at the conclusion, I'd like to thank you from my side. It was a wonderful talk, and your attire is making me jealous. You are so well clad in the winter of Delhi, and we are tropically clad. Anyway, enjoy the winter. Over to you, Dr. Manju.
Very cool. Thank you, sir. Thank you so much, sir.
Thank you, sir. Thank you, ma'am. I have only one. It's not a question, ma'am. I really want to. There's a lot of hype about the PUFA going on. Any advertisement you take, any oil ad, anything, it's only PUFA they speak about. But the slide that you put about the right fat, it was really an eye opener. The importance of having MUFA, medium chain fatty acids, whose percentages actually should be more than PUFA in the fat content, daily required fat content. Really, that should be actually advertised. Actually, and coconut oil contains MUFA. And I come from Kerala. So I always used to tell that if MUFA, once upon a time, MUFA was considered to cause MI, should not take coconut oil, such things were going on, then all Keralites would have ended up with cardiac arrest by this time, no, ma'am. So anyhow, your slide was very interesting, ma'am, and especially the right fat and the percentage of fat, types of fatty acids to be included, and the trans fats, the harm they do. But the next thing I also support, like Sarkar sir, what he was telling, we don't have a method to find out what is the percentage of, we have to just believe 0% trans fat in whatever, whatever cover, whatever junk food that we contain, it will be written. But we don't have biochemists, we don't have any method to estimate the percentage of trans fat as per now. I don't know. So anyhow, that was very good. If there are any other biochemists, young budding biochemists, they can take it up and come out with a method to find out the amount of trans fat also, ma'am. With this, I really thank you, ma'am. It was a very important topic that you have selected, and we loved hearing you, ma'am. Thank you so much for spending your time.
Thank you so much, ma'am.
Yes, and thank you.
Thank you so much, sir, again, and all the dignitaries.
Yes, thank you, sir. So though it is on a virtual mode, I would love to honor you using a virtual certificate and 1 minute virtual memento, ma'am.
Thank you, madam. Thank you so much.
Next, we would like to honor our chairperson, Dr. Gautam Sarkar sir, too, with a certificate and a memento.
Thank you so very much.
Thank you very much, ma'am. Thank you, sir.
Thank you so much.
Delegates, we'll have 5 minutes, 5 to 10 minutes tea break, after which we'll be restarting our session.
Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology
Starting
Yes,
at what
sir.
time?
Yes, sir. Shall we start still?
No, the speaker is ready.
Madam, are you ready? Dr. Jamuna, madam?
Yes, sir, I'm
Are
ready,
you
sir.
ready?
Am I audible?
Yeah, yes, ma'am, you're audible. Shall we start the session? Good morning, everyone. Good morning, everyone. Thanks for joining the session back. The session is about the application of organoids in biomedical research. So before starting the session, I feel proud to introduce the chairperson. The chairperson is Dr. B. Vishnu Bhatt, Director, Medical Research, Professor of Pediatrics and Neonatology, AVMCH, Puducherry, former Director and Dean of Research, JIPMER. Sir has published several papers, around 390 scientific papers, and 71 book chapters. He has edited books on protein energy malnutrition, NICU protocols of JIPMER, and NNF protocols in perinatology. He has a H index of about 37, I-10 index of 82. He has more than 4,000 citations. He is the associate editor of Indian Journal of Pediatrics. He is reviewing articles for many national and international medical journals. He has received several awards, of which only very few is mentioned here. He was awarded for the B.R. Santana Krishnan Award for the best paper in Tamil Nadu, and he received awards from various medical forums, including the Nagasami Oration of National Neonatology Forum, Oration of Kerala Nokon, and other scientific societies. He has got lifetime achievement awards from the Pondicherry chapter of Indian Medical Association, CAMR Conference of AVMC, and Dr. Rita Sud Memorial Oration Medical Education in India. He has also obtained the World Health Organization Fellowship in Basic MCH in 1989 and in 1992. I feel very proud to welcome our chairperson, Dr. B. Vishnu Bhatt, sir. So over to you, sir.
Yeah, good morning to everybody and our speaker, Dr. Jamuna. And thank you, Dr. Ganesh, for pleasant introduction. And it is my duty to introduce our guest speaker, Dr. Jamuna Bai Ashwath Narayan. She is assistant professor in microbiology in JSS Academy of Higher Education in Mysore. Her special interests are in bacterial infection, biofilms, antimicrobials, and various anti-infective activities. She had done her MSc and PhD from the Mysore University and did PhD, which was funded by ICMR for her research project. She was in the UK in the Cardiff University, trying in the field of meganomics. She has 21 research publications, 18 book chapters, and about 1,300 plus citations. And she's an active research worker and has got several projects under her. I'm sure that she will enlighten us on this topic of using organoids in biomedical research. So, good morning, madam. Please start your talk, and at the end, we will respond with questions and feedback. Thank you. You can start now.
Thank you, sir.
Can I allow her to share the slides?
Sir, is my screen visible?
Yes, ma'am, is visible. You can make it full screen.
Okay. Good morning, everyone. Respected dignitaries and the chair, as well as the delegates. I'm Dr. Jamuna Ashwath Narayan. I'm working as an assistant professor in the Department of Microbiology at JSS Academy of Higher Education and Research. And today I'll be talking about the applications of organoids in biomedical research. And recently I've started working on organoids. So this is a new field to me also. And I'll try to share my knowledge as much as possible in this advanced translational study. So today I'll be talking about the definition and the key characteristics of organoids, the various protocols and the requirements to synthesize organoids in the lab. And then I'll be discussing about what are the tools and techniques that we can use for characterizing the organoids, be it its structure, the composition, and functionality. And lastly, I'll be talking about the application of organoids for disease modeling and in drug discovery, regenerative medicine, and for studying infectious diseases, as well as for the personalized or precision medicine. And later, I'll be introducing to the two advanced technologies which have been used to improvise the organoid research. That is the use of microfluidics, and the other one is the 3D bioprinting. So lastly, I'll be discussing the challenges and the perspectives that are required for the organoids research. So there are two main issues when it comes to biomedical research and drug discovery. So one is we use invertebrate as well as vertebrate models to study the diseases, and we extrapolate these, whatever we observe in the mice and other animal models, to human biology. And the problem with the animal models and human cell lines is that they have little resemblance to normal or diseased tissue. So when we are translating this research from the labs uh, uh, to the patients, then we can find that some of the times the drugs that we have been researching on are not working in case of the patients. So the pharmaceutical company every year faces billions of dollars of losses because the drugs that have been tested in the labs do not work in case of the patients. So they all fail in the different clinical trial rounds. So that is the reason where we need to see to that the animal models or any other disease model that we use in our studies are having the utmost resemblance to the normal or the diseased tissue. And the other concern with the use of animal experiments is the ethical concerns. And that is the reason nowadays even FDA as well as Indian the drug and clinical trial rules has said that as much as possible we need to minimize the use of animals for research, especially in drug testing. And they have been asking us to, especially the researchers, to use non-animals and human relevant models such as 3D organoids, organs on chips, and advanced computational methods to study the efficacy and safety of the drugs. So that is the reason almost all the researchers are now taking an interest in the use of organoids with their research. So what are organoids? So these are actually self-organized three-dimensional tissue that is typically derived from stem cells. And these stem cells can be of different origin. It could be a pluripotent one or a fetal or an adult stem cell. And these are known to mimic the key functional, structural, and biological complexity of an organ. So we can consider the organoids as a miniature version of the actual organs. And they might be somewhere in size of around 1 to 3 millimeter or less than that. So when we compare these organoids with the conventional 2D cell cultures as well as animal models, we can see that these can be used to enable patient specificity, and they can be used to recapitulate in vivo tissue-like structures as well as functions. So organoid cultures are considered to be more accessible for manipulation as well as for in-depth biological studies in comparison to animal models or the cell lines. And organoid cultures can be used for personalized as well as cell therapy and drug discovery research. So here is a feature of the Science magazine, wherein we can see that the entire special issue was dedicated to the research that was carried out related to organoids. So next is we need to know what are the key characteristics of organoids and how is it different from the cell lines that we use usually in our labs as well as the animal models. So these organoids, they are known for the cell type diversity. Here we can get organoids from multiple cell types, and they can be found in the corresponding organ or tissue. So the diversity allows them to replicate the complexity of any of the native organ. And the other thing is we can expect the organoids to grow in a three-dimensional structure. So when we are using the traditional cell lines, we can see them as a 2D structure. They are grown in a flat layer as a two-dimensional layer, whereas the organoids can have a 3D structure. So this helps in representing the in vivo environment of organs as well as the tissues. And organoids are known to exhibit self-organization. So they have the intrinsic ability wherein they arrange themselves into complex structures, and they don't need to be having any external cues for them to develop into a 3D structure. So in this way, they will be mimicking the natural process of an organogenesis. And the other important things about the organoids is that they can display functionality, wherein they are known to be able to, on induction, they are known to secrete hormones, absorb nutrients, as well as they can also carry out the contraction if you are using muscle organoids. And the most important thing about an organoid is we can directly take it from a patient specific cells, and we can also go for induced pluripotent stem cells. So this will help us in personalizing the models for studying the diseases and testing the drug responses. So nowadays we can see organoids being used for various applications, which include studying genetic disorders and cardiovascular diseases, cancer, and also to understand or elucidate different disease mechanisms, pathogenesis, and also to test new drugs, its efficacy and safety, and finally to develop personalized as well as precision medicine. So this organoid technology didn't evolve it took a long time to evolve, and it initially started with the studies on the ability of sponge cells to organize themselves into whole organism. Later they came to know that in the year 1944 the amphibian cells could be able to reaggregate after dissociation. And later, in the year 1961, embryoid bodies could be differentiating under in vitro conditions. And in 1981, we could see that pluripotent stem cells could be used to establish embryos in mouse models. And in 1987, they could see that when breast epithelial cells were grown with ECM extract, they were able to evolve into 3D ducts and ductules. So this was the first study which showed that organoids could be formed from different tissues of different organs. So similarly, in the year 1998, the embryonic stem cell line was used for developing human blastocytes. And from 2006 onwards, they started using induced pluripotent stem cells from mice fibroblasts. And in 2008, they developed gastroid organoids as well as the brain organoids, especially the cerebral cortex tissues. And in 2009, the gut organoids were developed, and these were derived from the adult intestinal stem cells. And in 2010 between 2010 to 12, they had also developed retinal organoids, and these were also transplanted into mouse. And finally, we now have different organoids from various organs, be it the kidney, liver, pancreas, prostate, and lungs. And recently, they have also developed snake venom gland organoids for the venom toxicology studies. So these organoids have been developing over a period of almost around a century. And now we have got organoids for different organs. So how do we synthesize these organoids? So we have some basic requirements, and these are: we need to have a proper cell source. We need to identify what are the growth factors that these cell source required, and most importantly is the matrix that supports the growth of these organoids, and the various physical as well as biochemical cues that have to be provided. And we also can integrate these cues in a uh, microfluidic platform. So the most common methods for generating organoids include: we need to embed these cells in a 3D matrix, such as the matrigel. So this will provide a structural support and allow the cells to organize itself into a organ-like structure. And the other major thing is the choice of the growth factors and culture conditions. So this will help us in going for differentiating the stem cells into specific cell types within the organoid. And we also need to uh, use either the induced pluripotent stem cell or adult tissue specific stem cell based on our requirements. So firstly, let us know about the cell source. So we can either go for tumor derived cells or induced pluripotent cells or the circulating stem cells. So here, to establish a tissue derived organoids or cancer organoids, we can go for tissue resident stem cell or a progenitor stem cell or a differentiated cell. And these are usually obtained through an optimized tissue dissociation method. And for the induced pluripotent stem cells, we can use them to derive the organoids by establishing the induced pluripotent stem cell lines and characterizing the starting cell. So these are the three different types of cell source. So next is the matrix. So matrix is important because it needs to be the growth of the organoids. And here we can see that there is the matrigel, collagen, and synthetic hydrogels, which are the different types of matrix components that are used to grow the organoids. So once we isolate the cells, then we can seed them into biologically derived sources such as the matrigel or a natural ECM such as a collagen or into a synthetic hydrogel. So the most of the researchers in the initial days used to use matrigel. So this matrigel is made out of laminin, collagen 4, entactin, perlecan, and various growth factors, and it resembles the basement membrane in its composition. So there have also been other alternatives apart from matrigels, such as the recombinant human collagen or synthetic hydrogels. So most of the natural matrigels are produced from proteins or polysaccharides, and these are known to address the batch-to-batch variation that is seen with the use of the matrigel. And we can also use synthetic hydrogels, and in the recent years, these have been emerging as a powerful tool to have an independent manipulation of the biochemical as well as the biophysical matrix, which can control the organoid features and also improve its functionality. So overall, an ideal organoid matrix should have a stress relaxing as well as highly dynamic in its biochemical and biophysical properties. So this will help in accommodating or controlling the changes in the development of the organoid structure during their culturing. So the next important thing is the growth factors and small molecules. So over the years, the researchers have established the various soluble factors that are required to support the growth of different types of organoids, be it the intestine, liver. So here in this table, you can see that the various soluble factors that are required for the different types of the organoids. So most of them are WNT, EGF, and HGF, and commonly used ones are BMP as well as TGF. So again, the soluble factors depends on the type of the organoids that we are looking for. So moving on to the physical cues. So these are mainly important to support and signal for the communication between the cells in the organoids. So they are also important to mimic the nutrient and waste diffusion in the basement membrane. So we have so usually when we are using the cell lines, we carry out them in the T25 flask or in the microwell plates. Whereas when we go for organoids, and if we want to have a proper nutrient and waste diffusion, as well as a good nutrient accessibility, then we can go for their growth in shaking cultures, spinning bioreactors, or suspensions under continuous agitation. So we can also use bioreactors, which will help us in monitoring the pH, temperature, oxygen, and glucose levels, and also help us in maximizing the mass transfer and minimizing the shear stress. So here again, the use of a microfluidic chip will help us in carrying out perfusion experiments, and also aid in the long-term culturing of the organoids. So the other ways in which we can improve the organoid culture is by using bioengineering methods. And one of them is the bioprinting technology, wherein we can use bio ink, which is made out of biomaterials, to create three-dimensional structures using the bioprinting device. And here we can use different biomaterials to have a biological geometries, and this will help in mimicking the native tissue in a layer by layer way. And then microfluidic system can also be used to create miniaturized cellular models, and they can resemble the organ physiology. And when it is resembling the organ physiology, we call them as the organ on a chip device. So the next is to understand the protocols which are involved in the synthesis of organoids. So we can have, as I said earlier, we can either use the induced pluripotent stem cell or tissue-specific stem cells. So the induced pluripotent stem cells are the reprogrammed adult cells. So either it can be the skin or blood cells, and they are being induced to become the pluripotent stem cells, and these can differentiate into any cell type in the body. Otherwise, we can also go for the usage of tissue-specific stem cells, wherein adult stem cells from a specific tissue can be used, and it can be used to generate organoids. For instance, if we take the intestinal stem cell, we can generate the intestinal organoids. And then the next step is to increase the cell numbers. So we call it as cell expansion. So here the selected stem cells are expanded in culture to increase their number. The third step is to move this into the matrigel. So we expand the stem cells or embed them into a 3D matrix. So this matrix is, as I told you, it is made out of matrigel or any other types of synthetic hydrogel. So the matrix provides the structural support and helps the cells to self-organize itself into a 3D structure. And then the next thing is to go for differential induction. So to differentiate the stem cells into the desired cell types found in that particular organ, we can go for the usage of particular or specific cultural conditions and specific growth factors. So this will help in going for a natural development process. So the fifth step is to let the organoids to go for self-organization. So here the cells will have the inherent ability to arrange themselves into a complex structure, and they don't need any external guidance for this. And they start resembling an organ-like structure. So this takes somewhere around 15 to 30 days to reach the stage. So once we get to see the organoids being developed, then we need to allow them for further maturation and development. So here we have to, at this stage, we need to supplement them with the essential growth factors and nutrients so that their growth and function is being supported. So once we know that the organoids are completely matured and developed, then we can go for their characterization using various techniques, which could be either immunostaining or for gene expression analysis, or we can also look at their cell types and functionality. So if we have to go for any specific applications, then we can also go for different kinds of modification. So we can also use gene editing technology, and this can be used to carry out any modification, and We can also go for changes in the cultural conditions. So this can also be used to enhance the functionality of organoids. So in the case of studying genetic disorders, we can introduce genetic modifications at this stage, and this can be carried out by using specific gene mutations associated with the disease that we are looking for. So here, if you look in this figure, so these are the organoids that have been developed from the TDCs. So here we take the tissue samples from the humans or animals, especially from the specific organs such as the gut and stomach. And then, so here we have shown the example of intestinal organoid. So we can take the intestinal tissue sample and then open it, wash it, and dissociate into smaller fragments of 2 to 4 meter in size, 4 millimeter in size, so that we can increase its surface area. So this dissociation can be further enhanced using enzymatic methods or by mechanical dissociation. So finally, we can get single intestinal stem cells or crypts. So after a number of washing and purification rounds, we can harvest these stem cells or crypts and seed them and generate them into organoid cultures for expansion. So similarly, we can also use induced pluripotent stem cells. So this technology makes use of the genetic engineering concept. So here we take the iPSCs and maintain them and expand them as clonal populations on feeder cells or in extracellular matrix. So here these start aggregating and form embryoid bodies. And later these are harvested as cell aggregates and then preserved cell-to-cell contact, and we finally get populations with higher variability. So these aggregates can be later induced through germ layer specification. So we can expect them to develop into endodermal spheres or mesodermal domes or neuroectodermal matrix. So once we have the organoids, then next is we need to characterize them for the structural and functionality. So for the structural characterization, we use a number of visualizing methods or tools. So this is very important in understanding their structure and composition. And it also helps us to learn about the cellular organization, morphology, and molecular characteristics. So the simplest technique that we have got is the use of bright field microscopy. So this helps us in gaining an overall or visualizing the entire morphology and structure of the organoid. So we can use a bright field microscope to observe organoids cultured in the matrigel, which is actually transparent in nature. So we can stain them with HND, and we can go for contrast and imaging. So it will help us to highlight the cellular structure. So similarly, we can use fluorescence microscope, wherein we can use immunofluorescence staining to visualize the specific proteins or the cellular structure. We can also use fluorescent dyes and markers to obtain a live cell imaging. Then there is the confocal microscopy, which helps us to get a three-dimensional image of the organoids. So this is due to the scanning of the organoid using focus lasers. So we can expect a high resolution 3D imaging of the organoids using confocal microscopy. So we can also use TEM. So here in TEM, there is so in comparison to confocal microscopy, we can go for the live imaging. In TEM and SEM, we go for the we have to so the organoids have to be dehydrated and they have to be fixed, and then we have to go for ultra thin sectionings. So these sections have to be then stained with heavy metals and they can be imaged. However, the use of TEM and SEM gives an high resolution imaging, and we can see a clear cellular ultrastructure. Similar to TEM, SEM also provides a surface imaging of organoids, and here the organoids are fixed, dehydrated, and coated with a thin layer of metal, and the electron beam focuses and scans the organoid surface and provides a a high resolution image of the surface properties of the organoids. And we can also use fluorescent live cell dyes. So this helps us in live cell imaging and studying the dynamics in organoids in real time. So using fluorescent proteins, we can track cellular events and observe the migration, proliferation, and differentiation over a time. We can also use reporter cells and incorporate these fluorescent reporters into the organoids, and over a period of time we can see how the different cells express the fluorescent protein. And finally, we can also go for functional imaging, wherein we use fluorescent indicators or dyes which are having specific functional aspect. For example, if you use fluorescent 4am, we can go for calcium imaging in the neuronal organoids. So it helps us to understand the neuronal activity as well as track the metabolic processes that are occurring in the neuronal organoids. So when visualizing the organoids, it's important to choose the most appropriate technique. And this again is dependent on the research objective and the kind of information that we are looking for. So we can also combine the different visualization methods to gain a comprehensive understanding of all the features of an organoid, be it its structure, composition, and function. So there are, apart from the visualizing methods, we also can go for gene expression analysis and protein expression analysis. And so here in gene expression analysis, we can go for RNA-seq or as well as RT-qPCR to understand the expression levels of specific genes, and also to identify the differentially expressed genes. So similarly, we can use Western blotting as well as ELISA techniques to go for detecting and quantifying specific proteins in the lysates of organoids, and also for identifying or quantifying the concentration of specific proteins in the organoid supernatant. And we can also use flow cytometry to dissociate organoids into single cell suspensions and go for studying single cells using the fluorescent markers. So this flow cytometry helps in both quantifying and analyzing individual cells within a organoid culture. So we can also use electrophysiological technologies, which will help us in understanding the electrical activities and action potential. So this has been mainly applied for studying the neural organoids. So in this figure, we can see that how organoids have been generated and the various tools and techniques that have been used to characterize these organoids. So we can see that the development of organoids, this is a pancreatic islet organoid, and it has taken almost around 30 days for the initial cells to, that is the endothelial cells of this organ, to establish itself into an organoid. And they have used the fluorescent stains such as the DAPI to visualize the different parts and to see the live and dead cells. And they have also gone for the gene expression studies and seen which are the most expressed genes. And they have also planted it into a diabetic mouse model, and they have seen for the different functional studies such as after the transplantation, what are the changes in blood glucose tolerance, as well as the plasma insulin level and the body weight. So here we can see that the organoids can be grown in matrigel, and they are exhibiting specific spherical shape, and these can also secrete hormones. So this helps in validating it. So by using immunofluorescence technique, we can see which are the different kinds of secretions they produce, be it the glucagon or stomatostatin or pancreatic polypeptides. So once these organoids are matured, they have got us surrounding the EC matrix around it, and By using the DAPI stain, we can clearly see which are the nuclei in the organoids. That is the blue colored structures. And so these islet organoids, when transplanted into the kidney capsule of the diabetic mice, we can monitor for their physiological indexes and see what are the different types of diabetic phenotypes they are expressing. So later we can again remove the transplants and we can go for observing the different types of effects of the blood glucose levels. So moving on to the applications of organoids in biomedical research. So we can see that they have been used for a number of applications, which includes modeling genetic diseases or in transplantation therapy, in stem cell and regenerative medicine, and to study cancer biology, and in drug discovery programs, as well as for the toxicology studies. So here is a picture of the Nature journal, wherein it was published in the year 2018. So they have collected the protocols that were related to the production of or synthesis of different types of organoids from various organs, be it the kidney, brain, and liver cells, as well as liver, as well as the retinal. So when we go for genetic disorders, organoids can be generated from either the patient-derived induced pluripotent stem cells, and it helps us in understanding disease mechanisms and also to develop the personalized therapy. So by using genetic editing tools such as CRISPR-Cas9, these diseases that are associated with genetic defects can be easily studied. So in the recent years, a number of applications have been related more on cancer studies. So the organoids can be directly taken from the patient tumor tissues, and these can be used for developing personalized medicine. So here is an example wherein the cancer organoids, that is from the livers, were used to study a particular cancer subtype. So the patient samples were isolated and from them the cells were isolated and organoids were cultured. So the different types of the liver cells were used for developing a 3D organoid culture. And further, once they developed the organoids, which included the DNA and RNA, as well as all the other important components in a 3D structure. So these were further expanded, and later the IHC staining and xenograft model was developed. And later they were also used for the DNA analysis to see which are the various tissues or organoid specific biomarkers that could be visualized. And they also did the drug dosing studies in this, and saw whether any of the drugs that are used are resistant or it can be further used for the treatment of the tumors. So using the study, they could also establish the IC50 curves for the one of the chemotherapy drug, that is gemcitabine treatment. So this kind of studies helps us also to understand the organoid sensitivity to drugs. And so as these are taken from the patient sample, so it is easier for the translation into and for finding the right dosage for prescribing to the patients the different types of the chemotherapy drugs. So moving on to the application of organoids in drug discovery and development. So these have been used for high throughput screening, wherein automated systems are used to screen libraries of compounds for their impact on organoid viability. proliferation as well as have specific functional readouts. And then we can also use them to study the toxicity of the organs on specific toxicity of the drugs on specific organs. So here the organoids can be exposed to different organ drug concentration, and we can go for observing the cell viability, morphology, and the overall organoid function. We can also use organoids for the pharmacokinetic and PD studies, and this will help us in analyzing the kinetics of drug uptake, the metabolism, and the resulting physiological responses within the organoid system. So this helps us in understanding how the drugs are absorbed, distributed, metabolized, as well as excreted in an organoid model. And the drug resistance over time can also be studied using organoids. So when we expose the organoids to continuously to drugs, so these can help in mimicking the long-term effects and help us in identifying what are the mechanisms that finally leads to the evolution or development of drug resistance. And then we can also use organoids for screening novel drugs and seeing if they have got these bioactive compounds have got any therapeutic potential. And we can also go for either novel drug candidates, or we can repurpose existing drugs and study their effect on the organoids. And using organoids, we can also go for combination therapy testing, wherein multiple drugs or treatment modalities can be initially tested on organoid. And this can help us in providing insight on the potential synergistic effect of multiple drugs. Organoids have been helpful in tissue engineering and regenerative medicine. So they have been used to generate functional tissues and organs for transplantation, and also to study how the tissue regeneration mechanism occurs. And they have been quite useful in generating specific cell types, and when they are derived from the pluripotent stem cells, and these have been used in the stem cell therapy. So here is an instance wherein the retinoorganoid sheets were developed, and these were generated in vitro, and they have been transplanted into blind mice, and these have been used to study how the transplant develops and grows. The organoids can also be used in studying the host pathogen interactions, and these have been used for studying viral infection, bacterial infection, as well as parasitic infection. So the pandemic saw to that there was an increase in the use of organoids to study the effect of SARS-CoV-2 and other influenza-causing viruses in the pluripotent stem cells that were derived from the lung organoids. So this kind of infection models in organoids helps us in understanding and getting a realistic representation of the host pathogen interaction, and it will help us to study the various complexities of the infectious diseases, and also to test any potential therapeutics in a controlled and physiologically relevant system. So how do we study this host pathogen interaction? So it again differs for the different types of pathogens that we are is of our interest. So if it's a viral infection, then we need to choose an organoid type that is relevant to the target virus, and it could be either an intestinal organoid for enteric viruses or a lung organoid for a respiratory virus. So we need to infect the organoids with the target virus, and this could be either done through direct inoculation or exposing the organoids to virus containing media, or we can also use viral vectors and go for transduction. So next is we need to optimize the virus titer to get an infection rate that will be mimicking the in vivo scenario. So we need to carry out titration experiments and determine the optimal ID, or the infectious dose. And we need to perform time course studies to observe the progression of infection. And at this stage, we need to harvest organoids at different time points and carry out their imaging for post-infection analysis. And then we can also go for immunofluorescence staining to see the viral replication, its transmission, and cellular responses in the infected organoids. And we can also look for specific viral antigens and cellular markers. And we can also go for quantification of the viral RNA or DNA in the infected organoids and see what are the viral dynamics that are occurring during its replication. And we can also go for the measurement of cytokine and chemokines. So this will help us in understanding the host immune response. And finally, we can also look at the real-time viral infection dynamics and cellular responses within the organoids by live cell imaging technologies. So a bacterial infection can also be studied in a similar way. And we need to infect the organoids with the target bacteria. And this could be by direct exposure to the bacteria or using specific infection models such as invasion assay. And we can also, over the time, assess bacterial replication and clearance dynamics by quantifying the bacterial load. And we can also go for histological H&D staining and see what are the changes that are occurring within the organoid structure and how the cells respond to bacterial infection. And by using the IHC, we can visualize bacterial colonization, the host and cell interaction, the immune responses within the infected organoids. And by secretome analysis, we can look for the different types of antimicrobial peptides, cytokines, and other immunomodulators that have been generated due to the infection. And we can also use electron microscopy to visualize the ultrastructural details and understand how the bacterial invasion and replication occurs within the organoids. So moving on to the application of organoids in neuroscience, there have been lots of major milestones that have been achieved in the development of the brain organoid technologies. So here we can go for developing organoids from neural stem cells, which are known to mimic the complexity of the human brain. And this kind of the brain organoids is very useful in studying the neurodevelopmental disorders as well as various neurodegenerative diseases such as the Alzheimer's and Parkinson. And it also helps in screening for new drugs to treat the various neurodegenerative and neurological conditions. So here in this figure, we can see that the cerebral organoids have been developed by embedding embryonic bodies into ECM. So we can take human-induced pluripotent stem cells, which has the freedom to self-organize. And we can get organoids containing diverse tissues from these. So we can expect them to grow into retina, forebrain, midbrain, inbrain, and choroid plexus. And in the initial stages, brain region-specific organoid culture, we need to use the progenitor cells, and this will help in going for further differentiation. So we can also use 2 or more organoids that resemble various brain regions, and these can be later fused to get an assembloid and can also be used to simulate the interaction between the different brain regions. So here is an example of wherein different the brain regions have been developed, and these are called as mini brains, and these have been generated from pluripotent stem cells. So we can expect them to have, we can expect cortical organoids or midbrain organoids and striatal as well as cerebral organoids. So it takes around 30 days for these to mature into proper organoid-like structures. Organoids that have been derived from the patient tissues can be used to test the drug responses. So these have been the key reasons for going for personalized medicine and also in developing patient-specific treatment strategies. So here we can also establish the biobank of organoids by taking patient cells and storing them for further analysis. And whenever the patient has to be treated, their organoids can be developed from the patient tissues, and these can be profiled and they can be tested for the various biomarkers, and they can also be tested for the different types of drugs that can be used. So the other important application of organoids is in understanding the organogenesis. So here we can use organoids to understand how an organ development and morphogenesis occurs, and what are the cellular events that govern the organ formation, and also what are the cell fate decisions that occur during the differentiation process during a development. So organ on a chip or is a technique wherein we combine both the organoids development with microfluidic technology. So here we have a controlled and dynamic environment which helps in understanding better understanding of the biological processes. So we aim to mimic the in vivo microenvironment of organs within a microfluidic device, and this has been mainly used for biomedical research, especially concerning the drug development as well as the disease modeling. So here initially we need to go for the usage of microfluidic channels. The microfluidic channels can help in mimicking the physiological conditions such as the fluid flow, nutrient supply, as well as waste removal. So it helps us to have a proper spatial and temporal control over the organoid microenvironment, and also help us in getting realistic physiological responses. We can also integrate the organoids within the microfluidic device. So this helps in creating a dynamic and functional model, and helps in monitoring organoid behavior, its response to stimuli, as well as its interaction with the environment. And when we go for perfusion technology, we can provide the organoids with continuous flow of culture media. So this will support the organoid growth and maintain physiological conditions for a longer period of time. So this is mainly useful for understanding the blood flow and nutrient delivery in vivo. And also this kind of technology helps in increasing the longevity as well as the functionality of organoids. We can also use co-culture systems wherein multiple organoids or cell types can be integrated within the microfluid device. So this helps in understanding the interaction between different organs and different tissues. So mainly this kind of co-culture system is useful for studying systemic responses and the effects of drugs and its metabolism in liver and other organoids.
Madam, can you try to wind up?
So here, yes, sorry, I'll get up soon. So, so here is an instance where an organ-on-a-chip platform has used been for cancer research. And lastly, I would like to tell about the application of 3D bioprinting. So 3D bioprinting is a technique wherein we deposit cells and biomaterials, which we call them as bioink, layer by layer, and this helps us in creating a complex 3D structure. So this 3D structure is useful for mimicking the architecture and functionality of the natural organs and tissues. So we need to select appropriate cell type based models for this, and we also use bioink, which is made out of the cells that we are using or that we need to prepare for the organoids with. We mix it with hydrogels and other materials such as alginate, collagen, and gelatin. So this will help us in creating bioink. So this bioink can be later injected through extrusion based or ink based and laser assisted printing materials, or we also can go for a lithography technique. And using a computer-aided software, we can design the 3D structure, and then we can use the bioprinting device to deposit the bioink layer by layer, and finally we can get a proper 3D structure. So we can also adjust the printing parameters, which will help us in getting proper cell viability and cell functionality. And once the organoids are developed, we can go for post-printing maturation. And these organoids can be used for also going for addressing the challenge of vascularization. So here we can include channels for perfusion within the printed structure or integrate endothelial cells to help in angiogenesis. So this 3D printing technology helps in getting a more controlled and reproducible method for creating intricate cellular structures. So some of the challenges that are present in the organoid technologies are the organoids are lacking in vasculature and immune cells. So, and the size is also a limitation. So by using co-culture technology with endothelial cells, we can go for vascularization in a near physiological way, which will also help us in increasing the nutrient exchange. And another problem is it takes a lot of time to mature, somewhere we expected between 15 to 30 days. So slow maturation is another problem. So this can be overcome by using small compounds which can induce their growth. And tissue engineering, such as the biofabrication techniques and microfluidic technology and 3D printing technology, can be used to introduce vasculature and angiogenesis. And by going for co-culture systems, we can have the advantages of studying them in temporal and spatiotemporal interactions and go for diverse cell population. And if we use the principles of organogenesis during embryo development stage, if we follow them, we can go for an higher order of cell differentiation in space and time. And it will help us in having concentration gradients and signaling centers during the shape-guided morphogens. So I'd like to thank the organizers for giving me an opportunity to share my knowledge on organoids and its application in biomedical application. And I would also thank my institute for the grants and UGCBS as well as New York Academy of Sciences and Japan AMED. So along with my collaborators, we are working on developing lung cancer organoids, and we are studying how the microbial biofilms are known to cause any implications or interferences or modulation when we are going for any kind of immunotherapy such as the CAR-T therapy. And currently in our lab, we are working on the use of organoids for developing antimicrobials. So it's related to how exactly the lung organoids can be used for treating or testing the different novel antimicrobials when they are infected with escape pathogens. And we are also looking at how oncogenic bacteria and their biofilms can modulate the tumor environment using organoids of of the breast and colon cancer when they are infected with one of the oncogenic bacteria known as the Fusobacterium nucleatum. Thank you.
Thank you, ma'am. Thank you for the much detailed and impressive talk. Over to chairperson.
Yeah, it was a interesting talk. You given a lot of information. And earlier we used to do this cell culture studies, animal studies,
Yes,
and then go for human studies. Now with the organoids, I think, can we bypass the initial steps and straight away go to organoids?
We can do that only in cases of if you are derived directly the patient samples, and when we are testing, it is easier to go for such a method, like especially when we are going for personalized treatment. Otherwise, I think we need to initially do the studies in cell lines, and then we need to translate them into the organoid cultures. So whenever there is a possibility of avoiding the usage of animals, then we can go for these organoid models. And the other thing is it is quite expensive. So it will take a long time for these to be considered as an alternative for animal studies.
So many equipments are required. Are you outsourcing your sample or everything you have done in-house?
So the visualization and all, we can carry it in our lab itself, and not much of outsourcing we need to do. because we have an in-house hospital and the patient samples are also available. And the only thing is when we are doing this, the viral and bacterial infection studies, so that is the problem, and we need to establish a different lab for that, because we cannot see to that the organoids are like getting contaminated with the bacteria. So we need a separate lab to study host pathogen interactions.
How much will be the cost of establishing this lab for organized? Approximately how much? Any idea?
So like as of now, I cannot.
Because scientists often do not deal with economics. Okay, it was a good presentation, and I'm sure that our participants were enlightened. And if there is any question from participants in the chat box, anything is there?
I have a question, sir. I'm Dr.
Ganesh, you want to ask anything?
No, sir, actually. Dr. Ravi Kumar will ask some questions, sir. He is online now. He's a panelist.
Okay.
Madam, very good talk, madam. This is Dr. Ravi from Department of Medical Biotechnology. So I have a few queries, madam, regarding the reproducibility and standardization. So what real challenge in it, madam?
So reproducibility is such a problem. So that is why it is better to go for the use of bioprinting or 3D bioprinting technology. So wherein we can program every time and we get the same kind of organoids that can be generated. So when we go in for more of an automation, and where so we are knowing exactly what volume of the materials we are adding for the seeding or for the production of a matrigel or a matrix, then it is easier to go for the reproducibility. Otherwise, yeah, batch to batch variations are observed.
So if you're going for a patient specific cells for a disease condition or disease modeling, that will be very, very challenging. It seems, right?
So usually also we go for PDX models only, right? We take them and we test them. So I don't think it should be much of a problem, sir. But as you said, like, yeah, we do get like the batch variation.
So in your slide itself, you have been explained about the challenges in vascularization as well as oxygenation.
The lack of immune cells.
Okay. And what about the scalability, madam? That is a transition from a small cell culture to the large scale production. What kind of challenges you will face?
Yeah, we need to use bioreactors in such cases, a closed bioreactor system. And so that would be the way of going up for the scaling.
Okay. So definitely it is an ethical issues will be raised, right? How you are managing it, madam? Ethical and legal consideration for this one. So what are the committees you have been framed for this one actually? You need a biosafety committee.
Ethical issues comes, yeah, when we are doing ethical issues comes because we need to take the patient samples. So only in such instances we have to take all the clearance. So any human samples we are taking, we need to get the clearance from the institutional ethics committee. And other than that, like, we are not doing any viral studies, but only bacterial studies. So it's not much of an issue.
So you don't need a biosafety committee, madam, for this one? Or you don't need any stem cell committee? Who will monitor us actually if you are doing this organized study?
So we need to present it to the institutional ethical committee.
Okay.
And when we are applying for the grant, also we write to them that we'll get the clearance, and then only we'll be doing these kind of studies.
Biosafety committee will be needed because you are dealing with the cells. No? Because always when you study disease and all that, there is always a question of biosafety, whether it's maintained or not. So that committee is automatically should be there in addition to the human ethics committee.
The institution has the committees, sir, our institute.
Let's be there, otherwise it will be a problem as he's raising questions.
Okay, ma'am. Thank you. Nice talk.
Thank you, sir.
So once again, we thank you for your nice presentations.
Thank you, ma'am. Thank you for the much needed presentation, which you told in detail about the organoids, key characteristics, generation techniques, and its applications. So I hope in future this is a much needed research actually for the clinical research. People will replace the cell lines to the organoids. Thank you very much. So although it's virtual and we would like to honor both the speaker and the chairperson, so we would like to honor the speaker first with the e-certificate and the memento. Next, we would like to honor the chairperson with the e-certificate and the memento. Thank you, ma'am. Thank you, sir, for joining the presentation. I thank each and everyone for joining. Thank you very much. Please be online for the next.
As our chairperson joins, ma'am, shall we start the session? Good afternoon to all the delegates joined virtually to this significant scientific session in BMSECON 2023. Your presence adds value to the collaborative spirit that defines this international conference. It's my pleasure and privilege to introduce chairperson of this session, Dr. Ponni ma'am. Dr. Ponni is serving as a faculty of medical biotechnology at Adabari Medical College and Hospital, Puducherry. The insights of her doctoral research leads to fetch not one, two prestigious postdoctoral fellowship from national agencies like SERB and UGC, post at esteemed Pondicherry University. She had published articles in high impact journals. Her expertise and experience in the epigenetics, cancer biology, computational biology, and animal behavior suits her chair to this session. Once again, I welcome all the delegates. Over to you, ma'am.
Sorry. Thank you, Dr. Tiago, for the nice introduction. And I welcome all the participants for this conference, for this online conference. Very warm afternoon to everybody. It is my privilege to introduce the speaker of the session, Dr. Rajashekharan Ramalingam, who serves as a professor with the Department of Integrative Biology, BIT Vellore. So he has over 17 years of teaching experience. His specialization is bioinformatics, molecular modeling, and drug design. He has authored over 100 publications, and it is important to mention that his data has been featured on the cover page of the Genomics Journal. So he has many ongoing projects, particularly from the ICMR. He has served as many administrative roles, such as assistant directors, division chair, research coordinator, etc. He's serving as a reviewer for several peer-reviewed journals. Due to unfortunately he could not join us today, but he has sent us a recorded session. I hope it will be useful for all of you. So the recorded session will be started shortly on the topic bioinformatics and medical health.
Everyone, I welcome you all to my presentation on bioinformatics for medical biology, focusing on the application of bioinformatics in the field of medical biology. I would take about 20 to 30 minutes to complete my presentation. Before moving on to the topic, let us see the molecular level meaning of health and disease. What is the meaning of health and disease in molecular level? The state of health relies on a harmonious balance of biochemical reactions within the body. Conversely, disease manifests as abnormalities in biomolecules, biochemical reactions, or biochemical process. In other words, healthy state means our system follows normal physiological response. Diseased condition means our system follows abnormal physiological response. Diseases are concerned, what are the major factors causing them? In this slide, I have mentioned the major causes of the disease. The major factors causing the disease include physical agent, chemical agent, environmental factors, infections, genetic predisposition, immune system disorders, nutritional imbalance, hormonal imbalances, and other factors like lifestyle choices, age factors, poor healthcare access, even occupational hazards, and other factors like social and economic factors, psychological factors, etc. So based on these factors, we can easily classify communicable and non-communicable diseases. With this general information, let us see what is medical biology and bioinformatics and how they support the medical field. Medical biology is a branch of science that focuses on the application of biological principles and techniques to the practice of medicine. It involves studying the molecular mechanisms underlying various diseases, understanding the structure and function of biological molecules, and using this knowledge how to develop diagnostic tools and therapeutic interventions. One of the key areas to this field is bioinformatics, which utilizes computational techniques to analyze biological data. Bioinformatics is crucial for predicting three-dimensional protein structure, enhancing our understanding of their functions and interactions. This knowledge is invaluable for drug discovery and design. In essence, medical biology forms the foundation for therapeutic interventions, offering profound insights into biological basis of diseases. It aids in target identification, guides drug discovery, and supports the development of personalized and effective treatments. Actually, the knowledge of medical biology is used to develop diagnostic tools and therapeutic interventions, but my presentation focused mainly on the role of medical biology and bioinformatics in therapeutic interventions, specifically drug discovery process. Okay, when we need to design new drugs, means when the existing drug shows resistance or if there are no drugs available for certain diseases. In that case, how do we design a potential drug, or how computation methods support for designing the potential drugs? Here you can see the role of bioinformatics in therapeutics, which means how to use computational methods for drug designing. That is called as CADD, which stands for Computer Aided Drug Design. In CADD, we must know two important keywords. One is target molecule, the next one is drug. The target molecule is a specific biomolecule in the body, often a protein, nucleic acid, lipid, or other macromolecules, which is involved in a particular biological process or pathway. Target molecules are the focus of drug development efforts because they play a crucial role in normal or disease-related physiological functions. In this case, bioinformatics are used to analyze genomic data, proteomic data, and other sort of biological data to identify potential target molecules. Abnormal targets may be identified by comparing data from healthy and diseased tissues, looking for overexpressed, mutated, or otherwise aberrant molecules. On the other hand, the drug molecule is a chemical compound designed to interact with a specific target molecule in the body. The interaction between the drug and its target is intended to modulate the activity of targets, which leads to a therapeutic effect. The key point on the slide is drug is a molecule that interacts with the target in the body, which brings the normal physiological response. The nature of target molecule is mostly a protein, to some extent nucleic acid or lipids or carbohydrate. Additionally, the biological targets, that is biomolecules, are called as biological data, that is DNA sequence, protein structure, etc. As we know well that how they are interconnected in biological system through replication, transcription and translational process. Though the protein is formed by translational process, there is a great degree of complexity in the formation of 3D structure. The degree of complexity means the formation of protein sequence by the combination of 20 amino acids, followed by secondary structure, super secondary structure and motif formation, then domain and tertiary structure formation, and then dimer or trimer or multimer formation of quaternary structure by the influence of the various forces like peptide bonds, hydrogen bonds, electrostatic forces, van der Waals forces and hydrophobic forces, etc. We get these biological data. What are all the databases available to retrieve the biological data for computational analysis? There are various databases freely available to retrieve drug target information from sequence to structure level, where DNA sequence, that is nucleic acid sequence, can be retrieved from NCBI or GenBank database or EMBL and DDBG database. The protein sequence can be retrieved from UniProt database. The 3D structure of protein can be obtained from PDB database. Position or the location of drug binding pockets in drug target, that is protein. 3D structure is certainly required. To get the 3D structure, we need to go to the protein data bank and download the PDB file, also called as the structural file. The PDB file stored the structural information in the form of Cartesian coordinates. Let's see how is the format of atomic coordinates in PDB file. The first column denotes atom, second column denotes numbering or position of the atom, third column denotes a symbol of the atoms, fourth column indicates a 3-letter code of amino acids, fifth column denotes a chain ID, sixth column denotes a position of amino acids, followed by X, Y, Z axis values are given in column 7, 8 and 9 respectively. The next one is how to visualize the three-dimensional structure of protein. Three-dimensional structure of protein molecule, we need the visualization softwares, example Rasmol, Volview, DeepViewer, etc. Using this software, one can display the protein structure in different models. The model include a space fill model, ribbon, strands, cartoon, ball and stick model and wireframe models. Each model has some advantages and limitations. For example, spacefill model represents a 3D volume and shape, but secondary structures, atomic level details cannot be identified by this model. Ball and stick wireframe models are generally used to display all atoms and connections among them, but difficult to identify secondary structure elements. On the other hand, ribbons, stand, cartoon models are used to display secondary structure element, but difficult to identify the types of atoms and the nature of bonds. Next one is target structure. The target structure is very important for the development of potential drugs. If the drug target structure is not available in PDB database, what is the alternate way to obtain the structures? The structure can be obtained with the help of structure prediction methods. There are 4 notable modeling methods currently available for predicting the 3-dimensional structure of drug target. One is homology modeling, also called as comparative modeling. Next one is threading, which is also called as a fold recognition. Third one is ab initio, and the fourth one is alpha fold method, which is developed recently based on artificial intelligence technique. Next, we will see what are the major steps involved in modeling techniques. 6 major steps involved in modeling the protein structure. The first step is to identify the template sequences and their corresponding structures from PDB database by using either a BLAST or FASTA search method. And the second step is to align or compare the target sequence and the template sequences by multiple sequence alignment to identify conserved and variable regions. Third step is to construct the fragmented model of target sequence using the conserved region of template structures. Fourth step is to replace the template side chains with model side chains. Here the model side chain means our target sequence side chains. Fifth step is to adjust the model for insertions and deletions. And the last step is to optimize the modeled structure using geometry optimization technique. The geometry optimization, also called as energy minimization. If suitable templates are not available, then the alternate way is to find out the distantly related templates and follow the last major steps, which I discussed now, by which one can model the target sequence effectively, or else by predicting the secondary structure and the solvation potential from the target sequence, based on which find out the structural fragments from fragment library, followed by fragment assembly and optimization process, by which we can model the target sequence effectively. Minimization is generally employed under model optimization process while modeling the 3D structure of drug targets. In modeling approach, the initially modeled structure may be restrained with improper geometry. The potential energy or the total energy would therefore be high and unstable according to thermodynamics law. In that case, the initial structure must undergo energy minimization process. Thereby, improper geometry will be converted into proper geometry and optimized structure will attain minimal energy and become stable. In essence, the true properties of a molecule, that is our drug targets, will be better predicted by the conformation with its lowest energy. Moreover, energy minimization process is completely based on molecular mechanics calculation, which requires force field. Here the force field, also called as energy function, which is used to compute the total energy by summing up the bonded and non-bonded energy of biomolecular system. I have mentioned the tools for predicting 2D structure and 3D structure from protein sequence, namely GOR, which stands for Garnier-Osguthorpe-Robson method, Chow-Fasman method. These two methods are used for predicting the 2D structure from the protein sequence, followed by the 3D prediction tools. Example, Swiss model, 3D PSSM, Threader, Rosetta Fold, CPH models, followed by structure validation tools. Example, What If, ProCheck, Verify 3D, and 3D structure visualization tools. Example, Deep Viewer, RASMOL, PyMol, BallView, etc. By using this tool, we can model and visualize the static structure. With the help of this 3D structure, we can compute the stability and the functionality of protein molecule using various bioinformatics softwares like FoldX, Stride, Profun, etc. One is drug resistance. Drug resistance poses a significant challenge in the field of medicine, as it limits the effectiveness of antibiotics and antivirals over time. To address this issue and slow down the emergency of drug-resistant strains, healthcare professionals are advised to prescribe these treatments judiciously. There are 6 recognized mechanisms through which pathogenic virus and bacteria develop resistance to drugs. The first mechanism is a mutation of organism may result in a change in the shape of drug target's active site. The second mechanism for drug resistance is some pathogenic bacteria contain enzymes that break down foreign compounds. And third one is there can be upregulations of the drug efflux from the bacterial cell. And the fourth notable mechanism is bacteria can undergo genetic changes that results in decreased cell wall permeability. And the fifth one is there can be changes in the pathogen's biochemical pathways. And the sixth one causing the drug resistance is some bacterial colonies can excrete carbohydrates that join to form protective biofilms. Overall, the reduced efficiency of existing treatment is due to mutations, which in turn leads to modification of drug targets that highlights the urgent need for development of new therapeutic strategies in order to combat drug-resistant strains. Based on these facts, let us see what are the main aims of drug designing. Aims of drug design. The first aim is to improve the activity by increasing the binding affinity between the drug and the target. The second aim is to reduce the side effect by increasing the drug selectivity for the respective target. Based on these aims, the drug can be designed by following the three major steps. Namely, the first step is target identification, validation, and characterization. The second major step is lead identification and optimization. And the third major step is preclinical and toxicological studies. The first step, target identification, involves finding potential disease-related molecules, while target validation confirms their relevance. Advanced technologies like genomics are used for identification, and experiments on model are employed for validation. These steps guide researchers in focusing on promising target for drug development, increasing the chances of creating effective treatments. And the next one is target characterization, which involves a detailed investigation into structure, function, and relevance of chosen biological target for drug development. This process employs advanced techniques like bioinformatics and molecular simulation to understand how the target operates in normal and diseased conditions. The acquired knowledge guides the design of drugs that can interact effectively with the target, paving the way for more targeted and successful therapeutic interventions. Moreover, I have also discussed the computational aspects of target molecule. Now let's see what is lead molecule and their sources. The lead molecule. Lead molecule is the starting point when designing a new drug. It should have some desirable properties that is likely to be therapeutically useful. There are various natural and synthetic sources available for exploring lead molecules, that is bioactive molecules. Example, from medicinal plants and trees by the reference of historical record, and also from animal source. One can explore many natural products and screen out the potential lead molecules. Another example, penicillin, tetracycline, or the class of antibiotics derived from the source microorganisms. One more example, curacin, which is one of the anti-tumor molecule derived from cyanobacterium. In certain cases, even from synthetic source, lead molecules can be designed. Example, Prontos dye. The drug sulfonamide is derived from this dye called as Protosol. Some synthetic molecules are natural product analogs, which are derived from natural products by various chemical treatments. Computationally, by virtual screening, we can identify lead compounds from chemical library database. To analyze the small molecule, that is a lead or drug molecules computationally, we need a database to store and retrieve the small molecules. Some of the notable databases are Drug Bank, PubChem, CSD stands for Cambridge Structural Database. In these databases, the chemical structures are stored in one-dimensional, two-dimensional, and three-dimensional formats. SMILES stands for Simplified Molecular Input Line Entry System, or INCHI, which stands for International Chemical Identifier, or the standard way of representing one-dimensional format of small molecules. Line structures are the standard way of representing two-dimensional format of small molecules, where atoms are denoted by symbols and bonds are represented by lines. Lewis structures and skeletal structures are common form of 2D representations. In three-dimensional representations, the spatial arrangement of atoms in a molecule is considered. The 3D format provide information about the three-dimensional geometry of a molecule, which is crucial for understanding its properties and interactions. Files like PDB or MOL2 files stores three-dimensional coordinates of an atom and a molecule. In some case, if your chemical structure of interest are not available in chemical library database, what is the alternate way to get the structure? In this slide, I mentioned some of the notable softwares like ChemSketch, ChemDraw, Morven, MolView, Open, Wapel, which provides the environment for the user to draw the 2D structure of small molecule and convert the 2D structure into 3D. After getting the 3D structure, mostly the structure may not have attained the proper geometry. In that case, we need to go for optimization process, that is energy minimization process. Some of the tools, example Gaussian, MOPAC, GROMAX, GROMOS, AMBER, are generally used to perform energy minimization process. During this process, the improper geometry of small molecule will be converted to proper geometry by altering the bond length, bond angle, and dihedral angle with the support of basis set or force field. Now let's see what are the major methods available for drug designing process. One is molecular docking and another one is de novo design. Molecular docking is one of the important computational method which is used to determine how a compound will bind to the active site of a protein. Molecular docking is also used to determine the orientation of the compound, its conformational geometry, and the scoring. The scoring may be a binding energy, free energy, or a qualitative numerical measure. In other words, the docking process involves prediction of ligand conformation and orientation within the targeted binding site. Generally, molecular docking is used for hit identification and lead optimization in drug designing process. Some of the open source docking software are Autodock, Argus Lab, PatchDock, etc. The next one is a de novo program, which dock a large number of simple molecular fragments into the active site and then connect them together to build molecules by following the three basic steps. The first one is analysis of active sites. The second one is building molecules. And third one is sorting or selecting of best candidates. So the molecular docking and de novo drug designing belongs to the category called as structure-based drug design or direct drug design. More important computational methods for designing the drug are similarity searching method and QSAR, which stands for quantitative structure activity relationship. The first one is a similarity searching method, which is also called as a virtual screening. This method is used to identify small molecules from chemical library database with respect to the input molecule. Specifically, pharmacophore-based similarity searching method focuses on identifying common spatial arrangement of chemical features in molecules, regardless of their overall structure. And the second approach, QSAR, it helps in drug discovery by predicting molecular properties such as activity or bioavailability or toxicity of new compounds without extensive lab testing. It saves time and resources by guiding researchers to focus on most promising candidates. Specifically, a three-dimensional QSAR is a technique for predicting how a drug will interact with an active site when geometry of active site is not known. There are four major steps involved in QSAR technique. The first step is data collection. Here the data indicates the structure of known compounds and their corresponding activities. The second step is feature analysis, means identify the key structural features that influence activity. Third step is model building, that is develop a mathematical model that quantifies the relationship between structure and activity. Fourth step is prediction, that is by using the model, the activity of new or untested compounds can be predicted. Similarity searching method and QSAR belong to the category called ligand-based drug design or indirect drug design. In what situation and with what kind of approach is appropriate? For example, if target structure is known and ligand is also known, then we can use molecular docking. Second condition, if target structure is known and ligand is unknown, then we can use a de novo design. Third condition, if target structure is unknown and ligand is known, then we can use pharmacophore mapping, QSAR, and ADMET prediction. The fourth condition, if target structure is known, if the target structure is unknown and ligand is also unknown, then computer-aided drug design will not support in this case. And moreover, in silico drug design or computer-aided drug design is broadly classified into two types. The first one is structure-based drug design, also called as direct drug design. Two important techniques comes under this category, namely molecular docking and de novo drug design. And the second one is ligand-based drug design or indirect drug design. Some of the notable techniques comes under this category, namely virtual screening, pharmacophore mapping, QSAR, ADMET prediction, etc. In drug designing process, generally two types of calculations are employed to explore the properties of drug targets and drug molecules, namely molecular mechanics calculation and quantum mechanics calculation. Molecular mechanics calculation is suitable for protein, quantum mechanics for drug molecules. At last, even after designing the potential therapeutic molecule through in silico methods, the next step is to synthesize the molecule with the support of chemists. Then the synthesized molecule will be subjected to preclinical trials and clinical trials. Usually, preclinical trials precede clinical trials involving laboratory and animal testing. Clinical trials progress through 4 phases, starting with healthy individuals in phase 1, followed by patients with target disease in phase 2 and phase 3. Phase 4 involves post-approval monitoring of the drug's performance in the market. The overall goal is to ensure the safety and the efficacy of the treatment throughout its development and use. This note, I close my presentation. Before leaving, I would like to convey my thanks to the organizers for giving me the chance to express my thoughts in this forum. Also, I wish to express my thanks to the audience for your valuable presence during my presentation. Thank you all once again.
I would like to thank Dr. Rajashekar for sending us over this wonderful presentation. That was a very nice presentation detailing every information regarding total modeling and how to screen drugs for a particular protein. And since this is a recorded session, I invite all the questions from the participants. You can put it in the chat box. We will get back to you with proper explanations from Dr. Rajashekar. Thank you everyone for joining us in this session. Over to the organizing team.
On behalf of BMSECON organizing team, it is our duty to honor the speaker, Dr. Rajasekhar Ramalingam, with a certificate and memento. It is also our duty to honor our chairperson with certificate and memento. Thank you, one and all.
Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future.
Zolga is there? Good afternoon. Can you hear me? Smani Shankar is there? Okay. Ovia. Good afternoon. So the Prashant Kumar is there? Prashant Kumar? Shanmugha Velan? Vijay is there, Manishankar is there, and Meera. So shall we start? Yeah, Sumati. These of the presentations are 7 minutes of kindly try to adhere to the timings. And that is first, I would like to invite now Aparna. Good afternoon, ma'am. Now we can start to. Ovia, just accept the panel list. We can start to share. Sumadi. Yes, please accept the panel list. Sumadi, please accept the panel list. You have declined the panelists. Please accept the panelists. Meera is there? Meera? So we will start the section. Just wait for 5 minutes. We are waiting for our judge. So we will get back within 5 minutes. Is Meera is joined? Meera Noyer?
Good afternoon, Dr. Rita. Are you here? Dr. Rita?
Good afternoon, ma'am. I'm here. I've joined it.
Okay. Anyhow, thank you for coming. It will be actually, the session will be moderated by Dr. Prithviraj. Hello, can you hear me?
Yes, ma'am. Yes, ma'am. I can
Yeah,
hear
yeah.
you.
I just wanted to welcome you, that's all. Dr. Prithviraj will
Thank
be...
you, ma'am. Thank you for the
Yeah. He will be moderating, ma'am. Thank you.
Good afternoon to all. On behalf of BMSECON 2023, I'm warmly welcome you to our today's section, YRS UG. So today we are having three judges. The first judge from Dr. Catherine is Associate Professor from Stanley Medical College, Chennai. And second judge from Dr. Rita, who is a Professor, Department of Biochemistry from Mahatma Gandhi, Puducherry. And the third judge is from Dr. S. Ravikumar, Associate Professor, Head, Department of Medical Biotechnology from AVMC. On behalf of everybody, I want to welcome our judges. So today we are having near 7 participants are there. So for the talk, we have allotted for 7 minutes and 2 minutes for discussions. So now the session is open. So the first presenter is Ovia. So Ovia, you can share your screen on your PPT. Ovia? Yeah.
Shall I start, sir?
Yes.
Okay. The most creative approach is the most simplest approach. A warm greeting to one and all present here. I am R. Ovia Vallabi, first year MBBS student of Velamal Medical College, Madurai, standing before you to present my study entitled Effect of Acupressure on Pain Perception and Pain Threshold on Patients of Rheumatoid Arthritis. Imagine that your joints are slowly pulled apart. Now imagine that you're not imagining it anymore. This is what kind of pain is felt by the people who are suffering from rheumatoid arthritis. It is an autoimmune and an inflammatory disease where our immune system attacks the healthy cells of our body by mistake, causing inflammation and painful swelling, because of which joint mobility is affected. There are medications available such as non-steroidal anti-inflammatory drugs, glucocorticoid, biological agents, where we also encounter the adverse side effects caused by them. So acupressure is a self-reliable and a cost-effective alternative suggested for this. Acupressure consists of pressing the acupuncture points using fingers instead of needles. Acupressure is effective in treating conditions like insomnia, headache, nausea, muscle tension, and pain. Our aim is to study the effect of acupressure on pain perception and pain threshold on patients of rheumatoid arthritis. Our primary objective is to assess the changes in pain threshold and pain perception before and after acupressure intervention. Our secondary objective is to measure the time taken for the expected changes in pain threshold after acupressure intervention. So it is an interventional kind of study set at Department of General Medicine, Velamal Medical College Hospital and Research Institute, Madurai. The study duration was around 2 months and the sample size was 50, where in the study population, we considered 50 rheumatoid arthritis patients of both the gender, where the study group is 25 and control group is 25. In inclusion criteria, we consider RA patients of both the gender around the age gap of 27 to 70. And in exclusion criteria, we consider the patient who is suffering from osteoarthritis, varicose vein, and having bruises around knee joint. So the procedure includes getting the clearance from the institutional ethical committee, then taking the bilingual consent from the 50 willing RA patients after explaining the study procedure in detail. Basic details about the patient was recorded in the proforma. Reading for pain perception and pain threshold before the acupressure intervention was taken. Then we moved with 2-minute interactive session where we made them feel comfortable and relaxed. Then we proceeded with acupressure intervention where stimulation of local points, namely the eyes of knee point, ST36, GB34 and GB10 was done. So these are the points, and describing the acupressure intervention in detail, the patients were lying on supine posture, made relaxed in the cot. The points and procedure were explained to them in detail. 2-minute massage was given in the shown point GB10, GB34 and ST36, and the edges of the inferior border of the patella was given a 30 seconds of acupressure. After which we took the post acupressure readings immediately after acupressure intervention. That was taken as the 0 minute. Then we took after 15 minutes, after 30 minutes, and after 45 minutes of acupressure intervention as the 15-minute, 30-minute, and 45-minute reading. So for the data collection, these are the tools used. This is digital algometer, and this is a visual analog scale. For assessing the changes in pain perception, we used this. This is visual analog scale. Moving to the results part, the data analysis was confirmed by the statistical tools like PAD t-test, ANOVA, and Duncan's multiple range post-HOC comparison. It is a paired sample t-test used to measure pain perception before and after acupressure treatment. Here the t-value is 17.282, which falls on the significant two-tailed test with the p-value of less than 0.001, which is of high statistical significance, indicating the fact that there is a change in pain perception after acupressure intervention. So here we have used ANOVA to know whether any variation has taken in the mean value of pain threshold before and after acupressure treatment between groups and within groups. For better understanding and pinpointing the correct value and the correct variation, we use Duncan's multiple range test. If you look at the mean value in the before treatment, it was around 109.812. After treatment, the 0-minute reading was 113. After treatment, that is 15-minute intervention reading was 116. If we see the variation, it is of slight variation type. That is why they fall under single subset. If we look at the 30-minute and 45-minute reading, it is of high variation type, and that's why they fall under another subset, indicating the fact that the given acupressure intervention is profoundly effective after 30 minutes of intervention. So the changes in the pain threshold levels are depicted in a line graph. Coming to the discussion part, stimulation of local points, pressure sensation is carried by A beta fibers to the cortical areas. They are well myelinated and have high nerve conduction velocity, and they release endorphin, norepinephrine, enkephalin, and serotonin. No more production of substance P. This causes block in the transmission of pain sensation to the sensory area, thereby modulating the gate control theory of pain. This acupressure techniques is also believed to have anti-inflammatory action as they release CGRP, calcitonin gene-related peptide, from the nerve endings and inhibit the release of IgE, immunoglobulin E, responsible for inflammation, thereby reducing the inflammation and causes muscle relaxation. This acupressure also enhances the blood circulation over the point of stimulation. So, considering the past thing, this is the first study to focus on the time taken for assessing the effectiveness of given acupressure intervention in patients of RA. This we consider as our strength. And we have worked in a limited population size, and we have considered RA patient with a history of other complaints as a single group. So we see this as our limitation and would like to explore in future. Our highlights are very marginal difference in pain threshold is seen. So our area of interest is to use dolorimeter to record the changes in pain threshold. Through this study, we come to a conclusion that acupressure has significant effect on pain perception and pain threshold in RA patient. So introducing criteria like postural variation, etc. during the recording session will help us to explore the hidden mechanism in detail. So coming to the conclusion, there is a significant increase in pain threshold where the p-value is less than 0.01 was observed. Along with significant decrease in pain perception, a difference of 4 score was seen in visual analog scale. Pain threshold levels increased significantly within 30 minutes of acupressure intervention. So the implications include it's a cost-effective and a self-sufficient, self-reliable alternative. One can do it anywhere and one can say the points so that anyone can do that for them. If acupressure intervention is practiced along with the prescribed drugs, then it is found very effective to reduce the inflammation and pain. So these are the references. Thank you.
Thank you, Ovia. Now the presentation is open for the discussion. Over to the judges.
Hello, Ovia.
Yes, ma'am.
Congratulations on your presentation. Can you just decrease the zoom of your slide a little? Your screen zoom. Okay, you have stopped screen sharing. That's not a problem. Good. You have selected acupressure rheumatoid arthritis patients who are coming to the OPD. Did you decide on the duration of disease in them based on any criteria, or you have not subclassified that?
Ma'am, we have considered only the acute patients, ma'am, like patients who are having rheumatoid arthritis for around 2 years.
So that you have taken as a criteria?
Yes, ma'am.
Okay. Everybody is above 2 years, but there's no upper limit. Am I right?
No, ma'am. No, ma'am.
Okay. Did you repeat it after your time duration? Did you follow up the patients? No.
Yes, ma'am. Actually, we could initially we could work on a small population size only, and during the study period, we were having our college as well. We were having model exams as well. After all the exams got over, I went to the OP to check for any progress, ma'am.
Okay. Were the patients satisfied? Did you get feedback regarding that? You have taken the pain threshold, that would be even better. Okay, it will add value to your study. But your presentation was good.
Thank you.
Continue to proceed to see whether this effect of your study is persisting even after some time if the patients regularly do this. Okay, good.
Sure, ma'am. Thank you.
Thank you so much. Is there any other questions? Shall I move for next participants? Thank you, Ovia.
Thank you, sir.
The next participant is Nivedita. You can share your PPT.
Ovia, can you stop sharing the screen, please? Sure, ma'am. 1 minute.
Nivedita, you can share your PPT.
Yeah, no, can you please share it again?
So the screen, I'm not able to see the full screen, sir. Will I be able to adjust that? Or is it like that only for me?
Okay.
I can see only the heading. Even for the previous.
You can little zoom out, I think so. It is from your device only you have to correct.
It's mine, okay.
Yes.
Well, okay.
Go for the full mode.
Yeah, I've gone for the full mode only, sir. It's in the full mode only.
Okay.
Okay, sir. Let them continue. I'm not
Okay. Go for full mode.
You.
No, you must just click before the full mode. This one. Okay, you can start.
Edit, sir. Okay, let them start.
Slideshow. Yeah. Thank you.
Can I start?
Yeah, you can start.
Good afternoon, one and all. I'm Nivedita Satish, currently doing my house surgency in BLMR Hospital. The title of my study is a comparative study of visual and cortical auditory evoked potentials in Parkinson's disease patients and age-matched controls to assess their cognitive decline. Parkinson's disease, as we all know, is a neurodegenerative disorder characterized by the loss of dopaminergic neurons. It presents clinically as TRAP, which stands for pill rolling type of tremors, cogwheel rigidity, akinesia, and postural instability. All these are motor manifestations of Parkinson's. It has been noted that cognitive decline, which is one of the non-motor feature of Parkinson's, is becoming increasingly common nowadays. Evoked potentials are basically neurophysiological tests that help us assess the functional state of various parts of our brain. Here, visual and cortical auditory evoked potentials are used. So VEP is nothing but the expression of the electrical activity of our visual pathway, right from the optic neuron up until the calcarine cortex. Likewise, CAEB is the expression of brain activity that occurs in the auditory cortex in response to an auditory stimuli. As people age, their brains tend to shrink. So cognitive decline is inevitable in anybody. But Parkinson's disease being a degenerative disorder, the cognitive decline occurs at an earlier pace in these patients compared to the normal physiological process. The P100 component of VEP refers to the primary positive peak that occurs about 100 milliseconds after the visual stimuli is given in any young, healthy individual. And likewise, P300 refers to the positive peak that occurs 300 milliseconds after an auditory stimuli is given. So both of these components, latencies and amplitudes, were measured and compared between both the study groups. So the objectives of the study were to measure the VEP and CAEP in Parkinson's disease patients, and to measure the VEP and CAEP in age-matched controls, and to compare the changes that was present in the latencies and amplitudes of both of these components in both the study groups, and to assess its value as a sensible and reliable indicator of early cognitive decline and in the PD patients by correlating with the MoCA scores. The study was an observational cross-sectional type of study conducted in the Department of Neurology of 2 months duration. 25 Parkinson's disease patients aged between 50 to 80 years and 25 age-matched controls were taken into the study. Both typical and atypical cases of Parkinson's disease were taken into the study, whereas people with severely impaired visual or auditory functions or any malignancies or other psychiatric or neurological conditions were excluded from the study. A minimum of 27 subjects were required in each group. Institutional scientific and ethical committee clearance was obtained, following which informed bilingual voluntary written consent was obtained from the patients. Demographic details of the patients and history was collected, following which the inclusion criteria was applied. The patients were then split into 2 groups, Group A consisting of the control group and Group B consisting of Parkinson's disease patients. VEP and CAEP was measured for both these groups, and the P100 and P300 values, their latencies and amplitudes were obtained for both the groups, which was then subjected to statistical analysis by descriptive statistics, chi-square test, Pearson correlation coefficient, and independent sample t-test. Following this, the results were obtained. The data collection tools used were Ellinger's Evoked Potential Analyzer, which was used to measure the evoked potentials, and MoCA, which is the Montreal Cognitive Assessment Score, used to screen their cognition. Moving on to results, a p-value less than 0.05 was taken as a cutoff for statistical significance, and such significant difference was seen between both the groups with respect to latency of p100 component of VEP. Similarly, the latencies and amplitudes of p300 showed significant change as well, and the MoCA scores. The MoCA scores had an inverse relationship with the latency of p300, indicating that as there was an increase in latency, there was a decreased MoCA score, indicating there was a cognitive decline. So moving on to discussion, Parkinson's disease has been proven to affect many sensory processing pathways in many studies before. This P300 component and P100 component of VEP and CAEP were thereby chosen to assess the cognitive decline in these patients. So the neuronal generators of P300 mainly included the frontal cortex, reticulothalamus, and medial septal areas. These areas have an important role in attention, memory, learning, and motivation. So when the latency of P300 was delayed, it could be an indicator, an early indicator of cognitive decline or executive function. It has also been studied that the retina has a significant amount of dopamine neurotransmitter. And as we know, Parkinson's disease patients have a depletion in dopamine. So they might have retinal abnormalities, which can cause aberrant VEP. It has also been proved that patients with hearing loss tend to dedicate more of their resources to auditory perception. So this might lead to detriment of their other cognitive processes, leading to dementia by exhaustion of cognitive resources. So there are studies which have compared, which have used VEP and CAEP individually to compare the cognitive decline. But this study is one of the first Indian studies that has used both VEP and CAEP together and assessed them with MoCA scores to assess the cognition between both the groups. Few limitations of the study include a small sample size, so the results could not be. The results would have to be correlated in the light of other factors like gender, age, etc. Yet another limitation of the study was that the number of subjects were unequal between the mild, moderate, and severe categories of Parkinson's disease. So to conclude, Parkinson's disease patients had longer latencies of P300 and P100 components of both CEP and VEP. The P100 values showed an increase in amplitude, but the P100 did not show any discernible change. And this delayed latency of P300 was correlated with MoCA scores, which showed an inverse relationship. So this latency delay could prove to be an effective indicator of early cognitive decline in these patients. So this can be used for early diagnosis and to some extent to assess the severity of Parkinson's disease and thereby to improve the quality of life in these patients. The future direction of the study could be categorization of patients into mild, moderate, and severe. And the categorization could be in such a way that the number of subjects in each categories remain equal. These are my references. Thank you for your time.
Thank you so much, Nivedita. Now the PPT is open for discussion. Over to our judges.
Nivedita, congratulations on your presentation. It was a very clear one. And I was about to ask you about the subgroups and about duration. I think you have already added that at the end of your slide. That's a part of your future plan. You have not matched or correlated along with the duration of the disease, is it?
No, ma'am. We just categorize based on the MDS, UPDRS.
You can actually do a correlation study also, how your latency and amplitude compares with the duration of disease. If you do a subgroup analysis, then you can proceed as you have done now. It's a good study. You can proceed like that further.
Okay.
Good.
Thank you, ma'am.
Any other questions? Okay. Thank you, Nivedita.
Thank you, sir.
The next presenter is Manishankar. Manishankar, you can share your PPT. Just go for full screen. Yeah, you are audible. Just click the full screen. Okay, go ahead.
Hi all, this is Manishankar S, first MBA student from Velaman Medical College Hospital and Research Institute, and here I am to present my study on the topic effect of hunger, satiety and exercise on taste and smell threshold. Hunger is the best sauce in the world. Taste and smell perceptions are said to be better when hungry. A more relatable quote.
Can you be loud, please? Not able to hear.
Hunger is the best sauce in the world. Taste and smell perceptions are said to be better when hungry. A more relatable quote would be Pasi Usi Ariyar. All of us would have heard our elders tell this.
Manishankar, sorry to interrupt. Just you make your mic little nearer to you so that
Closer to you.
you'll be audible better.
1 second, sir.
No, you are audible. Just keep your mic close to your mouth.
1 second, sir. I'll just change. Hello, am I audible now, sir?
Yeah, you have to speak very loudly. Yeah.
Hello?
Increase the volume. Okay, go ahead.
Hello, am I audible?
Yes, you are audible. You can share your screen. Full mode.
I'm unable to hear. Sir, am I audible, sir?
Yes, you are audible, Manisha.
Okay, sir. Fine. A very pleasant afternoon to all present here. This is Manishankar S, first year MBBS student from Velaman Medical College Hospital and Research Institute. And here I am to present my paper on the topic effect of hunger, satiety and exercise on taste and smell threshold. Hunger is the best sauce in the world. Taste and smell perceptions are said to be better when hungry. A more relatable slogan would be Pasi Ruchi Ariyadu. All of us would have heard our elders tell this. Going a step further, we would have also heard that only when we eat when we are hungry, it is good for our body. There's even a Tirukkural which states, Marandana Vendavam, Yakaiku, Arandiyadu, Attradu Potri Unan. Meaning, there would be no need of medicine to heal your body if you eat after what already eaten has been well digested. So, as already told, it is generally recommended to eat after what has already eaten been digested. But in the fast-paced modern world, people tend to eat according to the physical clock rather than the biological clock of hunger. When we are not in a state of hunger and our taste thresholds are increased, as a compensation, we tend to overeat. Overeating ultimately causes obesity. There are already previously proven studies that obesity further increases the taste threshold, thereby decreasing taste sensitivity. This vicious cycle keeps on going over and over again, causing more and more obesity and its related complications. Reversal of this cycle during hunger prevents overeating and thus obesity and its related complications. Acute aerobic exercise is also expected to have a similar impact as that of hunger. So the aim and objectives of our topic is, the primary objective is to assess and compare the taste and smell threshold in hunger and satiety. And the secondary objective is to analyze the impact of short-term exercise on taste and smell threshold from a state of satiety. The operational definitions. The taste threshold is the lowest concentration of a taste scent that can be detected, and the smell threshold is the lowest concentration of an odorant that can be detected. Sensitivity is inversely proportional to threshold. Methodology. The study is designed to be an experimental study, and the duration was of 3 months from July to September 2023. The study was conducted in the Department of Physiology, Velaman Medical College Hospital. The sample size was 100. The study population included first and second year MBBS students. The inclusion criteria, students of age limit between 18 to 22 years were included, of both male and female genders, with normal BMI 18.5 to 22.9 kg per meter square on the Asian limit. Exclusion criteria, patients suffering from hypogeusia, ageusia, hyposmia, anosmia, and upper respiratory tract diseases like common cold, sinusitis, or other respiratory infections, anorexia, bulimia, like eating disorders, and subjects who were on taste modifying drugs were excluded from the study. So the procedure, the students were informed about the research topic during class hours, and those who were interested were asked to enroll. 126 participants gave their consent for this study. Written consent was obtained, and after applying the inclusion and exclusion criteria, 100 subjects were recruited for this study. These subjects had their last meal between 6 p.m. and 7 p.m. the previous day. They skipped breakfast the following morning, and the first reading, that is the taste and smell threshold in state of hunger, was measured between 9 a.m. and 10 a.m. after 14 to 16 hours of fasting. And the subjects were sent to have lunch at 12:30 p.m. The taste and smell threshold in state of satiety was measured 1 hour after food intake. And the pre-exercise threshold was measured after 4 p.m. So the pre-exercise threshold was measured after 4 p.m. The subjects were asked to perform moderate exercise on cycle ergometer for 30 minutes. Post-exercise values were measured immediately after the acute aerobic exercise. Data collection tools. Taste threshold was measured using sip, spit, and rinse test. Smell threshold was measured using Sniffen's method. The data collection was done in MS Excel, and the results were analyzed during the repeated measures ANOVA. These are the results. First table shows the taste threshold in hunger and satiety. We were able to find a significant decrease in taste threshold in hunger when compared to satiety. Similarly, a significant decrease in taste threshold was found after exercise when compared to before exercise. These are the results of smell threshold. We were able to find a significant decrease in smell threshold as well after exercise when compared to before exercise, but no difference was observed in hunger and satiety. So these are our graphical values. This is the taste threshold in hunger and satiety. We can see a significant decrease in taste threshold in hunger. And here we can again see a significant decrease in taste threshold after exercise when compared to before exercise. Similarly, here this 8 denotes the time to the power 10 power times of dilution. So the smell threshold again decreased after exercise when compared to before exercise. Conclusion: sweet threshold decreases significantly in hunger and on performing acute aerobic exercise, and the smell threshold decreases significantly on performing acute aerobic exercise. Now, coming to the discussion part, hunger and exercise both are states of physiological stress, and in states of physiological stress, sympathetic activity increases. So this leads to increase of three major substances: serotonin, noradrenaline, and endocannabinoids. Serotonin, the taste cells express serotonin, and this modulates the responses in the primary gustative neurons by acting via the 5-HT3 receptors. Also, serotonergic neurotransmission in the gustative nucleus of solitary tract and thalamus cause a decrease in decrease in taste threshold during hunger and exercise. Secondly, serotonin and noradrenaline as a combined effect have been found to affect taste cell excitability by altering the ion channel functions. Next, endocannabinoids affect appetite and stimulate food intake via two main pathways. Either they directly affect the central systems or they enhance the sweet taste responses in the peripheral receptors. Along with it, also new studies on neural circuitry have shown that the reward centers are stimulated more in, when palatable food is given to a person in state of hunger than compared to a state of satiety. The effect of exercise on smell threshold are majorly due to 6 conditions. The swelling of nares, moisture vapor in the exhale air increases, and according to Schneider and Wolf, odor sensitivity was best in the presence of a red, swollen, wet mucosa, provided the nares were not blocked. Along with this, there was an increase in neurotransmitters, cerebral blood flow, improved functional connectivity, and the neurotrophins increased during exercise that caused and decrease in the smell threshold and increase in smell sensitivity. The highlights of our study is that though lots of studies have already discussed on the effect of exercise after a meal, very less has been studied about the effect of exercise before a meal. The limitations are gender differences were not considered and age differences were not considered. Future directions: different types of exercise like isotonic and isometric exercises can be performed and assessed, and differences in smell and taste threshold in different BMI can be assessed. Implications: increased sweet threshold during satiety causes overeating as a compensation for the reduced sensitivity. This can be prevented by eating only when hungry. Performing acute exercise when not in hunger also creates an advantageous condition as that of hunger. This can be used to control feeding habits in subjects with obesity and its related complications, or subjects with binge eating disorder, hyperphagia, dyslipidemia, etc. These are my references. Thank you.
Thank you, Manishankar.
Thank you, sir.
Now the PPT is open for the discussion. Both our judges.
Manishankar, I appreciate you for selecting a purely physiology topic. Can you discuss why you have differences between sweet and sour sensation results?
In our report, we were not able to find any significant differences in sweet and sour sensation, but previous studies have shown a difference in sweet and sour sensation. Probably because of the sample size was inadequate in our case, we were not able to find any significant differences in sour sensation.
Okay.
Any other questions? Thank you, Manishankar.
Thank you.
The next participant is Prasant Kumar. You can share your screen.
Okay, sir.
Thank you. All the best.
Ma'am,
You can share your PPT.
1 sec, sir.
Okay.
PPT தான் சார் இதா வர மாட்டேங்குது சார் வர மாட்டேங்குது சார் டாக்குமெண்ட்ல.
Okay. Maybe I can share your PPT.
Okay, sir.
Okay, 1 second.
அவங்களே ஷேர் பண்ண சொல்லிட்டாங்க சார்.
Just thank you. Was you able to see?
No, sir.
1 second.
Okay, sir. Okay. Okay, sir. Go ahead. Shall I start, sir?
Yes, yes.
Good afternoon to all. Myself, sir. Myself, Prashant Kumar Gandhi, DMLT second student, studying at Stanley Medical College. Today I am discussing about topic about effects of electric interference on routine biochemistry parameters. Next slide, sir. Background. Bilirubin, a degradation product of heme, is found to interfere in the estimation of variable analytes. Electric samples are still the most common cause of falsely decreased creatinine values in biochemical assay. This study is to be estimate the effects of high bilirubin samples on other analytes. Next slide, sir. Sir. Next slide, sir. Objectives to evaluate the interference of high bilirubin on estimation of glucose aspartate transaminase, alanine transaminase, alkaline phosphatase. Next, sir. Materials and methods. Study designs. Observational study. Sample size. 10 serum samples of bilirubin more than 20 mg per dl received in our 24 hours clinical biochemistry lab. Government Stanley Hospital was utilized to analyze AST, ALT, ALP, and glucose values. After quality control, check all the above of 4 analytes were estimated in Excel 640 auto analyzer. Glucose by glucose oxidase and peroxidase method, AST and ALT by IFCC without pyroxyl phosphate method, and ALP by AMP method. After checking the baseline values, the samples were repeated with 1 in 5 dilutions. Next, sir. Results. When compared with the baseline values, glucose was significantly increased after 1 in 5 dilution. Other analytes like AST, ALT, and ALP were not significantly altered after dilution. It indicates bilirubin has negative interference on glucose. Bilirubin has no analytical interference on AST, ALT, and ALP. Next, sir. This graphical presentation shows that the glucose has significantly increased after 1 in 5 dilution, and other analytes like AST, ALT, and ALP has no change after 1 in 5 dilution. Next, sir. Discussion. Interference of bilirubin is due to spectral interference and chemical interference. In spectral interference, due to observation spectra of bilirubin and chromophore produced in the reaction. In chemical interference is due to portion of the reaction intermediate is being destroyed by bilirubin and decreasing the amount of chromophore formed. Next, sir. Discussions. Negative interference of glucose is by chemical method. Bilirubin interference in oxidase peroxidase based test system. Proportional to its concentration, bilirubin reacts with H2O2 formed, resulting in lower result of glucose. Next slide, sir. From this study, we can find that there was significant increase in the glucose concentration after dilution when compared to baseline. This shows a negative interference by bilirubin in the estimation of glucose at increasing bilirubin concentration. ALT, AST, and ALP shows no significant interference by bilirubin at increasing concentration. Each laboratory should evaluate yttric interference on each analytes before releasing the reports of yttric samples. Next, sir. Limitations. Sample size is less in this study. Bilirubin value of more than 20 mg per dl was taken in this interference study. Whether the interference is present even in less than 20 mg per dl of bilirubin concentration is to be evaluated. Next, sir. These are my references. Next, sir. Thank you.
Thank you, Prashant. Now the PPT is open for the discussion. Over to our judges.
Prashant, it was a good study, commonly affecting parameters due to interest. Okay, in that study about less than 20 milligram per deciliter bilirubin that was available in the lab, isn't it? It is only about 20.
Yes, ma'am. I only take 20 mg per dl, ma'am, more than.
Okay. So what other parameters can be affected? You told because oxidase and peroxidase method, those analytes which use this method will be affected. What other analytes can be affected?
Others like cholesterol, uric acid.
Yes. Okay. So you chose only to do these 4 parameters for the effect of trick sample?
Yes, ma'am.
Okay. Okay, Prashant. Thank you.
Thank you, ma'am.
Thank you. Thank you, Prashant. Next,
Thank
we
you,
can
sir.
move for the next participants. Sumati, you can share your PPT on full mode screen.
Yes, sir.
Yes, sir.
ಇಲ್ಲ ಸರ್ ಸಾಕು ನಾವು
Yes, you can share. Sumati?
Yes, sir.
Kindly share your PPT.
Okay, sir.
That I will do, madam. Next slide, I will know. You can start with this. Next, I would like to invite Dr. PC.
Yes.
To share the screen.
Good afternoon, everyone. I'm Sumati, studying DMLT second year at Stanley Medical College. Here I am to present the comparison of thyroid function test in fasting and postprandial samples. This is an introduction. Level varies with the time of the day and probably in relation to food. In routine clinical practices, much importance is not being given to the fasting or non-fasting status of the patient. Study to show clinical significant differences in the interpretation of thyroid function test in relation to food. Aim and objectives to determine the effect of fasting and postprandial state on thyroid and free TF. Disease and materials and method. Study design. It is a cross-sectional study. Sample size 15. Specimen. Samples collected from OPD at Government Stanley Medical College and Hospital, Chennai, from patients who had came to give blood for fasting and postprandial glucose analysis were taken. Estimation of thyroid stimulating hormone and free T4 by CLIA method in Beckman Coulter DXI 600 immunoassay. This is a result. The man and standard deviation for TSH and FT4 were calculated. Here the mean is 22.57% differences is shown between fasting and postprandial, which is 22.57% low in postprandial samples. FT4 shows 8.3% low in postprandial samples. This is a graphical representation of TSH value. Here the red line shows the postprandial value, which is less after, TSH value is less after the intake of food. This graph shows the FT4 value. Here the FT4 value is decreased after food. Here is the discussion. The TSH secretion is stimulated by the hypothalamic pituitary thyroid axis. The THS secretion is inhibited by somatostatin, which is stimulated after intake of food. Here is the discussion. The lowering of the TSH postprandially can label subclinical hypothyroidism as normal and mislead diagnosis. It will also affect the diagnosis of hypothyroidism in pregnancies, where the cutoff value of TSH is 2.5 mu international unit per liter.
leads to the unnecessary repetition of testers, wasting resources. Conclusion. TSH level shows a significant decline postprandially. These may have clinical implications in the diagnosis and management of hypothyroidism, especially subclinical hypothyroidism. It is preferred to take sample for TFT in fasting conditions only. Here is the limitations. Timing of sample have not been addressed in our study. Larger sample size may be required to confirm our findings. These are my references. Thank you.
Thank you, Sunadi. Now the PPT is open for the discussion. Over to our beloved judges.
Okay, ma'am. Sunadi, this study is useful for our daily routine examinations. It was a good presentation. I just want to know if patients that you have, samples that you've been included, no, that come for fasting and postprandial, were they normal or did they have any thyroid disorders? How could you,
No, ma'am, we just used our routine samples only. Patients doesn't any complaints about thyroid functions.
Okay. Okay, there were no inclusion of any pregnant women and all. Only other routine patients were included. Isn't it?
Yes, ma'am. Only routine patients only.
Okay, ma'am. Thank you.
Okay.
Thank you, ma'am.
Thank you, Sunadi. Next,
Yes, sir.
next we can move for the next participants. Shanmughavelan, you can share your PPT.
Sir, can you hear me?
Yeah, I'm hearing you.
Good afternoon, ma'am, sir.
Maybe you can
Myself,
have,
yes, sir.
you can share your PPT on full mode, screen mode, whether it is. Okay.
Sir, it's a PDF, sir.
Okay, fine. You go ahead.
Okay. Good afternoon, sir and ma'am. Myself, S. Shanmughavelan, studied DMD in Stanley Medical College. Today, a presentation about the change of urine pH with time and observational study. Introduction. Routinely, we are measuring pH of urine sample in our hospital for various conditions. Delay in processing the urine sample can give erroneous result in the pH value, resulting in wrong interpretation and unnecessary intervention. Storage can lead to overgrowth of urea splitting bacteria, leading to high urine pH. The study was conducted to show the change in urine pH with time. Aim and objective. Determine the change of urine pH with time. Determine the correlation of urine pH and presence of nitrate in urine. Materials and method. Study design, observational study. Sample size 5. Specimen. Urine was collected from 5 male healthy volunteers. pH estimation. Using pH meter apt calibration with known standard. At 0 hours, 2 hours, 4 hours, 6 hours, and 24 hours. Samples are stored in room temperature. Nitrate reduction by urine dipstick. Manufactured from herba. Result. There are the pH value of 5 urine sample. It gradually increasing in 2 hours and similarly increasing up to 24 hours. In urine dipstick, 24 hours, presence of nitrate in light pink color. In 24 hours, presence of nitrate in dark pink color. Result. Increase in pH value was noted from 2 hours of collection as measured by pH meter. Change in color due to the presence of nitrate was noted from 2 hours of collection as tested by urine dipstick. Discussion. Whole blood pH is maintained within an extremely narrow pH range of 7.35 to 7.45 at the expense of urinary pH. Urine is able to achieve a hydrogen ion concentration 1000 times greater than blood. Its range of pH is 4.5 to 8. Definition of urine pH. Urine pH helps to diagnose metabolic and respiratory acidosis and alkalosis. pH is important for renal stone formation. It helps in the identification of crystals. pH helps in the treatment of urinary tract infection. pH is important for diagnosis renal tuberculosis. Discussion. Bacterial combination of urine with microorganisms that split urea may yield urinary pH value greater than 8 because of bacterial decomposition of urea to ammonia. Organism makes pseudomonas, splitting urea into ammonia to make alkaline urine. Conclusion. Prolonged storage of urine at room temperature is associated with alkaline change of pH secondary to bacterial degradation of nitrogen urine analytes. It is mandatory that urine sample received for pH measurement should be tested as soon as possible. There are the reference. Thank you, sir.
Thank you, Shanmugavela. Now the PPT is open for the discussion. Over to our judges.
Okay, sir.
Shanmugavela, the presentation was good, and this topic is kind of usually we see about the effect of hemolysis, citric sample, and lipemia in the blood, but in the urine you have seen. It is good to know that. I have one or two questions regarding your study. Can I know what are the other factors that can affect the pH of urine? Hello, am I audible?
Audible, ma'am.
What
other
factors can affect?
Other factors affecting urine.
Like
nitrate and liquid.
You have taken, like you have inquired with the patient about the diet or any other intake of medicines?
Diet.
Yes.
After food, ma'am. Ma'am. After food,
no, I'm asking
diet.
you, have you asked about diet can affect the pH of the urine, and then any medicine that the patient is taking, all that can affect, no? Like, have you inquired regarding those?
No, ma'am. Ma'am, no, ma'am.
Okay, it only pH was measured, or any other like routine urine microscopy and all was done for these samples?
Only pH measure, ma'am.
Only pH was measured. Okay. Thank
Yes,
you.
ma'am.
Thank you, Shanmugavela.
Thank you, ma'am.
So before going for the next participants, there is announcements there. Dear participants, you are requested to join for the validated function at 4 p.m. So the link will be shared in the group. So our last participants, Meera.
Yes, sir.
Okay, to share your PPT.
Yeah, sure. One. Is this visible, sir? Sir?
Yes.
Is this visible?
Yes, yes, you can start.
Thank you, sir.
Thank you.
Good afternoon, everyone. My name is Meera and I am from Department of Microbiology, JSH, Mysuru. I am talking about my project topic, antibacterial effect of silver nanoparticles synthesized from different microorganisms.
Super.
Thank you, sir. So these are the contents I included in this presentation. Silver nanoparticles are nanoparticles of silver between 1 nanometer and 100 nanometer in size. Silver nanoparticles have been of most important nanomaterials in biomedicine due to their unique physiochemical properties. The synthesis of nanoparticles include physical, chemical, and biological methods. Antibacterial effect of silver salts have been noticed since the antiquity. It is currently mainly used to control the bacterial growth in variety of applications, include dental work, cattle toes, and burn wounds, etc. It is well known that silver ions and Ag-based compounds are highly toxic to microorganisms, showing strong biocidal effects. As many as 12 species of bacteria include E. coli, Klebsiella, and Staph aureus, etc. The mainstream recognition of antimicrobial mechanism of AGNP include affecting the bacterial cell wall, producing the reactive oxygen species and damaging the DNA and killing the pathogenic organism. Silver nanoparticles. Silver nanoparticles are, as I told, they are the particles of silver, particle size between 1 and 100 nanometer in size. Then they like gold nanoparticles. Silver ions has long history and initially used to stain the glass for yellow color. Typically, they are synthesized by the reduction of silver salt with the reducing agent like sodium borohydrate or in the presence of some microorganisms with the colloidal stabilizer. After the reduction of silver ions into metallic silver, nanoparticles will be formed through the nucleation. Silver ions have been used for the long time for their antimicrobial properties towards wide range of microorganisms. Antimicrobial properties of AGNP against gram-positive and gram-negative bacteria by the mechanism such as attaching to the pathogenic organism cell membrane, affecting the permeability and respiration, and other mechanism is penetrating inside the bacteria and damaging them, and through releasing the silver ions. It can be synthesized by two methods: extracellular synthesis and intracellular synthesis. Extracellular synthesis. Extracellular synthesis, it involves the trapping of metal ions on the cell surface and reducing the ions in the presence of enzymes. And then the preparation of the extracellular reductase enzyme in cell-free supernatant. Then the cell-free supernatant mediated bioreduction of metal salts. Then the formation of nanoclusters to nanoparticles within the cell-free supernatant. Intracellular synthesis means it involves the transporting the ions into the microbial cell to form nanoparticles in the presence of cell. Instead of cell-free supernatant in this extracellular synthesis, here the cell directly to the cell we are applying. Then the positively charged metal ions entrapment on negatively charged cell wall. Within the cell wall, enzymatic bioreduction of metal salts. Then the nanocluster formation will happen and into nanoparticles are diffused into the cell wall. So the selected microorganism for this experiment are Saccharomyces cerevisiae, Saccharomyces boulardii, and Fusarium oxysporum. Saccharomyces cerevisiae are also called as baker's yeast, are found naturally in many environment niches and are used to biosynthesize monodispersed and more important stabilize silver nanoparticles. They are important in traditional or industrial fermentation for beer or wine, breads, etc. Also used as nutritional supplement and an agent to treat disease-related diarrhea. Saccharomyces boulardii, which is unstable to protect intestine against Clostridium difficile and cholera toxins, also clinically effective in preventing antibiotic-induced diarrhea. Fusarium oxysporum. These are ascomycete fungus, saprophyte in soil and organic matter. It is look like white cottony in morphologically. Based on their phytopathogenic property contributing to hunger and undernutrition in the world, some strains are beneficial to the host and can provide protection against the root pathogen. These are the materials used, media such as SDA, YEPDA for the synthesis of nanoparticles and nutrient agar for the antibacterial checking. Then microorganisms such as I told, Saccharomyces cerevisiae, Saccharomyces boulardii, and Fusarium oxysporum, chemical silver nitrate. This is the method for the synthesis of Saccharomyces species. AGNP from Saccharomyces species. Saccharomyces species are cultured in YEPDA media at 37 degrees Celsius for 48 hours. After sufficient growth, the broth is centrifuged at 6000 rpm at 4 degrees Celsius for 25 minutes. Then the silver nitrate is added to the collected supernatant and incubated for 48 hours in shaker at 37 degrees Celsius and observed for the color change from pale yellow color to brown color. Then the cell-free supernatant was subjected to UV spectroscopy in the, for the characterization of synthesized silver nanoparticle. Then the filtrate is centrifuged at 6000 rpm at 4 degrees Celsius for 25 minutes and washed with the distilled water to collect the pellet. Then for the antibacterial testing, E. coli and the Staphylococcus aureus culture was swabbed over the nutrient media and silver nanoparticles disk is prepared and placed as Kibbe Boyer disk diffusion method, then incubated at 37 degrees Celsius, then inhibition zone is observed. This is the method for the synthesis of nanoparticles from Fusarium oxysporum. Here, the difference is the Fusarium oxysporum is cultured in the SDA media, and after sufficient growth is filtered. Then the supernet and the filtering, it is not the centrifugation is not required here. It's just filtered with the Watchman filter paper. Then the supernet and is collected and the silver nitrate is added and follow the same method. These are the result we got. This is the Saccharomyces organisms grown in the media. Saccharomyces cerevisiae and bollardi are grown in the YEPD media at 37 degrees Celsius, about 48 of incubation. Sufficient organism is observed. The first A and B, A is the Saccharomyces cerevisiae and B is the Saccharomyces bollardi, and Fusarium oxysporum is grown in SDA media for the incubation period of 14 days. After sufficient growth, it is confirmed with a microscopic observation with the lactophenol cotton blue method. Then this is the synthesis observation for the synthesis of nanoparticles. After the addition of silver nitrate, sulfur filtrate color is changed from pale yellow to brown due to the reduction of age silver ions, which indicates the synthesis of silver nanoparticles. The color change is because of the interaction of Ag ions towards the mycelial cells, followed by the adsorption of ions and reductase. These ions due to the carboxylate groups in enzymes. This is the characterization we done. Ag and P were characterized by dual beam UV visible spectrophotometer by scanning the absorbance spectra in 200 to 800 nanometer range of wavelength. Average wavelength at which peak is observed 350 nanometer. Then further characterization we performed by FTIR, XRS, and DLS. FTIR is a versatile tool for surface organization of nanoparticles. By this, we can monitor the molecular change in microorganism exposed to nanoparticles. XRS is the to know the crystallographic characterization of the nanoparticles, and DLS we can done for the behavior of nanoparticles in the suspensions. This is the result for the antimicrobial activity. Antimicrobial activity synthesis nanoparticle evaluated against both the gram-positive, gram-negative bacteria such as E. coli, Staph aureus by Kirby-Boyer disk diffusion method. A zone of inhibition was observed against the both test organism. By this, we can say the silver nanoparticle is resistant to these E. coli and Staph aureus organisms. Silver nanoparticle can be continuously release silver ions, which may be considered the mechanism of killing the microorganisms. Conclusion is Saccharomyces cerevisiae, bollardi, and Fusarium oxysporum strains are cultivated in a controlled condition. The color change from pale yellow to brown observed in compound when compared to control due to the reduction of silver ions in the solution. Zone of inhibition was observed against both the test organism as a result, which indicates the synthesis of silver nanoparticles showing antimicrobial action against both test organisms. So it may be inhibit the disease caused by E. coli and Staph aureus. These are the reference I referred. Thank you.
Thank you, Meera. Now the PPT is open for the discussions. Over to the judge.
Hello, Meera.
Yes, sir.
My question is that you have mentioned that you have done FTR, XRD, as well as another one is you have done UV, right?
DLS. Yes, sir.
DLS as well as UV.
Yes, sir.
So what about the result part of it?
So actually, I gave that samples. I didn't get the result yet. So I just mentioned that.
Okay. So what is your future direction about this study? So why you want to prepare a silver nanoparticles from this culture?
So actually, I had a paper about this nanotechnology. So I got interested in that and I approached to that ma'am and I did my project on this paper. because it is actually an important topic in the future. We can prepare the antibiotic from the organism. There is a Saccharomyces cerevisiae, Boladi, and Fusarium oxysporum as easily cultivated microorganisms and easily available things we can do in like in our laboratory and all with a very easily limited controlled conditions. So I took this one.
No, actually, you have to look into the application part of your nanoparticles. Okay, so now the people are start synthesizing the nanoparticles in several ways, in several methodology. The novelty is very much needed for synthesizing this kind of nanoparticles. I'm just asking why you prefer this methodology. Why do you prefer these two microbes for your preparation process?
So as a microbiology student, first thing going to my mind is from microorganism itself. That's why I took this one.
Okay. See, actually, the nanoparticles have been prepared for encapsulating the nanoparticles. You can add some biological material or chemical material. Okay. You just go and check it in your literature or in paper. What type of phytochemicals are being produced by this bacteria? So these these quinones or some other phytochemicals, what happens? This will encapsulate the what do you say, the nanopart, I mean the what silver, so that pure application part of it will be get generated. Okay.
Yes,
So
sir.
whatever you prepare, you need some kind of background information. Okay.
Okay, sir. Thank you.
Thank you.
Thank you, sir. Thank you so much. Dear participants, we are so much thankful for your efforts and sharing your presentations and your research area on BMSECONs. We wish you all the success for your future and also for your research. Thank you. Lecturers are requested to share the evaluation sheet to the organization secretary on before 3:50 p.m. Thank you.
Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education, we established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education, we established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future.
Good evening, everyone. There is some technical glitch which we are fixing it. We will start the valedictory function in 2 minutes. Good evening, everyone. I welcome you all on this wonderful evening who have gathered here for this valedictory function of the 4th International E-Conference BMSECON 2023, organized by the Departments of Anatomy, Physiology, Biochemistry, and the Center of Biomedical Research. Knowledge is power, information is liberating by Kofi Annan, and education is the most powerful weapon which you can use to change the world by Nelson Mandela. With these thoughts, the 3 days of proficiency and tedious conference coming to an end. Let's begin the end with an invocation song of Tamil Thai Vazhuthu.
வாழ்வினில் செம்மையே செய்பவள் நீயே மாண்புகள் நீயே என் தமிழ் தாயே வீழ்வாறு வீழாது காப்பவள் நீயே வீரனின் வீரமும் வெற்றியும் நீயே தாண்டிடு நிலையினில் உனை விடுப்பேனோ தமிழ் என் நாளும் தலை குனிவேனோ சூழ்ந்தின்பம் நல்கிடும் பைந்தமிழன்னாய் தோன்றுடல் நீ உயிர் நான் மறப்பேனோ செந்தமிழே உயிரே நருந்தேனே செயலினை மூச்சினை உனக்களித்தேனே நைந்தாயனில் நைந்து போகுமென் வாழ்வு நன்னிலை உனக்கெனில் எனக்கும் தானே முந்திய நாளினில் அறிவுமில்லாது மொய்த்தனன் மனிதராம் புதுப்புனல் மீது செந்தாமரை காடு பூத்தது போல செழித்த என் தமிழே ஒளியே வாழி செழித்த என் தமிழே ஒளியே வாழி செழித்த என் தமிழே ஒளியே வாழி.
Now, I would like to invite Dr. Ganesh, Assistant Professor, Department of Medical Biotechnology, to give the welcome address.
Good evening to everyone. Respected Provost, Dr. P.F. Kotur sir, respected Advisor, Medical Research and Publications, Dr. Vishnu Bhatt sir, respected IAC Convener and Adjunct Professor, Dr. Pushpa Kotur madam, in absentia, Honorable Chancellor, respectable Vice President, esteemed Vice Chancellor, eminent Board of Management, distinguished Dean HP, Medical Superintendent sir, Director, Hospital Growth and Outreach Services, Vice Principal Administration, Vice Principal Academics, Vice Principal Student Affairs, Deputy Medical Superintendent Dr. Arnachalam sir, and Deputy Medical Superintendent Dr. Shankar sir, and other heads of institutions. It is with immense pleasure and honor that we welcome you all to this prestigious valedictory ceremony of 4th International Conference on Beyond Boundaries, Exploring Excellence in Basic Medical Sciences. Your virtual presence to ceremony brings a profound sense of significance and pride to this occasion. In the realm of virtual conferences, we have transcended geographical boundaries, allowing ideas to traverse across continents, enriching discussions and fostering connections that transcend physical spaces. Over the course of this conference, we have witnessed a kaleidoscope of perspectives, groundbreaking research, and innovative solutions presented by speakers, esteemed scholars, experts, and visionaries. Let the connections made during this conference serve as a springboard for future partnerships, groundbreaking research, and transformative ideas. I extend my heartfelt congratulations to all participants for your unwavering commitment and enthusiasm. Your dedication has truly made this virtual conference a resounding success. I welcome you all for this valedictory ceremony and making this event an unforgettable experience. Once again, a heartfelt welcome to each and every one of you gathered here and joined virtually. Thank you.
Thank you, sir. As Carl Benson said, the best attainable version of truth is a good report. For that, now I would like to welcome Dr. M. Gopinath, Professor and Head, Department of Physiology, Organizing Chairperson and Scientific Head of BMSC 23, to read the conference report.
We gather here today to conclude the 4th International Conference of Basic Medical Sciences, a remarkable journey of exploration, collaboration, and knowledge sharing. Over the past 3 days, this conference has served a platform for the exchange of cutting-edge research, fostering interdisciplinary connections, and advancing our understanding of fundamental medical sciences. I have this privilege to read the report of the conference. The wagon wheel has started way before 4 months. We had discussions to conduct this mega event in physical mode. But by considering the enormous, huge responses from our dear delegates for the past 3 years, we had decided to conduct this year also in virtual mode. We have received almost 890 registrations, which includes all categories like faculty, postgraduates, PhD scholars, and undergraduate students. The enthusiasm and confidence in which the delegates had on us made us utterly spellbound. We had received 212 abstracts for free oral presentations and 32 Young Research Scholar Award category presentations. So in total, we have received 244 abstracts for our scientific presentations and 40 judges all over India for providing their valuable contributions. On these 3 days, on the first day, we had Dr. Deepak Sarkar, distinguished professor from Rutgers University, USA, provided his keynote address. The conference was declared open by our Vice Chancellor, Dr. P.K. Sudhir, and which was presided by Dr. Rakesh Sehal, who has released our e-souvenir. We had almost 15 resource persons, in which 6 were international speakers from United States of America, Singapore, Malaysia, Korea, and 9 other resource persons from various parts of the country, like renowned colleges and universities, AIIMS, NITE Bangalore, VMMC New Delhi, NIT Calcutta, IIT Kharagpur, JSS Academy of Higher Education Research, Bangalore, VIT Campus Vellore, in wide variety of topics which touched upon cell signaling, neuroinflammation, polycystic ovarian disease, heavy metal injuries, gut microbiome and health and disease, immunohistochemical oncological studies, bioinformatics, and modern technology tools. So in the afternoon session on all 3 days, we had scientific presentations from 2 to 4:30 p.m. in all categories. The scientific presentations created a lot of insights in the minds of attendees in basic medical research. One of the unique strengths of this conference lies in its ability to bring together all the professionals from diverse fields within the basic medical sciences. The interdisciplinary interactions have sparked new ideas, collaborations, and potential avenues for groundbreaking research. As we could conclude this conference, let us reflect on the insights gained and contemplate the future directions of basic medical sciences. The seeds planted here definitely will have the potential to blossom into transformative advancements that will shape the landscape of medical research. May the knowledge shared here continue to ripple through the realms of research, healthcare, and leaving an indelible mark for the future of basic medical sciences. We extend a heartfelt thanks to each and everyone who has participated in this conference, a resounding success behind these enlightening days. Thank you.
Thank you, sir, for such a detailed report. Assessment of the performance is needed for improvements, and feedback is one of the best tools of assessment. For that, now I would like to get feedback from the participants of the conference, for which I'm going to invite at least one speaker. I would like to invite Dr. Prasanpriya Naik, Professor, Department of Physiology from AIIMS, Jodhpur, to give his feedback. So, Dr. Prasanpriya Naik, sir, you can unmute and you can give your feedback.
Good afternoon, everyone. It's really enjoyable one, and I congratulate the organizing committee and all the members and the institute. Last time during my lecture also I have mentioned that without a support from the institution, any organizing committee may not be able to do these type of activities. However, it was a fantastic one, well planned and well managed. I think only during valedictory I saw some glitches. Otherwise, it was so smooth running. Fantastic. Congratulations to all of you who are involved in the process. And also, I would like to congratulate the delegates who have attended, and I am sure they might have enriched their knowledge And thank you very much. Thank you for giving me this opportunity. And obviously, I would again mention the support from the institute. Thank you very much. Thank you very much.
Thank you, sir, for your feedback. Next, I would like to invite one of the chairperson, Dr. Vidya CS, Professor, Department of Anatomy, JSS Medical College.
Thank you, sir. Gives me immense pleasure to give the feedback of a wonderful conference. The level of expertise and knowledge of the resource person were excellent. In addition, I appreciate the positive attitudes, willingness to explain concepts, and opportunity to ask questions. The insights we have gained from this conference far exceed any other conference, and I have attended many other, but this one makes a benchmark of future conferences. Wonderful, insightful, very well presented, and excellent sessions, scientific sessions, challenging and thought-provoking for the young researchers, both for undergraduates, postgraduates, and as well as PhD scholars. I definitely learned new strategies from this conference. Congratulate the entire team for successfully conducting this BMSCon 2023. Thank you.
Thank you, ma'am. Next, I would like to invite one of the judge, Dr. Vani, Associate Professor and Head in Charge, Department of Biochemistry, JIPMER Karaikal. Okay, next I would, I think ma'am is currently unavailable. Next, I would like to invite Dr. Grishma, Associate Professor, Department of Physiology, to give her feedback.
Thank you very much for the opportunity. At the outset, I would like to congratulate the organizers for conducting such a wonderful conference. All the sessions were very informative and it was very interesting because the topics were diverse and it was very interesting. I had the privilege of judging an oral presentation, which I found was very well organized and also very well coordinated. Not only that, the time management was also superbly done. Overall, the conference was a very unique and a wonderful learning experience. Thank you once again. I'm looking forward to attending the next year's conference. Thank you.
Thank you, ma'am. Thank you for your wonderful feedback. Next, I would like to again invite Dr. Vani, Associate Professor, Department of Biochemistry, JIPMER Karaikal.
Yes, sir. Am I audible?
Yes, ma'am, you are.
Good evening, everyone. First, I would like to thank and congratulate the organizing team for organizing an interesting and useful conference. Actually, currently, relevant topics were covered by eminent speakers from all over the world, which will facilitate interdisciplinary research. Personally, I have learned a lot and I have got a good sense of latest development. I'm looking forward for such events every year.
Thank you, ma'am. Next, I would like to get an feedback from one of our delegates, Dr. Avinash Thakre, Additional Professor, AIIMS Bhopal.
Yes, good evening, everybody. First of all, I would like to congratulate the organizing team, the management body of the institute, the resource faculty members, all the chairpersons, all the guest speakers for organizing this wonderful conference. It is the third year, I believe, and I have consecutively attending the same conference for past 3 years. And yeah, fourth year. Okay. So I wish all the best and hope in near future we'll see such conference and more researchful and insightful extravaganza to come up. Thank you. Thank you so much.
Thank you, sir. Our final feedback we'll get from Dr. Shiv Leela, Professor, Department of Anatomy, Siddhartha Medical College.
Good evening, everyone. So it was a wonderful opportunity to attend the conference and beautiful scientific sessions. And this is the continuously fourth time I am attending, and every time the quality is topmost. I think AVMC can do that. And beautiful technical support and oral presentations and even young research scholar presentations were very good. Overall, a wonderful experience. Thank you.
Thank you, ma'am. Thank you all for this wonderful feedback. Now, I would like to invite our beloved and energetic provost, Dr. P.F. Kotu, sir, for his valedictory address.
Very good evening to all of you here. I've been the first-hand witness for the 4 conferences. Those have happened consecutively for the 4 years here in AVMC. And all this will give a very explicit messages. One, first and foremost thing is that virtual mode international conference is a successful model. This is what the result shows. One. Second thing is, with the joining of the hands of all the 3 departments plus the Center for Biomedical Research. And now we talk of integration, this and all this. This is another example of such a wonderful integration of all the basic medical science along with the basic science research. That is another successful model, you can say, which is AVMC. That means, and third thing, very message is the commitment and dedication of the organizers. Certainly, you can realize, can make any dream true. This is what is the honest feeling I have today. Even though, there are some, this time they said that from next time onwards, we're going to have a physical conference, this and that. I'm not against that. But this model, when a model is so successful, doing well, improving day by day, so many feedback from the participants, the delegates, the resource person, judges, or everything is there, then you have to consider again. And of course, the advantages of having a virtual conference are many, and in the nowadays fast world and all, it's outweigh the disadvantages. That is a different issue. So this is a wonderful model. When it is successful model, I think it should continue. And I could be a part of some of the lectures, especially Dr. Sarkar's lecture. So it's a wonderful input, wonderful experience, wonderful knowledge. They have been shared on this for the benefit of all the participants. And having people from across various continents and bringing them together on one platform, and it's a Herculean task. And now when you start going on repeatedly that, you develop some kind of a mastery. I must congratulate the team by all these anatomy department, physiology department, then biochemistry department, Dr. Manju, Dr. Lakshmi, Dr. Gopinath, then Dr. Rajan, Ilankati, then Frank. See, everybody have been doing the things automatically, So that's what is the success story of this. And I am very sure. And in fact, I am coming back from some official commitment in my head office. And there, they once somebody, some other institute, they asked, how come you are doing 2 to 3 conferences in a month? They said, yes. How means we're doing it? It shows that the commitment. If you dedicate and commit yourself, anything is possible. That's what is the message. So I'm very, very thankful to be a part of this successful conference and followed by the valedictory address. And this will be a kind of a message for all the participants, resource persons, faculty from across the globe. A conference of this magnitude can be conducted successfully. It will bring in many confidence in the people. So with these few words, again, I thank and I hope to be a part of this even in the 5th year. AVMC has achieved one more milestone, I can say. Now the institutional character is the guaranteed or it is branded. Thank you very much. All the best for all of you.
Thank you, sir. Thanks for the supportive and encouraging words. So it's nice to get appreciation from you, sir. Thank you, sir. Next, I would like to invite our ever young, all loving Dr. Vishnubhat, sir, Advisor, Medical Research and Publications, to share a few words on the conference.
Respected Provost Dr. Kotur, the research persons from India and abroad, the organizers who have taken a lot of pain in organizing this program and the delegates. It is a great occasion. See, COVID has brought many bad things, but one good thing probably we have started taking online classes. We started conducting online conferences, and there is nothing like sitting in your own place and listening. See, now I am traveling. I can talk from wherever I am available. So it's a great thing. And getting nearly 900 delegates is not a small thing. Moreover, you could mobilize speakers from all over the world. And I was listening for most of the program on all the 3 days. And it's an occasion that we can listen to many people who are talking from their experience. And I'm sure that they would have stimulated the young minds and the faculty members to do more advanced research and also do better for the future of the country and the well-being of the people. I'm sure that it will be a memorable event for all those who have participated in this program, and probably they may start thinking of conducting 2 e-conferences every year rather than doing one, because I think all over people can assemble, learn from each other, and I wish everybody all the best. So I'm sure that it will go a long way, as Dr. Kotur said, in the history of AVMC, as well as in the history of the basic science research. Wish you all the best. Thank you for inviting me to talk.
Thank you, sir. As usual, ever loving words from you, sir. Next, success is not in the trophy or paycheck, but in the satisfaction of a job well done. Those said, appreciation is the biggest motivation of running forward, and appreciation is a wonderful thing. It makes what is excellent in others belong to us as well. Hence, it's time for appreciation. Now, we had Young Research Scholar Award presentations for faculty, postgraduates, and PhD candidates and undergraduates. So for that, I would like to invite our Provost P.F. Kotu sir to present the YRS to read away the e-certificates of the award winner.
Yes, what do I do, Frank?
Sir, I will Sir, I will share the PPT now, sir. You can just read the names out.
Yes.
Maybe it is difficult to read, otherwise we can
Yeah, I can. Yeah.
Yes, sir. You can.
Yeah. No, I'm very happy to declare Young Research Scholar Award faculty to Dr. P. Ati Sankaran, Assistant Professor, Department of Biochemistry, JIPMER, Karaikal. Then Young Research Scholar Award for a postgraduate, Dr. Parinit Sandhu, Department of Anatomy, Government Medical College and Hospital, Chandigarh. Oh, great. Congratulations, Dr. Parinit. Then Young Research Scholar Award for postgraduate, Miss Yelaki Ayam, JIPMER, Pondicherry. Young Research Award for a PhD scholar, Anamika Sekhar, Department of Medical Biotechnology, JIPMER, Pondicherry. Young Research Scholar Award for an undergraduate students, Dr. Mani Shankar S., Department of Physiology, Velamal Medical College and Hospital and Research Institute, Madurai.
Sir, he's a first year MBBS student, sir.
Oh, excellent. Congratulations, Mani Shankar.
that's it for the YRS presentation, sir. Thank you, sir.
Thank you.
Next, I would like to invite Dr. Vishnubat, sir, to read away the certificates for oral presentations and all presentations for the Department of Anatomy, sir.
This is a young research award already presented. Hello.
So this one you can read it out, sir.
Jai Prabha Banu. Hello, senior resident Jipman Pondicherry. Section 1. Is it the award what you are projecting?
Yes, ma'am.
Dr. Jyoti Naik from Karnataka. Hello.
Yeah.
I'm not able to read the topic. Or
Sir,
you just announced the topic?
Sir, need not, sir. Just the name and
Dr.
designation.
Priyanka.
Hello. Dr. Priyanka.
Dr. Priyanka.
From Jipman Pondicherry.
Vinuta.
And Dr. Vinuta, assistant professor from Medical College, Mysore.
Okay, sir. Topic, Dr. Ilankar sir will lead out the topic, sir. You can just read the name and the designation, sir.
Dr. Priyanka Clementia Stephan from Jipman Pondicherry, bilateral bifurcation of external carotid artery concurrent with variation in auricular development now and middle meningeal artery. Next is Dr. Vinuta SP, assistant professor, JSM Medical Mysore, on omphalocele and gastroschisis. Hello.
Yes, sir. Next slide, sir.
Dr. Vijay Kumar, assistant professor, Symbiosis Medical College for Women and Pune, India. I think his study is on effects of weight reduction and therapeutic exercise in reducing the severity of the flat foot, an observational study. The next award, session 6, Dr. Royal Aravind Kazar, assistant professor, Dr. B.C. Roy, Multi-Specialty Medical Center, IIT Kharagpur. His topic is on service learning as a trigger for self-directed learning.
Next slide, sir.
Session 7, Dr. Kalpana Ramchandra, professor and head, Sri Ramchandra Medical College and Research Institute, Chennai. Topic: an immunohistochemical study of CDR3 positive dendritic cell density in human being and malignant lesions of cervix. Session 8, Dr. Rituraj Majumdar, senior resident, Jipman Pondicherry. Topic is on morphology of first extensor competent tendons and potential rollout in first carpo-metacarpal joint arthritis. That's all.
Yes, sir. That's all, sir. Thank you, sir. Thanks for your giving
With
away
a lot
the
of difficulty, I was reading. The letters are small.
Yes, thank you, sir.
Congratulations to all the people who have got the award. I find a lot of JIPMER faculty and residents in the award list. And congratulations to everyone.
Thank you, sir. Next, I would like to invite our organizing chairperson, Dr. Ilangadar, Professor and Head, Department of Anatomy, to read out the certificates.
Thank you, Dr. Frank. It is for the oral presentation for the postgraduate session. On session 1, we have Dr. Lencham Deepak from Regional Institute of Medical Science, Imphal, on a topic ethical and affordable cadaveric bones procurement technique for advancing research initiatives. From session 2, we have Nitya D from JIPMER Puducherry on a topic morphological analysis of ligamentum mucosum of knee joint, a cadaveric study. From session 3, we have Dr. Nandini Rajaram from JIPMER Puducherry on a topic assessing the neurovascular pattern of the triangular fibrocartilage complex of the wrist joint, a cadaveric observational study. From session 4, we have Dr. Lekha from Government Medical College, Nalgonda on a topic, an evaluation of carrying angle of elbow joint in adolescents. With session 5, we have Dr. Hira Ahmad from Lady Hardinge Medical College, New Delhi on a topic, tortuous course of the peripheral arteries of the upper limb, its clinical correlation. For the PhD scholar, we have on session 1 with Mr. Ruby Mehta from Mahatma Gandhi Medical College and Hospital, Tapur, Jaipur, on a topic variation in the location of an ovale with respect to lateral pterygoid plate and its clinical implication. With session 2, we have Ms. Nagajyoti from Pondicherry Institute of Medical Science on a topic clinical and cytogenetic profile of disorder of sex differentiation and analysis of 85 patients from Territory Hospital in Puducherry. With to the e-poster undergraduate, we have with session 1, Dr. Samarin Bala from Government Medical College, Srinagar on a topic dermatographic study in relation to intellectual status of Down syndrome.
Thank
That's
you,
all
sir.
we have for anatomy. I acknowledge each and everyone who has participated. Thank you.
Thank you, sir. Thank you for giving away the e-prizes, sir. Next, I would like to invite Dr. Pushpa Kotur ma'am for reading the e-certificates for the Department of Physiology.
Am I audible?
Yes, ma'am, you're audible, ma'am.
The prize for oral presentation in physiology goes to Dr. Avinash Thakre, Additional Professor, AIIMS Bhopal. And another prize goes to Dr. Aisha Jyoti Juhi, Aisha Juhi, Associate Professor, Department of Physiology, AIIMS Devgarh.
Next slide.
Dr. Usha Dhanaradia, JIPMER Pondicherry. So congratulations to all these people. Mr. Anwar A.S., Yanapoya Medical College, Mangalore, Karnataka, where I work for short time. Congratulations, Anwar. Mr. Siddharth Malya, Department of Physiology, Velamal Medical College and Hospital Research Center, Madurai, Tamil Nadu. Congratulations.
Thank you, ma'am. That's for physiology. Congratulations to all the winners. Next, I would like to invite Dr. R.R. Kumar to give away the prizes for biochemistry.
Thank you, Dr. Frank. So oral presentation faculty, session 1, Dr. Shafalya Naik, Assistant Professor, SCB Medical College and Hospital, Cuttack. The topic is association of iris with insulin resistance in type 2 diabetes mellitus. Session 2, Dr. Vilma Delphine Sylvia, Professor and Head, Akash Institute of Medical Sciences and Research Center, Bangalore. Topic, it is establishment of biological reference intervals of thyroid hormones for preschool children. Oral presentation for the postgraduate, session 1, it is Dr. Sai Sarani, Nizam's Institute of Medical Sciences. Topic is association of serum lactate dehydrogenase with international staging system in patients with newly diagnosed multiple myeloma. Session 2, Dr. Renuka Devi M, Stanley Medical College, Chennai. Topic is reliability of blood gas analyzer for electrolytes, a comparative study. Session 3, Dr. Nandini J. Mohan Kumaramangalam Medical College, Salem. Topic is study of thyroid function and vitamin B12 levels in beta thalassemia patients. Session 4, Dr. Sangeeta Ghosh, Government Medical College, Nalgonda. The topic is impact of laboratory personal training on pre-analytical errors and laboratory reporting. For PhD scholar oral presentation session 1, Dr. S. Tejasvi Pulkanam, AVMC, Puducherry. Topic is investigating the impact of chemotherapy on hematological and biochemical profiles among breast cancer patients at tertiary care teaching hospital. E-poster for undergraduate, Dr. G.V. Soumya, third year MBBS, Savitha Medical College, Kanchipuram. Topic is Shankan-Poor syndrome, measuring prevalence among patients with normal renal function test.
Thank you, sir. Next, I would like to invite Dr. Deepthi Shastri, Deputy Dean, Vinayaka Mission, Kripananda Warrior Medical College, Salem, to give away the e-prizes for Biomedical Research Department.
Good afternoon, everyone. It's a pleasure to announce the winners for oral presentation, faculty in biomedical research, session 1. The winner is Dr. S. Mohan, Assistant Professor, Department of Biochemistry, AVMCH, Puducherry, and his topic was the association between PITX2 and leptin receptor gene polymorphism in obese young adult population. For oral presentation, postgraduate and PhD scholar, session 1, it is K. Aishwarya, postgraduate, Department of Community Medicine, Sri Satya Sai Medical College and Research Institute, Tamil Nadu. And the topic was need assessment and solution exploration on palliative care among community dwellings in Tirupurur, Chengalpattu district, Tamil Nadu. And the winner for the e-poster undergraduate, session 1, is Miss Jia Khanna, Savitha Medical College, Savitha University, Chennai. And her topic was measurement of surface roughness of implants fabricated using polyether ketone peak and compared with grade 5 titanium. Congratulations to everyone.
Thank you, ma'am. Thanks for giving away the e-prizes. Next, acknowledging the good that you already have in your life is the foundation for all the abundance, Eckhart Tolle. Hence, I invite the most apt person for this, none other than Dr. Manju, Professor and Head, Department of Biochemistry, and the Organizing Secretary, BMSECON2023, to deliver her vote of thanks.
Thank you, Dr. Frank. Good evening to one and all. As the Organizing Secretary, on behalf of the organizing team, it's my privilege and pleasure to propose the vote of thanks today. First and foremost, I wholeheartedly thank our Honorable Chancellor Dr. A.S. Ganeshan Sir and Madam Chancellor Dr. Anuradha Ganeshan, President of VMRF, for giving us the opportunity and support to conduct this conference. Thank you, Sir and Madam. Now I would I would like to extend our gratitude to Mr. Suresh Samuel, Member Board of Management, for his encouragement and support. Thank you, sir. I would like to wholeheartedly thank our Honorable Vice Chancellor, Dr. P.K. Sudhir, who has graced the inaugural program and declared our conference open. Thank you, sir, for your encouragement. My heartfelt appreciation to our beloved Dean, Dr. Rakesh Sehgal, for his unconditional and overwhelming support and timely advices. Thank you, sir. Next, I would like to thank our Provost, Dr. P.F. Kotu, sir, who was like a mentor for BMSECON for the last 3 years and inspires us to achieve our best and serves as a pillar of strength. Thank you, sir, for joining even today to encourage us, even though you are traveling from Chennai. Thanks a lot, sir. Now, I would like to extend my heartfelt gratitude to our respected Dean, Health Professionals Education, Dr. V.N. Mahalakshmi, ma'am, who has supported us with her timely advices. Thanks a lot, ma'am. Now, I would like to thank our advisor, Medical Research and Publications, Dr. B. Vishnubhat, sir, who has always been an integral part of BMSECON since its inception. Let it be chairing a session, judging, panelist in a panel discussion. He's everywhere. His help in getting the abstracts published in the journal is worth a big applause. Thanks a lot, sir. Next, I would like to extend our sincere gratitude to the Medical Superintendent, Dr. Satish Kora Kuruvilla, sir, Dr. Jay Singh, Director of Hospital Growth and Outreach Services, Vice Principal Admin, Dr. T. Rajan, sir, Vice Principal Student Affairs, Dr. Lakshmi Jatia, Vice Principal Academics, Dr. Lata, Deputy Medical Superintendents, Dr. Arunachalam and Dr. Shankar, Deans of the other medical colleges under VMRF, Dr. Deepthi Shastri ma'am, especially who is attending our function now, head of the institutions of the other colleges inside the AVMC campus like BPT College, Nursing College, Allied Health Sciences, and Rehabilitation College, university officials, members of the STB, HODs, faculty members, and students of AVMC. Next, I would like to thank our TNMC observer, Dr. Prasad, Professor Community Medicine, for his support and cooperation with the whole team for the whole 3 days of the conference. Thank you, sir. Our conference was appreciated for its scientific talks, mainly the topics and afternoon session presentations, and the whole credit goes to our outspoken, eloquent, and knowledgeable resource persons, chairpersons, and the judges. All of them were so humble and cooperative that all of us speakers, chairpersons, and judges, they logged in before their allotted time, which has helped us conduct this conference smoothly and on time. The whole organizing team is grateful to all of you, sir. Thank you. This conference wouldn't have been a great success without our delegates. We thank all the delegates for your overwhelming enthusiasm in attending the session as well as presenting your scientific papers. We received a lot of messages stating that they learned a lot during the paper presentations. Thanks to the judges. We had around 250 abstracts in various categories and we were in need of many Zoom links. I take this opportunity to extend my heartfelt special gratitude to a few people who were kind enough to help us by sharing their official Zoom links for this conference for 3 days. I thank the Dean of Vinayaka Missions, Kribananda Warrior Medical College, Salem, Dr. Elvendan sir, Deputy Dean, Dr. Deepthi Shastri ma'am, Deputy Registrar of VMRF, Dr. Rajshekharan sir, our own Dean, Dr. Rakesh Segal sir, Dean Health Professionals Education, Dr. V.N. Mahalakshmi ma'am, and the Principal of Vinayaka Missions College of Nursing, AVMC campus, Dr. Sharda Ramesh ma'am. We really owe you. Thank you all for the warm gesture, for lending us or sharing us your Zoom links for the whole 3 days. Thanks a lot. I wholeheartedly thank our sponsors, JP Publications, Biodiagnostics, Volta Kluwer Publications, and Elsevier for their financial support. I thank our virtual platform event management team, meetings and more, whom we have been in. They are our become our official event management team for the last all the 4 years. They were our event management team. This year too, they have done a wonderful job. They were always there with us in minute-to-minute planning and execution of the program since 2 months. Thanks a lot, Pankaj sir, Zia, Vandana, Rohit, and all others whose names I am missing. But I thank each and everyone in your team. Now I would like to thank our informatics department, Mrs. Vijay Chitra, Sam and Bala, and others also who has designed the website, the BMSECON videos, the advertisements in the social media, and all the mails that you have been receiving during registration and other times. Thanks a lot. I thank our publishing and designing division, Mr. George and Kavi, for our brochures, conference proceedings, virtual backdrops, banners, etc. Thank you. Next, the whole organizing team thanks our ADP section who were there with us all the 3 days and before, full time with us. Before we reach the venue, they will be here and they leave only after we leave the venue. The high bandwidth internet required for the conduct of the around 10 parallel sessions and morning sessions without any technical glitch. The whole credit goes to our efficient EDP team. Thank you, Mr. Manigandan, Sridhar, Raghupati, Rajan. Thanks a lot. A special thanks to our accounts department, Mr. Ravichandran and team, who have cooperated with us in managing the registration finances. I thank all the supporting staff of all the administrative officers of AVMC, the Annai caterers and Amut canteen, Mr. John specially for the delicious food. Here I should make a special thanks to Dr. R.R. Kumar, Assistant Professor, Department of Biochemistry, who has done a wonderful job in arranging and providing all of us the nice delicious food for 3 days. I thank the audiovisual team led by Sundaraman and team, electrical department, Dhananjayan, who has given us always backup, Dhananjayan and Vijay Kumar, housekeeping and hospitality team with Muthu and team, staff of the academic section, maintenance department for their support. Last but not the least, our vibrant organizing team. Each one of us have contributed and worked hand in hand for the success of this conference. I can proudly say that the real success of our team was unity, a lot of tolerance, and a commitment to the work allotted. I wholeheartedly thank each and every member of my team, especially the organizing chairpersons, Dr. Elangatha, Dr. Gopinath, Dr. Ganesh sir from BMR department, who have done a wonderful job for bringing out the e-souvenir, Dr. Frank Umarnath, who was managing finance and scientific team and many other teams. I'm not taking the names. Hema managing the registration, Saumya, lot others. So I thank each one of us from my team for cooperating and making this a huge success. Thank you one and all. And a very small announcement before I just leave. This is for the delegates who are attending. Delegates, you can download your participation certificates from the link that will be shared to your mail in the WhatsApp group in phases. We'll start from today evening, but it will take in a couple of days. Everybody will be getting your participation certificates. Don't be in a hurry. It will be coming in a phasic manner. Everybody will be receiving in a couple of days. The paper presentation and the best paper certificates will be dispersed next week. Please give us little time for that. And please don't exit the WhatsApp group till we share the list of delegates who have applied for TNMC registration. Next is the e-souvenir which we have made, the conference proceedings. The link also will be shared with all the delegates within 2 to 3 days. So with this, I end and I thank once again everybody for making this 4th International E-Conference BMSECON2023 a huge success. Thank you one and all.
Thank you, ma'am. We have come to the end of the sessions. Now I request everyone to rise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you all for the 4th International BMSE Conference. Let's meet on the 5th International E-Conference. Thank you all. Vandana madam, we can end the session. Thank you.